चलना। Good morning, everyone. I, Lakshmi Narendra Kumar, feel privileged to welcome you all on behalf of the organizing committee for the hybrid event of 2 days conference on antimicrobial resistance in human microbiome and the 2 days workshop on next generation sequencing and data analysis jointly organized by the DBT Translational Health Science and Technology Institute and the NIH National Institute of Allergy and Infectious Diseases under the aegis of the Indo-US Vaccine Action Program. May I request all of you to please keep your cellular devices on silent mode, and for those joining online, to please put yourself on mute as we will be starting the program now. To begin the event, I request our distinguished dignitaries to kindly come up onto the stage. First, I would like to welcome our Executive Director, Dr. Jayanta Bhattacharya. Sir, please. I request Dr. Arvind Sahu, the Executive Director, Regional Center for Biotechnology, to the stage. Welcome, sir. I also request Dr. Shinjini Bhatnagar, Distinguished Professor, THSTI, to come to the stage. Ma'am, please. Next, I request Dr. Liliana Brown, Director, Office of Genomics and Advanced Technologies, National Institute of Allergy and Infectious Diseases, to kindly join us on the stage. Welcome, ma'am. I also request Dr. Leah Vincent, Program Officer for STI's Basic Research, to kindly come up on the stage. Welcome, ma'am. To begin, we will have our traditional lamp lighting ceremony. I invite our distinguished dignitaries on the stage to kindly light the lamp. Light is a symbol of brightness and prosperity. Let us begin this event by seeking the blessings of Goddess Saraswati by lighting the lamp of knowledge and wisdom, along with chanting of Saraswati Vandana by Ms. Shruti Lal and Ms. Priyanka Kumar.
माँ शारदे वरदान दो, हमको विमल शुभ ज्ञान दो। माँ शारदे वरदान दो, हमको विमल शुभ ज्ञान दो। हे माँ हमें निज भक्ति दो, सुख बांटने की शक्ति दो। गुरु चरण में अनुरक्ति हो, ऐसा विमल शुभ ज्ञान दो। माँ शारदे वरदान दो, हमको विमल शुभ ज्ञान दो। माँ शारदे सब अन्य बंधन दूर हो, नेत्रा अविद्या चूर हो, सुख शांति से भरपूर हो, ऐसा सुरम्य संसार दो। माँ शारदे वरदान दो, हमको विमल शुभ ज्ञान दो। माँ शारदे।
Thank you, sir. Thank you, ma'am. Now, I call upon Dr. Shinjini Bhatnagar, Distinguished Professor, THSTI, for the welcome address.
Good morning to everyone in the auditorium and those who've joined online. A warm welcome and a very special welcome to all our guests who've traveled from different parts of the world, the country, to be here today. Antimicrobial resistance has been identified as a global challenge and is an area of priority for research and finding solutions by the Department of Biotechnology, Government of India, the Indo-US vaccine program, which is led by NIAID at NIH USA. It's a heterogeneous problem, complex, and because of that, it's absolutely critical to have identify multidisciplinary research platforms to find solutions for this complex problem. This conference and workshop is a step in that direction. It's an opportunity for us to share knowledge, to be able to share experiences, and hopefully by the end of this meeting and workshop, we have developed some very important collaborations. A lot of background work has been done by THSTI in collaboration with the NIAID faculty for setting up this workshop. And especially for our younger colleagues, we hope you imbibe the knowledge from our domain experts, share your experiences, and really come out, let's say, learn it from this whole experience. So with that, a warm welcome again, and all of you and to all the guests. We hope that it's a successful workshop and meeting. Thank you.
Thank you, ma'am. Next, I call upon Dr. Shushmita Chowdhury, Associate Professor at THSTI, for initiating the proceedings with the objectives and expected outcome of the conference and workshop. Ma'am, please.
Thank you, Lakshmi. Good morning, all of you, and to all the delegates and dignitaries. THSTI, with the help of DBT in collaboration with NIAID, has set up this conference, which is a 2-day conference on AMR and human microbiome, and 2-day workshop on next generation sequencing and data analysis. So I'll just briefly, give a preview of what we are trying to do and what you can expect out of this. So basically, AMR is, as Dr. Shindini rightly said, it's a multidimensional problem. So we have tried to carefully curate sessions here on the first 2 days with talks on different aspects of AMR. Though as a drawback, I'll accept that we couldn't touch upon all the aspects of One Health, but we have tried to, include the basic biology, discovery biology, the clinical aspects, and the data analysis aspect, which is coming up as the future of any disease management. So if you go through the latest agenda, which you might find in your abstract books, you will find that every one of you, whoever from different domains, have something of your interest. And also there is scope for a lot of cross-domain conversations. So on 17th and 18th, the workshop that we are organizing is for a selected smaller group of people. So the THSTI faculty is going to mentor for the hands-on training on NGS platforms, and the NIAID, especially faculty from BVBRC, the Bacterial and Viral Bioinformatic Resource Center, faculty from there are going to handle the data analysis part. So I hope all of you have a great time, and all of you find productive learnings from this experience. Thank you.
Thank you, ma'am, for elaborating on the importance and impact of the topic. Now, I request Dr. Jayanta Bhattacharya, Executive Director, THSTI, to deliver the opening remarks. Sir, please.
Good morning. Good morning. It is with great pleasure that I welcome you all to this very important conference in the area of antimicrobial resistance and human microbiome. As a scientist, I believe this is the right time. There could not be a better time that we actually gather to discuss and deliberate on what's the problem, how this evolving problem is actually making a lot of challenges to the human health at large. You know, I think, again, we have learned during the COVID times, I think COVID has helped us learn that whether it's a pandemic or an epidemic, I think antimicrobial resistance is an epidemic in nature, I think you all agree with me. There are 4 things that I think we have been hearing and we have learned that are pretty important to address and understand if we really want to tackle the antimicrobial resistance, for example. One is, of course, in the area of surveillance, and we really need to have a strong surveillance in place. And surveillance doesn't necessarily include just doing a simple epidemiological studies, just to observe, do observation studies. In recent times, we also have understood how pathogen genomic sequences can also help us identify pathogens which otherwise in a conventional method you cannot actually identify. And what does it lead to? It actually leads to identifying pathogens, understanding mechanisms by which you could actually diagnose those pathogens which otherwise we are not able to identify. So you actually have another component you add to that. We have learned during the COVID time with diagnostics. Diagnostics plays a very important role. I think I hope that in the next 2 days, we'll be also going to hear about how diagnostics plays a crucial role in identifying and delineating mechanisms by which you could actually identify pathogens, which are very difficult to identify. I mean, we THSTI, a lot of work is also going on in this aspect. And the third thing, once we actually have a good surveillance system, we understand what is causing the antimicrobial resistance and what is their mechanism of action. And then by diagnostic tools, you identify those critical pathogens. You would actually land to a situation where you can now think about how to mitigate that problem by discovering or understanding how to, new chemical entities. And today, we are also hearing about other modalities like monoclonal antibodies. And of course, there are areas where vaccines play an important role. So overall, I think that's what we have learned. And I think when I actually saw the program, I figured out that all of these aspects are going to be touched upon. And what is best that we have in the next 2 days, we're going to have the critical minds, all the critical minds in all these different domains are going to deliberate and discuss all these things. And that's something I believe is going to be an important takeaway from this conference. One of the things that is excellent, which the Bhaktos and the team has included is a hands-on workshop in the area of next generation sequencing. And that's something I hope is going to add value to this whole conference. Finally, I just want to thank the Department of Biotechnology. Department of Biotechnology has been playing an important, very, very important role in helping us conducting this kind of workshop. And of course, I also want to thank the NIAID team, the National Institute of Health, in actually joining hands with the Department of Biotechnology and THSTI in organizing this conference. And I'm sure in next 2 days, we'll be able to take away some of the very important aspects of information, which I'm sure will not only help us understand where the field is going, but also will help us understand who we should be collaborating with, how to complement each other, what are the gaps, and how do we actually mitigate those gaps. So I'll stop here. Warm welcome to all of you. And I wish the conference all the very best. Thank you very much.
Thank you, sir. Next, I request Dr. Liliana Brown, Director, Office of Genomic and Advanced Technologies, to say a few words about the conference. Ma'am, please.
Thank you and good morning. Want to thank our colleagues here at HTI for hosting us and really organizing this meeting together with us over the last 6 months or so, spending what I think is very precious times in the evenings calling into NIAID calls to organize the meeting. So we're very, very thankful for that time. I also want to thank the Department of Biotechnology for their support and continued engagement with NIAID through our almost 40 years of history of working together under the Indo-VAP program to advance understanding of infectious diseases and really trying to develop new vaccines. We're very excited for having this conference. We're very excited to have guests from the United States and guests from across all of India to address the issues of antimicrobial resistance, and of course, to see how we can advance continued research into the human microbiome as alternative mechanisms to treat and address antimicrobial resistance. So I think you've seen and we've heard from the different speakers this morning already that we have a very exciting agenda. We've tried to select speakers and people who would be really able to engage in collaborations moving forward after this conference. So it is my hope that there will be continued work, that people will get together, get to know each other, and build those projects moving forward that will continue to give us more understanding of how to address antimicrobial resistance. And I hope that through the posters, because we have a lot of different posters, will be some of us working and looking at your posters and engaging in conversations that way. But I also invite all of the scientists who are presenters to also go and walk through the posters and really try to discuss future collaborations in that way. Last, I want to thank our NIAID staff who works here in India for also all of their support and really helping us work through getting this conference together. Thank you for having us. Thank you for such a wonderful breakfast, and I hope delicious lunches that are coming. Thank you. I really hope we have a wonderful conference. See you around. Thank you.
Thank you, ma'am. With this, we are concluding our inaugural session. I request the dignitaries on the dais to kindly take their seats in the audience. Thank you, sir. Thank you, ma'am. We now begin our first scientific session of today on the theme, AMR heterogeneity in pathogenic and commensal microbes, genomic and metagenomic insights. I humbly call upon the session chairs, Dr. Shanta Datta, Dr. Jayanta Bhattacharya, and Dr. Anil K. Pandey to the stage. Sir, please. Dr. Jayanta, sir, and Dr. Anil K. Pandey. And Dr. Shanta Datta, she'll be joining us shortly. Dr. Jayanta Bhattacharya is the current Executive Director of THSTI. Prior to that, he was the Deputy Director and Head of the Division of Molecular Virology at the ICMR, National AIDS Research Institute, Pune. His research is on understanding AIDS pathogenesis with focus on how circulating HIV diversity can impact antibody-mediated prophylaxis and therapy. Thank you, sir, for coming over to grace the event. Dr. Anil Kumar Pandey is the Medical Superintendent and Professor at the Department of Physiology of the ESIC Medical College and Hospital, Faridabad.
Dr. Pandey has several collaborations with various research groups in THSTI and has been sentinel in many COVID-related projects. Thank you, sir, for your valuable presence here today. So in this session, each talk will be of 20 minutes. After the talk of all the speakers, there will be a 10-minute long question and answer session. I request the audience to kindly reserve their questions till the end of the session. So I now request the session chairs to introduce our first speaker and call them over for the talk.
Our first speaker is Dr. Ashley Earle from Broad Institute, United States. Dr. Earle is a research scientist and the director of the Bacterial Genomics Group at the Broad Institute at MIT and Harvard. She was awarded PhD in microbiology from the Louisiana State University and received a postdoctoral training from Harvard Medical School. Dr. Earle is working to understand the relationship between microbes and human health, including how multi-drug resistant pathogens emerge and spread. She leads a team of computational biologists to develop and utilize an array of omics analytical approaches to dissect bacterial and host contributions to several infectious diseases. She has brought in genomic approaches to the study of tuberculosis, urinary tract infections, and hospital-acquired infections caused by the enterococci and carbapenem-resistant enterobacteriaceae. So the title of her talk is Genomic Insights into the Global Spread of Antibiotic Resistance. So may I request Dr. Ashley Earle.
Thank you so much. First, I just want to continue the thanks to all of the organizers and especially to our hosts here. It's already been just a remarkable time, and I genuinely look forward to learning more about all of you, your science, of course, your culture, and I would be honored if we found ways to collaborate and continue the conversation after all of us go home. Okay, let me see if I can get my talk. Okay, here we go. All right, so I'm delighted to be telling you today about some of the work that we've been doing in the group using genomics and metagenomics technologies to understand the rise and spread of antibiotic resistance, which we've already heard today, I think very eloquently put, is degrading our ability to defend against even some of the most common bacterial infections. And obviously this is a big problem. And so I just want to start just reminding us that it's not just viruses that spread in our interconnected world, but we know that bacteria can too, and so can the genes that drive their resistance. So here we're seeing what happened in 2008 with the first discovery of the indium-1 gene known to confer resistance to carbapenems. It was identified in a patient from India being treated in Sweden. And what I hope you can see is that in just a very few short years, it went from that first discovery to basically following all over the world. We were finding this particular type of resistance. And so it's nice to have this. We can see, this was an alarming finding. A lot of retrospective work went into sort of creating this view. But the truth is, we know very little about how microbes spread around the planet, right? We know very little about how microbes spread in this very room. And so I think it's clear that without a good understanding of what's going on, what is the prevalence of resistance out there, how is it moving around, we're hard pressed to really come up with ways to intervene in it. Because clearly, I think this is, we would all agree, part of the problem, part of the challenge. So it was a delight to be invited to be part of a collaboration to effectively put travelers out there into the world and use them as kind of like a test tube to go out and see what they encountered during their travels, what they collected, and what they brought back. So these were international travelers from the United States going out into the world, and whether they liked it or not, whether any of us like it or not, as we're out there, we're interacting with the beautiful, microbial world that we live in, including those that live in other humans, those that live in the environment, those that live in animals that we interact with either directly or indirectly through food. So we thought this was like a really interesting way to kind of get that surveillance lens that we don't necessarily always have, especially since we focus so much on hospitals. And I'll just introduce our wonderful colleagues, Regina LaRocque and Ed Ryan here. These are fantastic physician scientists who, among many things in their great careers, have over the years worked hard with CDC in the United States to put together this travel clinic network called Global TraviEpiNet. So it's travel clinics across the country. This is where people come before they travel to get their vaccinations. We can help travelers learn more about how to stay safe. And it was from this travel network that we began our recruitment of travelers who allowed us to interrogate them about all kinds of things that were going on before they traveled, during their travel, and upon their return. And they also allowed us to request, and they gave us samples of their poop. And so this poop went through 2 different journeys so that we could look at what was going on. One was to the clinical labs at the hospital where Regina and Ed work, where the stool was put onto selective media for 3 different antibiotics, so that we're looking for organisms that are resistant to beta-lactam. So there are extended spectrum beta-lactamase producers, our ESBLs. We're looking for carbapenem-resistant Enterobacteriales, and we're also looking for those that contain the MCR gene, which encodes for colistin. And of course, once we sort of understand who's coming back with something they didn't leave with, we can get the organisms that grew up in the lab, we can sequence their genomes, and use this as a way to sort of do what we consider to be a kind of a targeted surveillance of what's out there in the world and coming back. And then separately, the samples go to the Broad, where we extract DNA in total from these samples, and then we sequence the whole gamish, so we can get sort of this non-culture based, less biased view of what's leaving and what's coming back. And so we consider this to be a bit of like an untargeted surveillance strategy. And it also gives us this really interesting opportunity to think about the ways in which the gut microbiome may be either promoting or inhibiting the process of acquiring these organisms. Okay, so that's the setup. And of course, we're creating lots of samples and lots of data sets, and we can ask a lot of questions with it. So I'm just going to take you through some of the questions, and let me check. I heard 20 minutes. I had prepared 25, and I just want to make sure I understand. But you guys, you know what? There's nothing more interesting at the end than at the beginning. So if we don't get all the way, it's okay. We can talk over tea and I can tell you the rest of this. So just give me the, Okay. So one of the questions, of course, is just how often are these travelers going out and coming back with something they didn't leave with? And so the answer is, I'll just cut to the chase, 40%. 40% of our over 600 travelers came back with one of these target AMR organisms. And I'll just tell you here that when this happened, one, we let the traveler know. We also, and we enrolled them, we attempted to enroll them for a year. So we asked them to submit samples for every 3 months for a year. We also asked if they would help us enroll their household contacts to look for potential onward spread of the organism they had acquired. And we also let the state departments of health know as well. So just in case something went wrong, everybody's on the same page. 0 of our travelers who came back with one of these organisms had any problem whatsoever. So that sounds great, right? That's great. And yet, I think for me and for all of us, it sort of underscores just how under the radar all of this movement and transmission and trafficking of antibiotic resistance really is. There's a ton of it, and we don't have a clue. Okay, and of course, where you go matters in the world. So here we're looking at the number of travelers coming back with MCR. So this is the colistin resistance. So we can see that depending where you go, you get different levels. Oh, great. Okay, so the travel regions that are, have the black outline are those where we had at least 10 travelers going. And you can see it's 12% of our travelers to Southeast Asia came back with MCR, and often none of our travelers to other parts of the world came back. And if you look at something like ESBL, it's a different story. So as many as 50% of our travelers to South Asia came back with ESBL, and as little as 10% came back with it in other regions of the world. Okay, so that's part of the story, but what do the genomes tell us? So that gives us a sense of what kind of resistance may be out there in the world. But what can we learn from studying the genetics? And so here I want to introduce Colin Warby and Sushmita Sridhar on the team, who are really impressive scientists who are responsible for the vast majority of everything I'm going to share from this point on. So one of the things the genomes told us is that the 95% of the organisms that grew up in the laboratory were E. coli. So that's one. And when we looked at the relationships between the E. coli that came back, using our phylogenetic techniques, which I'm showing you the result of. So here's just a tree. The tips obviously represent each of the little isolates. And I'll tell you, if you're not kind of an E. coli nerd like I am, these big sort of larger clusters on the tree represent a lot of the diversity that we understand within the species. So, all these different color clusters tell us that people are coming back with basically the diversity of E. coli that we understand in the world. So lots of diversity coming back in these people. And when we ask whether there's any connection between where the travelers went and the kind of E. coli they came back with, the short answer was no. There really was no pattern whatsoever. And this jives with other kinds of other research and meta-analyses that really suggest E. coli and all the different types are kind of everywhere. Okay, but what was super cool is that sometimes we saw some interesting patterns that suggested some regionality. So we saw examples like this one. So here now we've got two independent travelers to Peru. Their E. coli sit on very distinct parts of the E. coli tree of life. But when we looked at their plasmids, so these are these extra chromosomal elements that are absolutely at the heart of AMR for bacteria, for a lot of the bacteria that are really important in the clinic, we see that these plasmids are nearly base by base identical. So, and they encode for the MCR gene here, this little red dot. And at the time when we first discovered this, Colin looked in the databases, and the only other example of this plasmid happened to be in the southern neighbor, Bolivia. And recently, as I was preparing for this talk, he checked again, and the only other sighting of this plasmid was in the northern neighbor, Ecuador. So for reasons we can't fully explain, but it's fun to speculate, and I love to chat about it, this plasmid seems to have a sense of place. We don't really see it anywhere else. So I think that's really interesting. And we also see examples like this where, it's not just looking at plasmids, we can also look at the variants within the resistance genes. So just like we looked at variants of SARS-CoV-2, we can look at the variants in these genes. And, again, the story, two different independent travelers to parts of West Africa, each came back with a variant of MCR1 that we had never seen before. Today, there's multiple sightings of this variant. It seems to now be showing up in patients in different parts of the world. And most of it is either contemporary with the sighting of this one or later. And for now, this variant is still the only one that we've seen. So it's kind of, I think, underscores the potential power. Is that my time? No, okay. The potential power of this approach to really, potentially tell us about emerging threats that are out there. Okay, so of course, everybody wants to know I get this AMR that I didn't leave with. How long do I carry it? So the answer is not terrible. So many people lose their AMR organism by about that halfway mark in the year. So we've got about a 40% probability of continuing to carry your AMR organism by about 6 months after return. And by about a year, we see about a 20% probability. So many people lose them, but for some, they stick around. Okay, so we all want to know, are there things that we can do, of course, as travelers to protect ourselves against acquiring these organisms? And it's a mixed answer here, but what we did was a risk analysis, and what Colin showed was basically what we already knew. We didn't really need this study to tell us that certain regions of the world put you at higher risk for acquiring certain resistances. So I'd already showed you the maps before. We saw what I think had been appreciated before was visiting certain behaviors during travel, visiting family, friends also puts you in increased risk, as well as having diarrhea during your travel, and particularly having diarrhea that you treat with antibiotics. So that seems to be a risk factor. It's been known before, we see it here. But here we have an opportunity to actually look at the microbiome. And I think the hypothesis was, what's likely behind this is that, like when patients in the hospital are treated with antibiotics, their gut microbiota gets messed up, right? You get a breakdown of this colonization resistance that we think is one of the functions of our gut microbes that allow things to come in. And sure enough, when we looked at our data, that's what it told us, or at least supported. So here I'm showing you for each of our samples. This is from travelers who had diarrhea during travel. So each circle is a sample. These had diarrhea plus antibiotics. We can see that many of them, statistically significant fraction of them, had less diversity, so lower on this y-axis post-travel than pre-travel. And of course, we see it even more for the antibiotic treatment. So that seems to support this idea. What we didn't expect, but what we saw is even for travelers who didn't have diarrhea, we also saw a statistically significant drop in diversity, which was interesting. Now, from a risk perspective, when we run the analyses and ask if it matters for AMR, it doesn't seem to matter. But I think this is an interesting area and more to develop here. Okay, for time, I'm just going to tease, if you're interested in hearing about risks associated with acquisition of specific genes like resistance genes and animal contact, let's talk. If you're interested in animal contact and risk of picking up virulence genes, let's talk, but I'll skip it for now. Okay, so another thing that we were very keen to look into with our collaborators was whether the pre-travel gut microbiome could it have a protective effect on whether or not you acquire an organism. And so here's the answer. It's no. So what Colin has done here is each of the travelers is a column. All the different colors represent different bacterial families that we could predict from the microbiomes. And here the Y is just showing the relative abundance of each of these portions of the bar. So here are the travelers that didn't acquire. Here are the travelers that did acquire. Trust your eyes. There's really no difference here. And we've looked at it in all the different taxonomic levels, no difference. We've looked at it with diversity, no difference. So it seems that there really isn't something you can do to sort of prepare your gut prior to travel. That doesn't seem to be part of the story. Okay, what happens? How am I doing with time? 5 minutes? All right, please, you can cut me off. So what happens when we sort of widen, our view of things and we go beyond the culture, and now let's just let the metagenome do the talking? What do we see? Does it change our perspective at all on what's happening? And the answer is yes, it did. In fact, what we saw was that there was indeed widespread acquisition of resistance genes. And just to go quickly, so each of these circles represents an antibiotic resistant gene or an ARG. And here we're just seeing the proportion of travelers that lost that ARG after they've come back and the proportions that acquired. And just to cut to the chase, I think this little effervescent sort of, bubbling up along this y-axis here of these genes that I'm circling really tell the story. There are a ton of genes that are coming in and coming home with these individuals. And many of them are, we predict are leading to resistance to things beyond what we screened for in the lab, including for fluoroquinolone resistance, aminoglycoside resistance. And we also see things that we consider high risk based on their mobilization and pathogen association. Okay, so what's bringing these things in? So that was a big question. As we did some analysis, I'm going to skip the slide on it, but bottom line is it's our friend E. coli. What we saw was that what seemed to associate best with the increase in these genes was an increase in the relative abundance of E. coli and its cousins like Klebsiella, Enterobacter, Citrobacter. And in fact, what was so fascinating is when we now take into account the rise in relative abundance levels in the guts of these travelers post-travel of the Enterobacteriaceae, so the family of E. coli klebs, we see across, it doesn't matter where you go. Everybody's basically coming back with more of these organisms. And so one of the questions we had, of course, was, is it the organism you left with that's now growing in abundance, or are you picking up new things? And so I'll just flash this for a second. We developed a tool and published it last year that allows us to really look at the high resolution at strains at very low abundance in complex community samples. So we developed this for recurrent UTI studies. So if there are folks in the crowd who are interested in that kind of research, I'd love to chat about that as well. But we applied it here, and it was a great tool because it gave us this interesting perspective. So here it's just a rolled up view of the analysis where we pointed strains at all of our stool metagenomes, and we just asked, what kind of strains are in these metagenomes prior to travel? So here I'm showing the fraction of travelers that had certain types of E. coli. So again, here's the big, very diverse clusters. We saw the things we expected, a lot of, good proportion of B2 and D, sort of the Western gut kind of E. coli. So again, this is pre-travel, but look what comes back. Totally different, right? Like these are different E. coli. And so I think what we're sort of bringing it all together, where we're coming to in terms of like thinking about the risk of AMR, it's really the coming together of two probabilities. It's the probability of going out into the world and encountering an E. coli, and the probability that that E. coli is going to be resistant to a certain drug. And we think that, of course, is really dictated by the prevalence in the community. Because you can see, like, sometimes the fraction of travelers acquiring an organism in a particular region agrees really well with the fraction that comes back with new E. coli, but a lot of times it doesn't. So it really seems to be like that. Okay, so what? Here's my so what's. I'll tell you more about those later. Here's my partners and funders who I just bring a lot of joy to the work that we do together. I want to mention Sushmita again, and my team of just wonderful people that make life productive and a lot of fun. Okay, with that, if I have, I guess, questions later at the end. So thank you.
Good morning. Now I would like to invite Dr. Bhabatosh Das, who is the Associate Professor of THTSI. Dr. Das is a molecular microbiologist and Associate Professor at Translational Health Science and Technology, Faridabad. He obtained his PhD degree from Indian Institute of Chemical Biology, Kolkata, and received postdoctoral training from Institute of Integrative Biology of the Cell, France, and Osaka University, Japan. Dr. Das's research focuses on the ecological principles of the human microbiome and their role in health and diseases, the emergence and spread of multi-drug resistant bacterial pathogens, and the vaginal microbiome of Indian women, and its importance in birth outcomes. Dr. Das is a recipient of the prestigious National Bioscience Award and serves on the editorial board of many high-impact journals. He will be speaking on genomic insights into AMR heterogeneity of bacterial pathogens for drug discovery. Dr. Das, please.
Thank you, ma'am, for your so nice introduction. And I'd like to thank all the participants for coming this beautiful campus and joining us in this conference. So I'm Bhabatosh, and I'm working in this Translational Health Science and Technology Institute in almost last 12 years. And in my labs, we have the two major research focus. So the first part, we are working on the human microbiome to understand the role of human microbiome in health and disease. And the second part, we have a large group who are interested to explore the genomics and genetics of antimicrobial resistance, mostly on the critical priority and high priority bacterial pathogens. So today I'm going to discuss only these genomics and genetics of antimicrobial resistance in the critical priority and high priority bacterial pathogens. So I don't think we need to introduce the audience why antimicrobial resistance is a global concern. We know that almost like each 3 minutes, one child die because of the multi-drug resistant associated infections. And if you look in 2019, almost 4.5 million died only because of the resistant infections. And the way the resistance is going on, if it's not tackled properly, by 2050, almost 10 million people will die because of the antimicrobial resistance. And this resistance is not restricted only to the bacteria. You could find that resistance is widespread, including fungi, parasites, and also the viruses. And based on their impact on the health sectors, now WHO categorized them in the different groups. So we are mostly interested on the critical priority groups and the high priority bacterial pathogens. So when we start our works on the antimicrobial resistance, the first questions we try to understand how fast this antibiotic resistance can emerge. So once you introduce an antibiotic to the clinical practice, so how long it will take to develop that resistance. So we started working in silico. We analyzed almost like 5,600 articles that time it was available. And what we observed that once we introduce an antibiotic, like within 2 years of times, all the times the resistance evolved. And in some of the antibiotic, even before introducing into the clinical practice, resistance been already reported. So that means that antibiotic resistance is already present into the environment. Once you are giving the antibiotic, you are giving some selective advantage to the resistant variant, and they are basically parasites. Now, we know that resistance is evolving very fast. So definitely second questions that comes in our mind, what's the functions that confirm the antimicrobial resistance in the microbes? We are focusing mostly on the bacteria, and what we observe that there are mostly 3 different mechanisms that help this bacteria to reduce the efficacy of antibiotics. Simply, this bacteria can change their target molecules either by acquisitions of some mutations or some functions that can transfer a chemical groups or by excess production. But if you look to the gram-negative escape pathogen and the bacteria that resides in the microbially crowded environment, so the acquisitions of enzymes that modify the antibiotic is the most predominant. Either they degrade the antibiotics or they modify the antibiotics. And the third mechanism that bacteria use, they reduce the effective concentrations of antibiotics simply by increasing the efflux pump activity or decreasing the permeability. So this is the way basically antibiotic resistance bacteria reduce the efficacy of antibiotic. And if you know that this is the resistance, so the second and the third questions we had in our mind to understand how heterogeneous are the antimicrobial resistance function in the different pathogens. Do you have any pathogen specific signatures? So we started working here that we have made a good collaboration with almost like 12 different hospitals across the country. And we collected more than 6,000 bacterial isolates. And those isolates are mostly collected from the sepsis, diarrhea, gastrointestinal tract infected people, UT infection, respiratory infection, and the skin infections. And the bugs that till now we characterize, they are mostly the high priority and critical priority bacterial pathogen. In addition, what we did, we selected some bugs, particularly the diarrheal pathogen, where antibiotic is not the preferred way to cure the infections, like diarrheal pathogen, Vibrio cholerae. If you look, the people use mostly the ORS to treat the cholera. They don't use antibiotic, but still you'll find that the pathogens is basically have huge number of antimicrobial resistant genes. That means that it's not only the use of antibiotic into the health sectors that drive the infections, that drive the emergence of resistance, but antibiotic that present in the environment could also play very important roles. We started this work. So Pawan is one of my PhD students who initially started this working on this antimicrobial resistance, and first he published this work on the scientific report. So we analyzed the genomes of the gram-negative escape pathogens, and then later on we also did Pseudomonas aeruginosa and Enterobacter Klebsiella pneumoniae to understand what are the genetic traits that confirm the antibiotic resistance. So what we observe that if you look chronologically, this antibiotic resistance is basically evolving very fast. The isolate that we had from the 2019, the average number of resistance was less than 5, but 2015, you can find that it's then close to the 10. And depending on the bacteria, you could find that some of the bacteria, particularly the Klebsiella pneumoniae, they have a huge amount of resistance reservoirs compared to the Vibrio hermeticus. We are continuing this study, and if you look to 2023, you could find that the average number of resistance is coming close to 15. So it's almost each and every antibiotic that we use in the clinical practice, you could find that bacteria acquire this antibiotic resistance. Now, what we find that when we analyze the genome, that it's not only the resistance allele for a specific antibiotic, but you have the multiple allele against each antibiotic. Look at beta-lactamase. If you look one isolate of Klebsiella pneumoniae, they have almost 13 different beta-lactamase from all the 4 subclasses. It's true also for the aminoglycoside resistance, effects pumps everywhere. So they have the resistance functions, and the resistance allele are also quite heterogeneous. Now we ask another one question to understand whether the AMR genes have any pathogen-specific signatures. We have the multiple pathogens, although the functions are very common, but is there any pathogen-specific alleles? So the Tansi is another one of my PhD students. You can say the very intelligent PhD student who basically majorly done this work. So here what we did that we tried to understand the antimicrobial resistant heterogeneity in gram-negative pathogens. We started with isolating this bacteria, then did the whole genome sequencing. And once we predicted that sequence, we also validated for the proteomic analysis in the presence and absence of antibiotic, whether those resistant genes are functional or not. And what we find that yes, if you look at the different pathogens, each of the pathogen, they have a pathogen-specific allele. Just look to the beta-lactamase resistant. If you go from the Acinetobacter baumannii, this is mostly dominated by the BLA ADC and BLA oxa. You just go to the Klebsiella pneumoniae, Pseudomonas aeruginosa, it's BLA oxa and BLA pau. You go to the Klebsiella pneumoniae, BLA SSV and E. coli blandindium. This is also true for the aminoglycoside resistance, macrolide resistance. Resistance functions are common, but the allele, they confirm the antibiotic resistance. They are quite pathogen specific. Now, how actually the genome plasticity link with the emergence of extensive drug resistance bacterial pathogen. That is important. We know that this is the resistant genes that present in the bacteria and they make them resistant, but how the genome plasticity play this role. So here we did with Jyoti. So Jyoti was also my PhD student. She just completed her PhD and now joined TGST as a research scientist. So what she did very intelligently selected the bacterial isolate chronologically in the different periods of time. So we had a very good collections of Vibrio cholerae from 1960 to 2023, where we selected cholerae, those are totally sensitive, then resistance, multi-drug resistance, and pan-drug resistance. And when we analyze, again, we found that almost all the resistance in the Vibrio because of the aqua function, the enzymatic acquisitions, and each and every those acquisition functions are linked with the mobile genetic elements. That means you have a huge threat that once you have that resistance genes, the mobile genetic element, they can disseminate. And if it's acquired into the genome, so how long those antibiotic resistant genes stay in the bacterial genomes? Is antibiotic play an important role for the stability or the acquisition of the genes? That is the questions we also answer through this paper, how stable are these MGE linked with antimicrobial resistance? So this study is done by the Archana. Archana also completed her PhD from our labs and now she joined as inspired faculty in the NII. So what she has done, try to understand how efficiently these antibiotic resistance linked mobile element can integrate into the bacterial chromosomes and how stable this molecule in vitro and in vivo experimental settings. So what we observe that these antibiotic resistance linked mobile elements present across the genomes. In Vibrio, there are two major hotspots, PRFC that integrate the IS element and superintegron that basically capture the antimicrobial resistant genes. Other than that, you could find also that there are so much genomic islands or also IS and TN element that present across the chromosome. When we study the efficiency of integrations, what we observe that if you are giving only the integration modules of SXT that carry a lot of antibiotic resistant genes, they integrate very efficiently, almost like that with 0.1% efficiency they can integrate. But in terms of the excision efficiency, we did both in vitro and in vivo. We find that the in vivo, they are very stable, although in vitro, their excision efficiency is quite high. And then we did the study with Dr. Ramurthy, which also here. Now here we looked at the wild type condition, how those resistance allele basically differed with the periods of times. We had the isolate from the different part of India, in addition also from the Bangladesh and Thailand. And what we observe here that the resistance allele, some of them are very stable. Okay, if you look at the clompenical resistance allele, clompenical is never ever been used to treat the cholera. But this allele present in the microbial genome, that makes that a huge sense. It's not antibiotic that used in the health sector, antibiotic that used in the environment or present in the sewage. They do also play very important roles. So this is the another one study. We started working with Neisseria, Calcutta, in collaboration with Dr. Ashish Mukhopadhyay and Dr. Ramurthy. Here, what we did, we basically now collected these bugs from an endemic area. So each year we have collected this bacteria, then did their antibiotic resistance susceptibility, and we selected sensitive resistance, multi-resistance, extensive drug resistance, and contact resistance bacteria to understand whether it is only the acquisitions of new functions or is there any mutation within the allele and how these resistance alleles are stabilized. So when we analyze, we again observe that depending on when also the serotypes, the cholera have the more than 254 serotypes. What we observe that the O1 has the highest resistance capacity, and O139, non-139, their resistance profile is quite low. That make another sense. If you look to the structures of these cholera pathogens, O1, they don't have any capsules. But the environmental and non-1, non-1,3, they do have a capsules, and capsule is one of the barrier for the natural transformation. That's why even if they present in the environment, they might not be able to uptake the resistance allele. They're still sensitive. But O1, since they're naturally competent, they can uptake this DNA from the environment. They're getting huge resistance. So not only the pathogen, we also try to understand that the bacteria that reside in the human body, what is their resistance profile. So Soto was our technical officer. Now he joined the Black Biotech and he's heading that the R&D section. Aini was also doing postdoc in my labs. Now he had an independent assistant professor position in IIT Delhi. They basically isolated all those commensal bacteria from the gut and then their genome analysis. And what we observed that like the pathogen, commensal are equally resistant. And these resistance allele are also linked with monogenetic element. So that makes very interesting story. Whenever we take an antibiotic, does not matter you have a pathogen or not, the commensal is always there. So they also try to survive. That's why they acquire the resistance function. And when a pathogen comes to your body, even they're sensitive, what they can do, they can pick up those resistance allele from the commensals and they can disseminate. That exactly proved we experimentally. What we did, we isolated this DNA from the guts of the healthy individuals and then give those DNA to the naturally competent the bug. And what we find that within 36 hours of time, those bugs basically acquire the resistance. And when we did the genome sequencing of those bugs, what we found that some of the elements like ICE and TN, these bugs can acquire and they go and integrate into the chromosomes. So we have all those informations regarding the extracellular bacterial pathogens, and those are living in the microbially crowded environment, and they have the acquired function. But there are microbes that live in the harsh environment where the natural competent is not very efficient, that one of them are H. pylori. So we started working on the H. pylori genomics, again, the Tansi and the Sokti. Sokti also have his new labs in the HPI Lucknow. So they analyzed the genome of H. pylori. We analyzed almost 142 genome. And when we look to their genome, what we find that H. pylori resistance are only because of the acquisitions of mutation in their target genes. All the resistances happen only because of the mutation in the target gene, and we could not find any resistance functions. That might be the regions of the environment is primarily resides that is not favorable for the natural competence. We have genomic information. We know what types of allele confirm the resistance. We know the resistance crisis. Now, currently, lab is majorly focusing to develop some therapeutic against these resistance pathogens. What we are trying to do that whether it's possible to resensitize this resistance back, then the existing antibiotic may also work. So for that, you need an effective systems. So what we did that, what are the different way this bacteria can be resensitized. So we have a very talented group of the PhD student and postdoc. So Minal is doing PhD and she started working on the aminoglycoside resistance functions. Lakshmi is the MK1 fellow. She started working on the beta-lactamase potentiator, and the Dipojyoti started working on the macrolide resistance bacteria. So we work on the existing literatures, and then we try to understand what types of molecule can inhibit this resistance function. Since these resistance allele are enzymatic in nature, if you could find any molecules that inhibit that enzymes, then your antibiotic will work. So that is the way we are working. So this is the common mechanism of how potentiator work to resensitize. Sometimes they can also destabilize these plasmids, but in our lab, we are mostly focusing on to identify a molecule that can basically inhibit the resistance functions, and then we are giving the antibiotic tokens. First, we developed a reporter strain. So bacteria, you know that sometimes even they're not resistant, but they have some intracellular signaling molecule that can sense and reduce the sensitivity. So one of those molecule is the PPGP, and what we observe that it reduces the efficacy of antibiotics and also increase the effects from activity. So we develop a reporter strain where all the enzymes involved in the PPGP biosynthetic pathway we remove, and we cure the reporter strain, and we check that this box is highly permeable and their effects from activity is drastically reduced. Then we transport all these resistance functions from the pathogen to this reporter strain. So we transport it into the chromosomes with the native promoters or the PBAT, that is the individual promoter, and also the constitutive promoter, and check that whether these resistance allele are functional. If they are functional, then we started screening the compound. So this is the way we are working. So we introduce those resistance allele from all the bacteria to our reporter strain, that is in Vibrio and E. coli ATCC, and then we screen the library. So currently we have the two library. One is natural compound library from the CellChem, and second one from the MedChem. We grow these bugs in the presence of antibiotic. They grow very happily. And then we started giving that compound, and we try to identify any compound that basically inhibit their growth. So once you find that compound, that could be one of the three ideal potential outcomes. It could be an antibiotic potentiators. They inhibit the antibiotic resistance function. That's why the antibiotic works. It could be a synergistic compound, or it's a novel antimicrobial. So currently, we have few hits that actually inhibit the aminoglycoside resistance that Minal has found. The Dipojyoti has found lots of hits on the cloramphenicol resistance. Lakshmi found few hits against beta-lactamase, but what we observe that is not actually the desired compound, but most possibly some of the contaminant that give this potency possible one. With that, I just want to summarize my talk. What we observe that the antibiotic resistant genes are widely disseminated with clinically important bacterial pathogens. Mostly the extracellular gram-negative pathogen, this is acquired function for the horizontal gene transport. Resistance functions or dynamics can be inactivated by the natural or synthetic compounds. I'd like to thank all my collaborators, my funding agency, especially to the Department of Biotechnology for the generous funds. And also I'd like to thank all my lab members, and thanks for your attention. Genomic insight into AMR history of discovery. It's really very good to talk looking into this global challenge of resistance. Now I would like to invite Dr. Jonathan of University of Michigan, USA, For this talk, Dr. Gola is a physician scientist with more extensive clinical and research training. He received his PhD and MD from University of Washington. The journey started with a foundation in engineering encompassing computer science and biomedical engineering, and is an enthusiastic teacher of computational analysis of data. He is an infectious disease specialist, further specialized in the care of people with immune dysfunction, with cancer, post-transplant, and infection. He was involved in the University of Michigan response to COVID-19 pandemic, both providing some of the earliest direct patient care as an infectious disease specialist and clinical research, controlled observational studies, and randomized clinical trial for COVID-19 therapeutics. His talk is on the evolution of spread of antimicrobial resistance elements. Dr. Jonathan Gulab, please welcome to the session and your lecture. Thank you.
Thank you. I feel so welcome. This is my first time to India, and it's been an utterly delightful visit. And so I'm very appreciative of the opportunity. And I'm appreciative of the previous speakers in this session, because they have gone over much of the background that I would need to do. Perfect. I am going to make a provocative statement, what I think a provocative statement, that leaning into my computer science background, I think using a slightly different method of analyzing our data, particularly when we get into metagenomic data or 16S-based data, might yield us a little bit more information about specifically antibiotic resistance. I'm going to go through and start with a brief example of some data and work done by one of my colleagues that was an inciting insight for me about this exact question. And here I'm going to start on talking about a slightly different approach, one that Dr. Earl had, both of the speakers had really emphasized, which is this notion of starting with isolated microbes with a known phenotype of antimicrobial resistance, and then working with the genomes that can now, in this era in particular, be derived quite inexpensively. So, and here I'm going to narrow on one specific example, one of these antimicrobial modifying enzymes, the carbapentamases. And by studying isolates, one can do an approach called molecular epidemiology, which I think many people in the room are familiar with, where one can then back work out the chain of transmission when a new antimicrobial allele arises or a new antimicrobial resistant strain arises. One can retrospectively go back and track how it spread, combining the information gained from the genetic sequences of the isolates to the epidemiologic facts of when the specimens were collected and which patients and which facilities. And beyond these networks, I want to highlight some work done by one of my colleagues, Evan Snitkin at the University of Michigan, who, using these sort of isolate approaches, noticed a couple things. One is there's a slight difference between colonization in a patient, but a relationship between that, as well as how often the patient is infected, converts to infection. So one can, by using screening specimens, identify the microbes that then later on resulted in an infection in that patient. This in itself is not a huge burden, but gets into a question about how are these microbes, once introduced, or the phenotype of antibiotic resistance, spreading? And has been intimated in some of the previous talks, there are really two ways by which the phenotype of antimicrobial resistance can spread. It can spread vertically, which is to say you have a strain that has been transformed either by modification of one of their chromosomal enzymes or gain of a mobile element containing a protein coding sequence that confers resistance. And then that microbe with that advantage is able to grow and spread containing that. The other way the phenotype of antimicrobial resistance can spread is horizontally. So there, the key elements, the key peptides, the key protein coding sequences are transferred as mobile elements and transform completely different microbes that then gain that phenotype of antimicrobial resistance. And the degree of importance for vertical versus horizontal transmission remains an active area of study. And so I wanted to then show what Dr. Snitkin had done. He had gone back to when the carbapentamases first arrived in the United States and looked at isolates from 3 distinct centers, each of which had outbreaks of this phenotype of carbapenem resistance for the first time, roughly in the same time window, but not exactly overlapping. And then use these tools of genetic epidemiology, a study of the genetics of the isolates retrospectively to try to characterize the relative importance of vertical versus horizontal transmission. And one of the ways this can be identified is how often is the phenotype being imported into the network or into the facility versus how often is, once the strain is established, is it spreading? And a very interesting finding came from this study. In 2 of the 3 initial outbreaks in the states of Connecticut and Minnesota, the cases that those facilities saw were primarily imported. The 3rd site in Tennessee, there was more of a mix. And in fact, one of the dominant ways that that particular phenotype of carbapenem resistance spread in the facility was through vertical transmission as opposed to horizontal, or through not importation, but expansion. And that's really interesting. Another way to kind of see this is to sort of cross-reference how much of the KPC was shared between different isolates of different genetic backgrounds. You can see Tennessee on the left, in the Minnesota outbreak on the right, and you can see in the Tennessee outbreak, there's a lot more of sharing. You get the reds and oranges showing that once there was a strain that was very favorable, it just spread and spread vertically. And there are 2 important things that were inciting to me about this. One is in the other 2 sites, and in fact, even when you add in all together, the majority of way by which the phenotype of carbapenem resistance spread in this initial contact in the United States was by horizontal gene transfer events, where this allele is spreading not in an organism that has increased fitness and then spreading from person to person, but as a mobile genetic element, then suddenly finds itself in microbes with quite different genetics, including other mechanisms of antibiotic resistance and other abilities to adapt. And then that in turn seems to have an effect on driving the evolution of that allele. That allele then has an opportunity to adapt in a new host, and in both a host in the sense of the prokaryote and bacteria that is hosting, as well as the patient and the patient conditions that host today. And I just want to highlight the tool that was used here out of Dr. Snitkin's group to very quickly assess, is an allele spreading or is a microbe, are things spreading horizontally or vertically? A tool that I found to be useful. And I just want to highlight another point that has been made. There's an entire panoply of ways by which microbes can adapt to the pressure of antimicrobials. And again, I completely concur. This speaks to my clinical practice. All 5 of these distinct mechanisms are a routine problem and often interacting together. So you have modification of antimicrobial targets, you have regulation of the antimicrobial target. And I'll give an example of that in just a second. An anaerobial modifying enzyme like a carbapenemase reduced access to the target either by efflux pumps or reduced permeability. And then this odd phenotype, which I'll also touch on at the very end of my talk, of alternative physiology of cells engaging completely alternative physiology that de-emphasize the role of the pathways which the antimicrobial is targeting. So why do I bring this up? So for example, now, in the United States, we do not use typically colistin when we encounter a patient with a carbapenem-resistant isolate. We use the beta-lactam-beta-lactamase combination. But very quickly, in a patient, multiple of these mechanisms are engaged. Changes in permeability, changes in the regulation of, in this case, the penicillin binding proteins, that join with the antimicrobial modifying enzyme that the microbe already has to then result in the emergence of antimicrobial resistance, which is in a way not genetics in the sense it's not a change in the DNA that is spreading from person to person, but an inheritable, repeatable phenotype that can occur in the circumstances of the patient. I just wanted to bring up this regulation of an antimicrobial target in a little bit more detail. This comes up quite often. And the classic example for me is the Visa phenotype, or vancomycin intermediate Staph aureus, where the phenotype is not a change in the penicillin binding proteins themselves. So this is not like vancomycin resistant enterococcus, where there's no longer the target for the vancomycin to target. Here, ironically, it's increased production of cell wall components with reduced expression of the organizers of cell wall. And in effect, the cell wall becomes thick and disorganized and acts as a suicide inhibitor for the vancomycin. And in turn, this results in relative resistance against daptomycin, one of our backup antibiotics. And this phenotype is a little bit, again, tricky to imagine. It's not quite spread vertically. These strains, once they emerge, don't tend to spread from person to person, but they can be reliably regenerated with the similar set of exposures. And this is a great example in my mind because it's been known for over a decade, and the underlying genetics of these changes are better understood. Another one that comes up, and as I mentioned, porn mutations, particularly in Pseudomonas, is the dominant way in the United States by which I, as a physician, encounter carbapenem resistance. So I have a patient with leukemia or other very poor immune function. They get a pseudomonal infection that's carbapenem susceptible, and then it becomes resistant by changes in the endogenous chromosomal enzymes within that organism. And here, the phenotype that is critical is the genetic plasticity of the Pseudomonas and their ability to have these mutations and remain viable. I want to highlight a resource, particularly for some of the learners here. There is the CARD database, which is at least my go-to to identify examples of mutant alleles or variant alleles where this phenotype has been established. There are certainly others, but this goes through and it's a very nicely organized ontogeny. Okay, so lots of wonderful work is and can be done with isolates, but it doesn't tell the whole story. And one of the examples I give here is the ability to now study wastewater and waste sludge out of water treatment plants, which themselves can act as a reservoir for these antimicrobial resistance alleles that can be in environmental organisms unsuited to life inside the host, but then through this horizontal gene transfer process, end up back into pathogens that are relevant for human health. So can we study it? And there's a variety of very mature methodologies that are complementary and have distinct strengths and weaknesses to take, say, a complex microbial community, whether in a patient like in stool, just which microbes are present, but also which antimicrobial resistant alleles are present and relate them back to antimicrobial resistant genes. So I'm going to highlight one of the techniques, one of the ones I feel like is the most viable and efficient from a computational perspective and wet work perspective, which is used a pair of PCR-based techniques. You have a list of antimicrobial resistant genes you are interested in. You do PCR to try to identify alleles of them, and then cross-correlate that computationally with the composition as determined by, in other words, which microbes are present by amplifying a taxonomically informative gene. In this case, most commonly the 16S rRNA small subunit gene, which is ubiquitously present in all microbes with intensely conserved domains that then are intertwined with highly variable domains that can be used as a sort of barcode to identify which microbes are present. And I want to highlight one of the strengths of one of the earlier studies I showed that combined data not just from one site, but multiple sites. Combining data from studies done with these broad technique, but slightly even the minus differences in technique can result in completely non-overlapping sequences being resulted, and a real challenge in combining data from different studies and revisiting the wealth of data that already exists in studies of not just antimicrobial resistance, but of other ways of the microbiome. I'm going to do a metaphor. Please excuse me for this. I give this as a question of like sending children to a zoo with cameras and ask them to take pictures of animals, and then try to figure out which animals they encountered after going to the zoo. And children being children, some take pictures of heads, some take pictures of eyes, some take pictures of bottoms. And then from a computational perspective, this is incredibly challenging to figure out what animals and which two kids saw the same animals. And it's even more when they've all just seen a bunch of cats. So, and one insight that I had come up with my collaborative team is that even when you have completely non-overlapping sequences, if you use a technique called phylogenetic placement, where you take these short sequences and place them on a set of full-length references, they tend to converge on the same part of a phylogenetic tree comprised of full-length references. And therefore, this distance on the phylogenetic tree can be used to make a novel unit, a way of tracking which microbes are present that is independent from taxonomy, And not only is it able to be done, but it can result in a finer grain separation of organisms than taxonomy, the dominant technique that we use. Why does this matter? Particularly again for antimicrobial resistance alleles, where a majority of the events are occurring or can occur by horizontal gene transfer events. Even getting to the ability to identify a strain is not necessarily a great way of tracking a particular microbe, because it's not the strain, it's the gene, and combined with a more stable long-term genetic background. And so that's my proposed model for how to revisit this data that is being stored at immense cost, to go back to the raw reads and perhaps get a better look at how often antimicrobial alleles are being spread between microbes in complex communities, as well as getting a better understanding of comparing data and seeing how consistent that effect is across different sites and different studies. Shifting to metagenomics, I have a similar sort of suggestion and thinking about maybe less usage of intense reference-based approaches. As been previously described, and I won't belabor this. This is a slightly different technique where the DNA is randomly sequenced, and then there's an attempt to redo it. And the dominant approach that is used here is to map these small reads to reference genomes, and both then get a sense of what taxonomic, again, species and strains were present, as well as the genes based on the genes that have been previously identified in reference genomes, and quantify it. In contrast, I advocate a slightly different approach of using references only at the very end as a decoration, to use de novo assembly into contigs and then extraction of protein coding sequences from those contigs as the base rather than reference genomes, quantify through alignment, and then identify antimicrobial resistance genes by then going back to resources like HARD, where you can compare the new alleles that have been identified strictly from the raw data that has been generated from these specimens based on their similarity to known alleles of previously identified genes. And then finally to relate them to all the other genes, but then go through a step of dimensionality reduction. I'm going to give an example. I think in a simpler problem, which is relating the microbiome to, in this case, efficacy of a cancer therapy. This approach can do a remarkable degree of reduction and concentration of the information in the data from billions of reads to a couple thousand really potent features that then can be used. And really importantly then, again, this is a technique that can allow the combination of data from distinct studies show reproducible associations. So bringing us back to antimicrobial resistance genes, my proposal is to use this broad approach and really look in detail for these changes. And I think in particular, this is a rich approach for looking for the first example, modification of an antimicrobial target. The classic example here would be emergence of mutations in the DNA gyrase gene for quinolone resistance. And I'm going to skip this step and just sort of looking at the time and emphasize summarize a little bit of what I've talked about here. I think that there are alternative approaches, whether to use taxonomy-free ways of representing the composition of microbes and microbial communities, or to allow our data to drive what alleles are present, rather than go forward and try to use reference genomes as our starting base. And although I wish I had a little bit more, this is certainly ongoing work with my colleagues and collaborators. Then I want to bring up this last story, which is about this notion of alternative physiology. And the clinical example I encounter is this phenotype of small colony variants in Staphylococcus aureus. This is a case of often starting as a completely susceptible isolate of Staphylococcal aureus in a patient with a complex long-term infection, a device infection, with a device that is not removable and not easily removed, where one is then attempting very long therapy, either for cure or for suppression. In this case, the way the microbe develops antimicrobial resistance is not by gaining, say, an ability to break down a beta-lactam or changing the target. Here, the cell is broadly changing its entire metabolic structure to make it less susceptible, make less dependent upon production of new cell wall components. In the case of beta-lactams, less dependent upon folate in the case of trimethylthermal sulfide. Likewise, less dependent in general of protein synthesis when you're using a protein synthesis inhibitor, like a doxycycline. Another very interesting aspect of this is this overall phenotype is, it's often reversible. It develops in a patient, is reversible in a patient. And the phenotype is the heterogeneity of the community now in that patient. So the phenotype you'll see is that in the patient, for example, trimethylprim sulfa is failing. But when you culture the organism from that patient, you sample it, and you take it to the microbiology lab, the trimethylprim sulfa works perfectly well. And so I find this a very tricky problem, and I feel like there are some interesting opportunities to use some of the emerging technologies, including combined microbial metagenomics to get, and transcriptomics to get a better understanding of what are the drivers of this phenotype. Because I think this is an important complement to the ability of carbapenemase resistance to actually cause trouble for our patients, particularly as we struggle and develop novel antimicrobials. With that, I want to thank my colleagues and my wonderful team of people from all over the world. And I guess now we'll answer some questions. Thank you.
Now this session is open for discussion. If you have any questions, you can ask. The speakers are here to respond to your questions. Yes.
So this is a question for all the three speakers. So, we do use a few isolates of Acinetobacter, Klebsiella, E. coli, and basically which are causing neonatal sepsis. What we see is that almost all of these isolates in the clinical settings have a wide range of resistant markers, and quite possibly most of them coming from the plasmid. This is in fact a collaboration between me and Dr. And what we see is that the frequency at which they are able to do the natural transformation is quite extremely low. I mean, we do get the variants basically having a wide range of, the natural transmission frequencies. And even the conjugation frequencies is quite low. But if you see these isolates, I mean, you pick them up from different environmental reservoirs across the hospitals. You do see most of them being MDR and XDR. So what could be the possible natural ways by which these are able to, get these particular gases before they become MDR and XDR? Any insights? Thank you.
This is very interesting question. So what, as per my understanding, when you are testing the natural competence, how sure you are this is the optimal conditions? Because depending on the box, those conditions differ. So when you are testing those natural competency in the lab, they might not the actual environment where these bugs stay and occur. Just for example, Vibrio. If you are taking Vibrio in M90mm medium, their efficiency of natural transformation is quite low. But if you are incubating Vibrio with the shrimps with chitin, then you can find that they have high level of efficiency to acquire streams. So when you are taking Acinetobacter bovani, I think we need to really know at what environment do these bugs stay. And if you can mimic, that will give the idea. Even in the animal model and in vitro model, when we test it, we find there's a lot of differences. So that might be one of the reasons that in lab condition, they might not be very efficient, but natural condition, they can acquire this DNA very efficiently.
We are trying in our lab, given that that is the major challenge, because we do not know what is the environmental setting, natural environmental setting at which you can see this. I would want to hear any insights from the other speakers as well. Thank you.
So I also think this is an important question of sort of translating what's happening in the natural ecologies to the, to the place where we need to be studying these organisms, which is largely the bench, right? It's really a big tension, and we need to understand more what's happening naturally. The only other thought I had was just numbers, especially that we don't necessarily understand where these transformations, conjugations are taking place. It's hard to know what the density of the microbes are, the complexity of the communities that are there. So I think even for rare events, if the numbers are high enough and then subsequent selection allows for, that's very rare species to grow and emerge, I think it's still a big problem, even if it is rare. That was my thought.
I'll add the thought that the host itself is an important factor. And even the great example of F. presnitzii, it's murderous to grow that in the lab. And it's literally the one microbe 100% prevalent entire across the world in all human guts. It doesn't even take in mice. And so I think from the perspective of the microbe retaining fitness when gaining a plasmid that may have a fitness cost, the host itself might be a favorable environment. And then I think of some of my patients who do not have immunity, have leaky guts, are maybe even a more favorable environment to support the microbe through its transformation and be able to maintain adequate fitness to maintain a foothold in the overall community. And so that's the other factor I usually start to think about when I think about this divergence between what we find in the bench and what we're finding in the real.
Thanks to all the speakers for the very fascinating talks. So I have a question specifically for Ashley. When you talked about the loss of the antimicrobial resistance over the period of like 30 days or so, so was this just the loss of the plasmid or the phenotype, or was it the complete loss of the?
Okay, I love that question. That's a great question. And it's one where we are attempting to dig into more, because at the moment we don't know. Because though the graph that I shared in the talk and our understanding to date is largely based on isolating things in culture. But we have the opportunity to dig into this further with the samples we have, because we've got the full community. Now, with the short read technologies, of course, it's difficult to sort of piece together what genes go with which things and whether we feel confident that, what we're seeing is, there's a lot of confidence in the technology. I'm not saying that, but it's hard to dig into these questions. Like, do you see the same strain minus a plasmid present, right? That's the question. And so we've just recently started to do more long read sequencing. And now with technologies like the PacBio Revio machine, which has incredible throughput with a much reduced price, suddenly going to the depths required to get into some of these questions becomes feasible. So anyway, I love it. We're on it. I'm excited to maybe hopefully soon tell you something more concrete.
Okay, maybe a small follow-up to all the panelists there. So all of you talked about most of the bacterial AMR resistant strains and everything that you see. Do you see any sort of correlation with eukaryotic parasites, or any hotspots or any crosstalk happening between bacteria or parasites for that matter, where you see AMR also? Because there was a talk about fungi also being one of the focus areas of the whole program. Thank you.
So there are a lot of article already been published that there is a cross kingdom cross form. So bacteria could also interact with fungi, bacteria could also interact with parasites. And some of the molecules, when these fungi secrete, that can also induce the resistance in microbes. But this resistance happens mostly because of the change in the target sites. But you could hardly find any function is coming from the parasite to the bacteria to get the resistance. You want to add something?
Yeah, I think it's an insightful question. I think the interactions between Canada and both commensals and pathogens also are just now fully being appreciated. I have a colleague, Teresa O'Mara, at University of Michigan, who studies this as well as in the context of Canada ORIS. And there can be both mutualistic and competitive relationships that both, I think, are compellingly identified, but I think this is an area that we certainly need more development to understand. And gets back to the other questions have been asked about when are conditions favorable for colonization, and then likewise horizontal transfer of antimicrobial resistance.
Hello, everyone. Okay. I am Dr. Madhav. I am from Scan Research Trust. So my question is from Ashley. Did you check the dietary recall of the candidates which you have chosen for your study? Like you have collected the poop sample, right? So dietary recall might have played an important role. So have you checked that with your findings?
Yeah, so it's a great question, and I didn't get into it here, but partly because the result is negative. So we did collect dietary information. Now I think there's a lot of caveats in the way that we did it. So I don't think it was the most robust method. However, we asked lots of questions about what the travelers were, eating, drinking, and of course, we looked closely at any connection between the diet and whether, they picked up one of our target organisms, whether they picked up more E. coli. We really ran all the data through the ringer. The only thing that was significant, but only in a unique variable model, was, and it makes no sense. It was, you are less likely to get an antibiotic resistant organism if you eat uncooked meat. That makes no sense. That makes no sense. And we think that the reason it showed up and we quickly batted it down is because it happens that people who eat uncooked meat, at least in our cohort, were likely in places where there's less prevalence of resistance. So I think it was confounded. Ultimately, I think there's more to be done in this space. I don't want to say we've closed the book on it, but so far everything we've looked at, there's really no connection.
Okay, thank you. And also one more question from my side. If you collect samples from different parts of your intestine, then it might give you different picture. I have gone through different literature which shows that at different parts of your intestine, there are different bacterial community. Like if the poop you collect from outside the body, there is different bacterial this thing. So have you checked that too?
Yeah, so you're totally right. And I think, we're kind of lazy in the way we talk about it, but we really are studying the poop microbiome, right? It is. And so I think that you're right. It's the lumen. It's likely quite different if you were to get, even closer to the epithelial layer and the mucus. So, and then there's the small intestines versus a lot. So yes, I think that all of that is really important. The other thing that's running through my mind is I think, one of the proposed reasons we see differences in the communities, the stool communities of people with diarrhea is because you're getting more of the microbes in the small intestine that we don't necessarily. So I totally appreciate that that line of thinking. These were healthy people. We didn't ask them for other kinds of samples, but I kind of doubted we would get them. But I agree, it would be so cool to dig into the biogeography and start relating that to some of these phenomena that you're curious about.
Thank you.
We can take two more questions.
First of all, I would like to thank all the distinguished speakers for giving such intellectually stimulating talks. And I'm okay. Yeah, so thank you to all the speakers for giving such intellectually stimulating talks. So based on my limited understanding, I could understand that from all of your talks that how the genomics of the organisms are playing such a huge role in acquiring the spread of the antimicrobial resistance genes. I was just wondering whether the epigenetics of the host can play a role in acquiring as well as the spread of the antimicrobial resistance. And as we know that the epigenetics is so variable and it can vary from host to host. And if there are approaches to study that, I was interested in knowing some computational as well as non-computational approaches from which we could figure out and derive any kind of meaningful conclusions to understand the effect.
It's a lovely question. Thank you. There are some evidence, getting back to, I think, one of the answers to the earlier questions about an interaction between diet and the state of the host and ability of organisms like E. coli, particularly more with the EPEC phenotype, to be able to stably colonize. I had a lovely collaboration with Nobu Kamada, a faculty member at Michigan, where we use an organoid-based model system complementary with murine to understand the role of mucus degraders in lysine-deficient diets, and allowing then EPEC to be able to engage and more thoroughly colonize. And gets into the short point is the microbes are content to take from the diet, but in the context of dietary deprivation, where mucus is available as a source and mucus is degradable, that sets up a positive feedback loop where the mucus degraders support the E. coli and the E. coli support the mucus degraders, and the end result is further damage to the host tissue and the phenotype of diarrhea. I think it's a rich area, and I really am optimistic with these new organoid-based model systems where it truly recapitulates the tissue, and that's where the epigenetics come in. You have goblet cells and intestinal epithelial cells, and depending on the condition, even intestinal stem cells all together, but in a very controlled system without any lymphocytes to get in the way, to really, really start to, in a reductionist way, understand the interplay between these factors.
Thank you so much.
Last question.
Okay, Eitan from Stanford. I have a question for Ashley, for everyone. Hear me?
Yes, yeah, oh, yeah. Oh, yeah. Yeah.
Okay. Question.
Someone help me.
I can hear you. If you want, I'll repeat your question.
You're audible, sir. Okay, now it's more stable. So it's a question of speculation. And in your study, the people living in the US, 40% of people got back with resistance. So it's for you, but actually all the panel. What do you think? What do you speculate would happen if somebody would leave India and travel the world and come back here back to Delhi? Would they also gain resistance or maybe more other types of antibiotics or not at all? Do you have any thoughts about that?
We should do the study.
It's good.
Yes.
Where are the funders? Let's do that study. No, I think it's a fantastic question. So my speculation is that the guts of people from India are not any less likely to interact with the microbial world than the guts of people from the United States who live in the United States. I think that there is a high likelihood that we would see things coming back that are new to them. I could also, like, if we really sat down and started to, like, map it out, like, their competition, and like, we could talk about this for a while, actually. But my gut tells me that their guts are not any fundamentally different. We really are kind of test tubes for particular types of organisms. I think we encounter lots of things that are in the environment that can persist there and live there, so that when we take them in whatever route we do, that they can have an opportunity at least to colonize. And so, yeah, maybe I'll just stick with that. I know we're short on time, but let's talk more later.
Thank you.
And I'd like to hear your speculation later too.
We should conclude now. Thank you, Dr. Arthur, Dr. Jonathan, and Dr. Bhabatosh Das for wonderful scientific session. And thank you and congratulations to all the lovely audience for a very interactive scientific session. And we managed the time well. We just finished whatever time allotted. Exactly within the time. Thank you, everyone. Now over to the organizers.
Thank you to all the session chairs and speakers for the exciting and informative session. Now I request the session chairs to kindly felicitate the speakers. Dr. Shanta Datta, to hand over the token of appreciation to Dr. Ashley Earle. Dr. Anil Kumar Pandey to Dr. Bhabatosh Das. Dr. Jayanta Bhattacharya to Dr. Jonathan Golub. Now I humbly request Dr. Rupa K. Bhadra to kindly come up to the stage to felicitate the session chairs. Sir, please. Our session chairs, Dr. Jayanta, sir, Dr. Shanta Datta, Dr. Anil Kumar Pandey. So all of you are requested to have a group photo for this session. All the speakers and chairs. Please join to the picture. Thank you, ma'am. Thank you, sir. So we now break for tea. I request the conference volunteers to assist the dignitaries to the area for tea. And the participants who are presenting the poster on AMR today are requested to be present near their respective posters for the evaluation. I request everyone to assemble back by 11:30 a.m. for the start of the next session. Thank you.
हाँ जी।
नीचे तो करो, कैमरा नीचे तो करो। नीचे करना। हाँ, जूम करो, जूम जूम करो। ठीक है।
ये बुक से शुरू करना. So we'll be starting with our next session shortly. So all are requested to assemble back in the auditorium. Welcome back, everyone. After a very exciting and informative session and some brainstorming over tea, now we begin with the second session of the conference on AMR in bloodstream infections, sexually transmitted infections, urinary tract infections, and respiratory tract infections. I humbly call upon the session chairs, Dr. Rupak K. Bhadra, Dr. Ranjan Nanda, and Dr. Tushar K. Methi to the stage. Please, sir. Dr. Rupak K. Bhadra is the Emeritus Scientist at the CSIR, Indian Institute of Chemical Biology, Kolkata, India. His research area is focused on understanding the molecular basis of stringent response regulation in the cholera pathogen, Vibrio cholera, under nutritional stress conditions, and elucidation of the role of intracellular signaling molecule in regulation of gene expression related to growth, virulence, codon sensing, and biofilm formation. Thank you, sir, for accepting the invitation and joining us today. Dr. Ranjan Nanda is the Group Head of the Translational Health Group at the International Center for Genetic Engineering and Biotechnology, New Delhi, India. His areas of research is to understand host-pathogen interaction in tuberculosis disease, with major focus on clinical patient-based studies for hypothesis building and employ in vitro and in vivo body systems for mechanistic and validation studies. He's also keenly observing the gut microbiome dysbiosis and altered fecal metabolome in tuberculosis patients. It's a pleasure to have you here with us today. Dr. Tushar K. Methi is a professor at the Regional Center for Biotechnology, Faridabad, India. His research is focused to delineate the mechanism of protein misfolding, aggregation, and protein quality control systems in cancer and neurodegenerative diseases. So in this session, each talk will be of 20 minutes, followed by a 10 minutes long question and answer session after all the speakers finish their talk. I request the audience to kindly reserve their questions until the session is completed. So I now request the session chairs to introduce our first speaker and call him over to the stage. So please.
Good morning, everybody. The first speaker in this session is Dr. Purva Mathur. And Dr. Mathur is a professor and MD at All India Institute of Medical Sciences, New Delhi. Her field of research is microbiology and includes hospital-acquired infections. She has pioneered the development of the first surveillance system for hospital-acquired infections in India. She monitors the nosocomial infections and assesses the traits of antimicrobial resistance. She is the member secretary of the Hospital Infection Control Committee of the center and is in charge of the dietary services at the JPNA Trauma Center. With this short introduction, I would like to request Dr. Purva Mathur to give his speech. Thank you.
Thank you so much, chairpersons, and good morning to everyone. I work in the field of hospital-acquired infections, and I was hearing the lectures in the first section, which were purely on genomics and antimicrobial resistance. Through this lecture, I would like to bring the spectrum of antimicrobial into the clinical domain of how antimicrobial resistance impacts healthcare-associated infections, because it is those patients admitted in the ICUs and wards who are really largely impacted by these highly drug-resistant bugs. And to bring into your understanding how the outcomes of very good surgeries, transplants, ICU care is now being impacted through healthcare-associated infections. So we know that as the surgical practices, the critical care is advancing at a very, very high speed. The impact or the outcome of these very good surgeries is now being, held at ransom by these highly resistant bugs, which if the patient acquires and the patient is immunocompromised, there is a high chance of antimicrobial resistance. So just a brief review about what we are doing at All India Institute of Medical Sciences, along with a big network of hospitals across India to develop surveillance system for healthcare associated infection. So this is the figure from CDEP, which does global antimicrobial resistance surveillance. And you see that India is in the highest prevalence of carbapenem resistance, which are almost the last generation antimicrobials amongst the beta-lactams. And this is true for almost all classes of antimicrobials. If you see the ICMR's annual report, which was published last month, ICMR publishes its annual report, we see that the carbapenem resistance has increased exponentially, and now we find colistin resistance to be also very high amongst enterobacterials and non-fermenting gram-negative bacteria. So we are almost at the last resort antimicrobials, as we see in clinical practices. But the critical question is that does this MDR or XDR pathogen prevalent everywhere in hospital? How do they impact patient outcomes? I mean, they do cause infections, but ultimately, are they really the attributable cause of mortality in ICU patients? So we did the study and we found that we really have to see that how many patients are actually colonized with these multi-drug resistant bacteria, how many actually get infected. So it's very important to distinguish patients who are probably colonized, because the hospitals are a hotbed for multi-drug resistant pathogens. You find MDRs in every environmental niche in a hospital. So how many of these are actually clinically infected by these MDR bugs, and how many have a fatal outcome? So if you see that MDRs or XDRs are all present everywhere in hospital environment, and very few of these patients will get infected, and a part of this will have a fatal infection. But even if you see that the magnitude of patients who are colonized and then get infected and have a fatal outcome, it's a huge burden for all healthcare facilities. So it's very important that we estimate the burden of healthcare associated infections, do it properly using proper denominators and numerators and proper metrics. So if we talk about healthcare associated infections, we know that more than 1.5 million people suffer from HAIs at any time. Prevalence is more than 40% in developing countries, and HAIs kill more people every year than AIDS, breast cancer, and accidents put together. Of course, it increases the cost, prolongs hospital stay, increases the morbidity and mortality. And on top of that, we have the antimicrobial resistance with no new antimicrobials. As we often hear, the WHO estimates that 10 million people will die annually by 2050 because of AMR alone. The ironic part of this is that almost 70% of infections acquired in hospitals may be preventable. So there is a cost factor. So there was a very last study in US in which it was found that if a preventive program for healthcare associated infection can prevent just 7% of HAIs, your entire program cost is covered. So it's very, very cost effective to have an HAI preventive program. So prevention, of course, need priority. And what we are now moving towards is a bundle approach. So bundles are what they are a set of practices. When you implement them together, they have a good clinical outcome, better than if individual elements of prevention are applied in a patient. Do we have Indian bundles? No, we don't have Indian bundles because we really didn't have data of what is the actual burden of healthcare associated infections, bloodstream infections, urinary tract infections, ventilator associated pneumonia or surgical site infection. So that's where we were around 2014 and 15, that we thought that in India, there was no systematic surveillance across a network of hospitals where we could say that, okay, this is our benchmark, this is our rate of bloodstream infections, UTI, VAP, and SSI across India. So one of the seminal study, which was done in late 70s in USA, which was the CENIC study, found that if you do HAI surveillance properly, you can prevent almost 30% of infections just by doing surveillance. When we see literature, we found that only 15 to 16% of developing countries have any system of surveillance, So doing a literature search, we found that very few countries have a system of surveillance for healthcare associated infection. So we know that ICMR and NCDC have parallel verticals doing AMR surveillance. They are established networks working on tertiary level or medical college level. These AMR networks are bringing in quality AMR data in their network. At AIMS Trauma Center, we were doing HAI surveillance for only our center since 2008. So we had a system of HAI surveillance for the last 5 years. It was in 2015 that we got together with a grant from CDC under GHAC's program, and we started developing the first of its kind HAI surveillance network building on ICMR's and NCDC's AMR network. So we started with ICMR's network initially because they had 20 hospitals which were generating quality AMR data. So our original objective was to layer on HAI surveillance into these hospitals which were doing antimicrobial resistance surveillance and slowly build upon the capacity for infection prevention in these hospitals. So we started with 5 hospitals in 2015, geographically into North, South, West, and East. And we started with HAI surveillance. We also started a tier 2 external quality assurance system so that all these labs had quality AMR data. We also started laboratory strengthening for quality assured AMR surveillance and helped ICMR in its antimicrobial stewardship program, the initial trainings. So we started developing with a baseline assessment tool. We developed our own SOPs, training modules. We started slow with only doing central line associated bloodstream infection surveillance. We developed the first of its own kind database. It's an indigenous live database, which now all the sites are using. We've conducted trainings, and from 2015 to 21, we have increased the geographical reach as well as the scope of HAI surveillance and coupled this with database action. From 2022 onwards, we've expanded this network to medical colleges and district hospitals. So this is how we've geographically expanded, and we had 13 centers in 2017, then 20 centers in 2018. And by 2021, we had 121 facilities doing surveillance using the same module, the same SOPs, and the same methodology. So these guidelines were adopted from CDC's NHSN, but tweaked to suit the Indian medical system. So the definitions and protocols were slightly different from CDC's NHSN. So more than 190 ICUs were using this database, doing the surveillance using the same protocol. From 2022 onwards, we started partnering with multiple Indian agencies and hospitals to strengthen hospital infection control and expand this network. So we are in the process of signing an MOU with the National Health Center Resource Center, the NHSRC, which works under the National Health Mission, looks after the district hospitals. So we have trained more than 700 district hospitals for doing HAI surveillance. We are now partnering with National Centers for Disease Control, which looks after the state level medical colleges. So more than 80 medical colleges have been trained and are part of this network. And we are already, as I said, working with the ICMR's network, and a couple of hospitals are participating through your own voluntary participation. So AIMS New Delhi is now collaborating with ICMR, National Centers for Disease Control, NHSRC, working at the district hospitals, all the new All India Institute of Medical Sciences, the Laksh Quality Control Program for surgical site infection surveillance, and is supported from CDC for technical and financial support. So this is the proposed framework for infection prevention and HAI surveillance. The AIMS is now mentoring around 7 to 8 regional centers in different parts of the country. And these regional centers are liaising with individual healthcare facilities and the state level ministerial health missions so that it becomes a self-sustained activity. So we are working in 34 states and union territories. Problem. So currently we have trained 717 district hospitals, 90 medical colleges, and we are present in 34 states and union territories. So this is the distribution of the participating hospitals. We are working on laboratory assessment and infection control assessment, working on HAI surveillance, which includes bloodstream infections, UTIs, VAP, and surgical site infection. We are using the data generated to have our own preventive bundles and IPC activities, performing lab strengthening through external quality assurance system, the Tier 2 EQAS, which is now handed over to ICM. And also, we helped ICMR in the initial steps of training for AMSP. So this is the database, which is www.haisindia. All the training modules and everything is available on this website. All the sites use this for real-time data entry every month. So it's a very live database that we are using. These are some of the graphics of our database for BSI, UTI, SSIs, and VAP. So from May 2017 to June 2023, we have a data of 2.5 million patient days in our database with more than 700,000 central line days. The bloodstream infection rate in our network is 6.36 per 1,000 patient days. We have more than 1.3 million urinary catheter days, the denominator data, and the UTI rate in our network is 1.85 per 1,000 patient days. When we see the organism profile, we find that in bloodstream infections, the most common organism is Klebsiella and Acinetobacter, followed by Candida species. In UTI, Candida is the most common organism, followed by E. coli and Enterococcus species. And in VAP, again, gram-negative pathogens predominate. Coming to the antimicrobial resistance profile, it is similar to what is published in the ICMR's AMR network. We have a very high rate of resistance of bugs causing actual infection. So this is where, colonization versus infection comes. So these are actual bloodstream infected patients who are clinically defined to have bloodstream infection or VAP or SSI or UTI. And the rate of antimicrobial resistance is extremely high. So our carbapenem resistance rate is more than 80%. And even colistin resistance is increasing in these pathogens. So whether it is bloodstream infections or urinary tract infections, you can see that the AMR profile in these infections is extremely high. We are now using this data for infection prevention and control. So it's a circular cycle. If you're only doing surveillance, you're only collecting data. So we're encouraging and training each hospital to start quality improvement based infection prevention and control protocols. So each hospital now presents their own work on infection prevention control through online once in 2 weeks meeting, and it helps other centers to, take up those actions. So this data-based action is now being used for central line associated bloodstream infections, surgical site infection, or catheter associated urinary tract infections. So these are the trainings which are given through AIMS and CBC and all the sites participate for this quality improvement based IPC training. And these interventions, which are low cost interventions, are adapted by other hospitals, and it helps in prevention of infections, and ultimately in the long run will help in the overall burden of AMR. So this quality improvement program is helping in getting knowledge across all hospitals. As a team, all these hospitals develop their own bundles and implement them. And it helps in training the different cadres down to nursing staff in implementing these QI-based protocols. These are some of the manuals and SOPs which we have prepared. They are there on the website, and any hospital which is voluntarily keen to join our network, we give them training and encourage them to participate in these. So these are all the SOPs that we have developed. As I said, we've also started a tier 2 EQUAS. We know that in India, there are 2 EQUAS. One is in North India and the other is in South India. We started to have a tier 2 EQUAS through this program, and in which the PT panels were sent to all the hospitals and the reports were given to them. And this has been now transitioned to ICMR. ICMR is now hand-holding these through a grant which is given to CMC Vellore. So these are the PT panels which we sent across to all the hospitals, and then it was transitioned to the Indian Council of Medical Research. As I said, from 2021 onwards, we started publishing our annual hospital-acquired infection data in the ICMR's annual report. So this was the first time that the network level data is now available in the ICMR's database portal and the ICMR's annual report. We've been actively involved in training, so more than 100 healthcare cadres have been trained in the last 5 years, and we continue to do that. This number has increased much more now, with us going into the district hospital levels, where the microbiologists and nurses are being trained for hospital-acquired infection surveillance. So these are some of the training programs that we did. We continued these trainings during the COVID period through online mode. So a lot of trainings happened even during COVID times and included COVID-based training also. One of the main objectives of this program was also to have a Center for Advanced Research and Infection Control, which would cater to the training needs and for AMR research. So ICMR has granted us a Center for Advanced Research on Infection Control, and we are collaborating on a number of, now we're moving to genomic-based surveillance. So we are planning to, collaborate under ICMR's genomic surveillance program, because we have a huge number of isolates which are known to have caused infections in ICUs. So it will be good to have a genomic database of these pathogens, and because we know the outcomes of these patients also, our database and our data is such that we know the 14-day and the long-term outcomes. So it's a huge data which we can leverage if we couple it with the genomic surveillance and actually what kind of genes are there in these infections. So the achievements of this network is that this is the first national level HAI surveillance and infection control network. All the hospitals are using standardized methodology. There is coordination between different agencies like NCDC, ICMR, NHSRC, all the All India Institute of Medical Sciences, a number of tertiary care hospitals and private hospitals. This network is generating quality data. We are now using this data for action. We have an external quality assurance system for antimicrobial resistance. We have a laboratory capacitation program in which all the sites are visited and any loopholes are plugged. And we've also been able to investigate a number of antimicrobial resistant infection outbreaks. So this has been one of the other major achievements of this network, and of course, the training component. So the first 18 months of data from this network was published in the Lancet Global Health in 2022. And we've also published the reports of the baseline assessment in American Journal of Infection Control. So we had the facility level WHO tool, which is filled in by all the participating hospital, and it gives very good, approach of finding in which we know that what are the areas which really need to be filled through this surveillance network. So some of the outbreaks also have been reported, published through us using the surveillance system in our network. The challenges have been that this being such a first of its kind initiative in India, we really need to work very hard to get local buy-ins by state and national levels, the sustainability and resources to sustain it for long term, homogenization of various activity, and now percolation to district hospital level. So going forward, we envision that there will be a national infection prevention and control program under NCDC. We know that we are in the phase 2 of antimicrobial resistance national program, and the IPC will be made a part of that. And there will be phased implementation at state level under NCDC and Ministry of Health, and district level implementation through NHSRC. These are some of the national level trainings done through NCDC at the state level program. I really would like to thank the organizers, thank the All India Institute of Medical Sciences, the administration, ICMR, NCDC, CDC, NHSRC, and the other bodies which have been, and of course, all the participating hospitals who've been actively participating in this network. Thank you.
Thank you, Dr. Mathur, for finishing the time. Our next speaker.
Yeah, our next speaker is Dr. Stephen Becker. So, Dr. Becker is a molecular microbiologist with an interest in the microbiology, genetics, epidemiology, and treatment of enteric infections in developing countries, with the emphasis on focal pathogens that include novovirus, Shigella species, and Salmonella typhi. Dr. Becker obtained his PhD from Imperial College of London, studying Salmonella genomic diversity. So his group uses various genomic and laboratory approaches to understand how bacteria and viruses that cause the infectious disease in humans in low and middle income countries and evolve and spread. So they study the phylogeography of enteric bacteria and look more closely evolutionary adoptions and clonal replacement of GI pathogens in endemic settings through cohort studies and hospital surveillance. So in addition, they are also interested in impact of antimicrobial access and treatment on the gut microbiota and the generation of drug resistance pathogens. So he'll be talking, where do we go next with AMR? Dr. Becker, please.
Thank you. So thanks for the introduction. Also, thank you for the invitation to speak to you all today. It's always a pleasure to come to India and talk about AMR. So I have a split personality. So I'm here on behalf of both the University of Cambridge in the UK, but my partial employer now is IAVI, and I'm here for the next couple of weeks in India to try and drum up some interest in some of the work we've got going and hopefully move some AMR research to the next step. So this session is about bloodstream infections. So what I'm going to do is not talk about bloodstream infections. I'm going to talk about something completely different. And hopefully my job is not only to interest you, but also to inspire you a little bit in where we can go next with not just thinking about AMR, but how we can do something about it. Yeah, so my background in microbiology really comes from spending many years working and living in Southeast Asia and using applied tools such as epidemiology and then moving into genomics to understand key problems. And probably two of the bits of work that I'm well known for are demonstrated here, one of which is the first real paper using genomic technology to map typhoid fever. So we're using genotyping tools and then geosurveillance mechanisms to understand how typhoid is distributed, in this case, in a particular region of Kathmandu. And secondly, more recently then, is using microbiome-based technologies, including with cohort studies and also genomics, to understand pathogen diversity, but also plasmid diversity. And as you can see from the title, the take-home message here is that colonization of the gut with drug-resistant E. coli leads to the generation of drug-resistant pathogens by active plasmid transfer in the gastrointestinal tract. So moving on from this, we kind of know how these things work, but actually we have a kind of slightly superficial view of the world of microbes and what we're doing to them. So we live in a bacterial world, whether we like it or not. Human beings, as we're doing a very good job of, are shortening our existence on the planet, that microbes were here before us and they'll be here long after us. And actually, I've not done the count myself, but I'm trusted to hear that we have more bacterial cells living in us than we do have human cells. So whether you like it or not, you're more bacterial than you are human. And what we've done is probably the biggest and worst scientific experiment in history in the last 70 years by exposing them to a whole cornucopia of different chemicals which impact on them. And they will impact on all aspects of our microbiome and every organism we have in us. From a clinical point of view, we aim to treat the infection, but we actually ignore the bigger problem largely is what we're doing to every other microorganism in our body. Because antimicrobials are beautifully sophisticated chemicals in the way they interact with bacterial cell, but what they actually do is not so sophisticated in the way they interact with everything else. They work like an atomic bomb. Anything that is susceptible to them, they will destroy, and anything that's not, they will interact with and create different metabolic processes that we don't really understand, but lead to ongoing inherent resistance. And this experiment, as demonstrated quite nicely by this slide, and I was listening to Babadash yesterday talk about this, has basically led to the fact that there isn't a single new drug that's been released since the start of the antimicrobial era that we haven't had resistance emerge to within 3 years. And we've burnt through these at a relatively alarming rate, and now leading into the discovery void, which is fueled by various things, including probably a lack of targets and also a lack of the pharmaceutical industry's interest in to generate any profits from this area, and therefore pulling out of it. So we really are at a bit of a kind of juncture in humanity of where we now are largely at the end of the antimicrobial era, unless we can do something about it. And again, not to labor the point, but we all know this information. If we believe the predicted patterns as estimated by Jim O'Neill in the O'Neill report in 2014, that AMR is going to be the biggest killer of humanity by 2050, greater than cancer, and also probably, if we want to get people interested in the economic impact of what we're doing with drug-resistant bacteria globally, is going to bring us all to a kind of financial meltdown. So those are the kind of start facts, and I think probably we're all in this room, this is like preaching to the choir. We know a lot about this, and this is why we work on it and why we're interested. And I'll give you a couple of examples. So I have put almost here in italics. So Asia is a very interesting region for drug resistance. Now, not all, there are certain clones of certain bacteria that have originated in hospitals in Europe and the US, but almost all, including most of the organisms with that are drug resistant in most parts of the world. And there is a reason for this. The reason for this is, as we know, large human, large animal husbandry density, access to pharmaceuticals, and also manufacturing, loads of other potential purposes. This a real kind of stand for studying AMR. So India is the right place. India has to lead the charge in understanding what we can do next with AMR, because this is the place to do it. You have access to the best hospitals, the best facilities, but also potentially this is where all of the new infections will emerge from. And hopefully this will work. So this kind of depicts the point of some work we've done in the past using genomics and how AMR spreads as a model. This is Shigella sonnei. This is ciprofloxacin-resistant Shigella sonnei. On the bottom, we have a timeline ranging from 1996 up until 2015. The blue dots are susceptible to ciprofloxacin, the red dots are resistant. We have a phylogenetic tree there of the genome sequences and then a map of where they've emerged from. So we have the first organism emerging in whatever that is, 2008. And then we see this rapid clonal expansion moving internationally, radiating out onwards. And this then is a really good example of the way drug resistance emerges and then spreads relatively rapidly. Now, almost every Shigella sonnei in the world is ciprofloxacin resistant. This is a cluster we identified in the UK. This is how we picked it up originally when I was working in Southeast Asia. And just to highlight my previous points about where the origins were, how they spread from, this is the main cluster coming out of South Asia. So this is a fairly dramatic turn of events. The acquisition of 3 simple mutations led that suddenly we had a clonal replacement spread across the world. And now every Shigella sonnei that's resistant to fluoroquinolone has come from those 3 events. And this is obviously not the case for every organism. This is one of the best models because we can follow it so accurately, but lots of other bacteria will follow similar suits. And again, some work just to demonstrate why important this is. This is then some work again that we've published and some opinion pieces. If you've got insomnia one evening, you wish to read about something we've written about, you can read these. But this demonstrates the changing trends in multi-drug resistant organisms isolated from bloodstream infections in patients in a major hospital in Kathmandu. The blue line there depicts the non-MDR organisms. The red line depicts the MDR organisms. You can see a period where MDR was not that common, then a period where they tracked each other, and then they took over from about 2010 onwards. And now probably upwards of 70–80% of people hospitalized in South Asia with a bloodstream infection have a bloodstream infection caused by an MDR organism. And this is common everywhere. So we're quite good at kind of collecting information, describing this, but where do we go? And without being kind of overly pessimistic about it, I would put this as a bigger challenge as something like climate change for where we go next with the destiny of humanity in the next 50 years. And this then, because of the scale of these problems, what we're really good at is doing nothing. We don't know where to go. This is a major problem that we don't really know how to tackle. And therefore, any degree of coordination is going to be problematic. Now, this would be pessimistic, and I do believe that we can do things. And what we're very good at as a community is collecting information. So microbiologists, and now the advent of genomics, we're very good at piecing together these puzzles of working out how these things emerge and spread. And that then depicts the nature of the problem that we have to take on. But we need new ideas. We need new science. We need new creativity. We need to move it forward. Clearly, we need new drugs. We need new diagnostic methods to actually diagnose the organisms that were actually causing the infections in the first place. Bacterial vaccines is still a bit of a backwater of vaccinology. Monoclonal antibody therapy, which I will talk about a little bit as I go forward, is clearly an avenue that we need to explore. Repurposing existing drugs. Are there drugs out there that are used in other places for other presentations that can actually help us treat these infections? Alternatives. Everybody asks me about bacteriophage. Don't ask me about bacteriophage. I'm not a bacteriophage expert. But these are alternatives, mechanisms that we can think about killing bacteria. Reversion. Are the mechanisms to actually then reuse the drugs we've got by actually preventing these resistance pathways working? And also then clearly in places such as South Asia and other places, reducing the amount of antimicrobials we use to try and have better access to what we've got at the moment. But realistically, this is not a one-stop problem. We need to do all of these things and considerably more. And I would say from my personal point of view, this is a massive area of potential expansion for microbiological research and where we go next. There isn't anything that I don't think should be excluded from the potential argument of where we can go next. Now, I don't have the solutions, but what I'm going to show is some work since I've been in Cambridge that we've been doing to really think about how we can use more applied techniques to understand AMR and what we can do about it. So something that we've not done particularly well historically, and now we can do because of the power of genomics, is consider bacterial variation. Bacteria are different to one another. Let's take Klebsiella pneumoniae, for example, one of the most diverse bacterial species imaginable, almost impossible to find two the same. So if we're going to then generate new vaccines or interventions against Klebsiella pneumoniae, we need to understand Klebsiella pneumoniae. And like many others year round, have collections of these things that can be catalogued and characterized and used. And we've generated one that we think is globally representative of what's going on in Klebsiella pneumoniae at the world. We can identify resistance genes, we can make phylogenetic trees, and this becomes a stock kind of panel of things that we can then use to develop future kind of interventions. New toys. Science is going through a real big expansion in technology at the moment, and there are new things available. A toy that we've got is a 384-96 well confocal microscope, which allows us to image bacteria at really high resolution and understand things in a way we weren't capable of before. This is a single image of a Klebsiella pneumoniae, a single clone of Klebsiella pneumoniae stained with a single antibody. And you don't have to be a trained expert to realize there are so many different phenotypes just on one plane of bacteria. And also, quite conveniently now, because we've got it to work, is we can do this in real time. And also then we can actually grow the bacteria and understand how resistant phenotypes emerge in real time, but also how particular subpopulations expand within this plane over a given period. So just a couple of where we're with this. So this is a relatively simplistic that we're just about to publish now, of using machine learning. Each time we take an image of a single microorganism, we take more readings of every bacterial cell. Expose these organisms to different concentrations. We grow them over 24 hours, and we image them over 24 hours. That's trying to understand the morphological impact of what the drug is doing to the bacterial cell. And we can pick out variables, as shown here, 10 of them as it happens, that depict how the bacteria is going to behave when we expose it to a drug. Now, there's a lot of information on this slide, but to cut a long story short, we can predict whether a salmonella organism is resistant or susceptible to ciprofloxacin without ever exposing it to ciprofloxacin just by imaging it. So we can take a detailed image of a microbe and predict how it will predict to a drug or potentially any future perturbation just by taking a picture of it from having a large amount of data. So something else we've been doing then is thinking about immunizations. This, as I alluded to, bacterial vaccines are a bit of a backwater. We've been using outer membrane vesicles, and we can demonstrate quite nice kind of cross-reactive protection by simply immunizing mice with outer membrane vesicles purified from our collection of different Klebsiella that represent a different catalog of sequence types, but also K types and O types. And we can put this information back of our polyclonal sera back onto our organisms and image them and understand antibody binding. So the picture on the right there is all of our 180 or whatever they are, 200 Klebsiella pneumoniae, and across the top there are our different, or across the bottom, are our different polyclonal sera from immunized mice populations. So we can use this as a surrogate for understanding polyclonal antibody responses that we think are largely protective, and how much cross-reactivity we get, and how many different combinations of outer membrane vesicles we have to purify and mix together to make a cross-reactive Klebsiella vaccine, for example. So these are things that relatively kind of modest technology, using new approaches, we can understand to do this. So really taking on the kind of vaccine field and using more applied techniques in the laboratory to understand how we generate the appropriate immunological responses we need to do. Something I'm particularly excited about, and hence my reason for wanting to work more with IAVI, is the use of monoclonal antibodies. So whilst working in Vietnam, we identified a clone of Acinetobacter baumannii that was associated with infections in our ICU. Through a collaboration with a friend of mine, we isolated one of these bacteria, we purified outer membrane vesicles, and we immunized chimeric mice containing a humanized B cell repertoire. So these mice produce human antibody. So as shown from this sun plot here, we get really nice germline proliferation of certain clones of these B cells, and we can generate really nice binding antibody, human antibody, IgG, against these Klebsiella organisms. And this graph here shows we can also get complement deposition. Going back to our imaging, we take our collection of Acinetobacter, also a highly variable organism. We purify our monoclonal antibodies, and we stain them and measure for binding antibodies against the surface of the bacteria. And we can identify clear clusters of organisms, as those highlighted in black there, that have antibody binding, and also a main cluster here highlighted in green, which we think is probably an important target, which happens to be the capsular antigen of a particular variant. And again, to cut a long story short, in the interim period, updating our culture collection, including a study we did on neonatal sepsis with a new collection of Acinetobacter baumannii, taking one of these and setting up a mouse challenge study, we can show not only a dose response, but also a complete blocking of infection with a single intravenous monoclonal antibody before a 24-hour challenge. Meaning we can protect a mouse against not only lung, but also systemic Acinetobacter spread by one of our monoclonal antibodies that we put through this system. And this is our first go. And I think, therefore, monoclonal antibodies offer a big potential for doing things slightly differently. Diagnostics. Diagnostics is something that I know is very strong in this organization. And again, something that I see a big opportunity to change the way we're doing things. So this then, I have some other information, but this will be the last kind of thing that I will finish with saying. So I don't see a reason for developing diagnostic platforms for individual organisms. Whatever we do that's diagnostic needs to be ability to identify as many things as possible in whatever body sample we're looking for, viruses, fungi, bacteria. Now, this then is a DNA nanobase system whereby we make the actual DNA ourselves, and then we pull out small RNA populations and feed it through a glass nanopore. And then we can detect variation or fluctuation in the voltage, which will tell us what is in that sample. This is enzyme-free, so therefore we don't need a freezer for the samples. You can run it off a car battery, and we can detect 220 different RNA populations in an individual sample. We've done it for COVID, and we can detect different COVID variants, and also with a recent kind of breakthrough in our ability to identify 16S RNA targets, we can now do it for bacteria. So it takes 5 minutes. It can identify 220 different targets. It's enzyme-free, and you can run it off a car battery. Suddenly changing the paradigm of not only trying to grow or identify particular pathogens by using whatever lateral flow system or whatever we want, but also a mechanism to really change the game in ability to identify what's in what sample. So the last thing I'm going to say is then some work on typhoid fever. We've also contributed to typhoid fever by understanding not only surveillance mechanisms through genomics, but also then using this information to try and identify alternative targets. We've identified a new oral carbapenem We can debate the use of oral carbapenems for treating community-acquired infections later, but we've got a drug that will work against potentially treating XDR infections. And also then we now have the ability to identify people that have seroconverted to typhoid rather than naturally been exposed. And if I just whiz through these slides because I've overrun my time, this is the last slide. We've worked backwards and identified a number of immunogenic antigens on the surface of the bug, which we think are evidence of seroconversion for this particular pathogen. Now, why is this useful? This is useful to determine disease prevalence with a disease which is difficult to diagnose by a lack of sensitivity around blood culture. So if we want to introduce a vaccine into a region where we think we may have typhoid fever, we now have the ability to measure antibody as an indicative marker that someone's been exposed to it. And from a study that we did in Dhaka here, the incidence or the crude incidence is 417 per 100,000 children aged 0 to 4 years. Just as a kind of example of what we're showing, and this has been cross-validated, we estimate the incidence to be closer to 85,000. So in a year, 85% or seroconverting to typhoid fever. So therefore, independent of and a mechanism to introduce vaccine. AMR, we know that vaccines are important for limiting disease, and the more vaccines we can use to prevent disease caused by typhoid or other organisms by reducing antimicrobial use. So just to summarize, I'm a bit of a kind of whistle stop. I'm an applied microbiologist interested in. We know that AMR is a big challenge. We need policy to shift to think about how we can apply some of these things at a bigger scale. There's a whole host of possibilities. The only good thing to come out of the pandemic is the fact that actually now some of these technologies are more widely available, and we in our field should plug into them. And some mechanism to think about how we approach this challenge, repurposing what we have, new diagnostics, new vaccines, new epidemiological approaches, including seroepidemiology and the use of monoclonal antibodies. And also, we really require some new direction and innovation that clearly isn't going to be supplied by the pharmaceutical industry, and us as kind of academics need to really challenge forward. So I'd love to say I've done all this myself. I haven't. There's been a lot of people I've worked with over the years, people at Cambridge, my new employers at IAVI, the people, the folks when I was previously in the Oxford units working overseas, and the various academic and kind of industry collaborations I have, and these various organizations that have been generous to fund some of this work over the previous years. So that's me done. Thank you very much.
Thanks, Dr. Vikas. Next speaker.
So on behalf of the organizers, I take this opportunity to invite the next speaker, Dr. Datun Mortimer. She is currently serving as assistant professor in the Department of Population Health at the University of Georgia College of Veterinary Medicine. And she has lots of fantastic research experience. I'll not go into the detail, but her today's talk title is Genomic Analysis of Antimicrobial Resistance, Diagnostics, and Interventions for Neisseria gonorrhoeae. Thank you.
Hey everyone. First, I want to say thank you so much for inviting me to the talk, and thank you to the organizers for this conference. Today, I'm going to tell you all about our work using whole genome sequencing data from Neisseria gonorrhoeae to understand the evolution of antimicrobial resistance and the potential impact of new diagnostics and interventions. So Neisseria gonorrhoeae is a gram-negative Diplococcus. I will probably also refer to it as gonococcus for the rest of the talk. It is the causative agent of the sexually transmitted infection gonorrhea. And during uncomplicated infections, it infects mucosal surfaces in the rectum, urethra, cervix, and pharynx. However, it can also cause more severe disease like pelvic inflammatory disease, conjunctivitis, and disseminated gonococcal infections. And in the United States and also worldwide, we've seen a large increase in incidence in the last decade, with over 80 million infections estimated to be reported by the WHO in 2020. Antimicrobial resistance in Neisseria gonorrhoeae has also been deemed an urgent public health threat by both the CDC and the WHO. Like many other pathogens, gonococcus has acquired resistance to many classes of antimicrobials shortly after they're introduced into clinical use. So you can see that in the timeline on the left, where light blue is the first clinical use of an antibiotic and purple is the first report of resistance to gonococcus. Well, most gonorrhea are treated empirically, and that's because they're diagnosed acid amplification tests. So we're really just looking at the presence of Neisseria gonorrhoeae in a patient sample. We don't really know a lot about susceptibility of a particular in the clinic. And of the population is resistant to a particular antibiotic, that antibiotic is no longer used for treatment, which means that for most of these drugs that you can see in the darker blue color, that we no longer use them in the clinic for treatment of gonorrhea. And while there is geographic variation in the prevalence of resistance to different antibiotics in gonorrhea infections, we also see internationally transmitting MDR strains. So for example, just this year, there have multiple reports, including in what used to be my backyard of Massachusetts, that there is a strain of gonorrhea that has reduced susceptibility to ceftriaxone, which is our currently recommended therapy, as well as resistance to every other drug we've ever used to treat gonorrhea. And has been nicely demonstrated already in this conference. We really don't have a lot of options left. There's not a lot of new drugs being developed. And so it's very important that we understand how to more effectively use the antibiotics we have to treat gonorrhea. Now, I've told you that there's an increase in incidence in gonorrhea and also an increase in resistance. And you might imagine that this increase in resistance is really driving the increase in incidence. For example, if patients aren't being treated effectively, that really presents an opportunity for ongoing transmission. However, some work we did in collaboration with the New York City Department of Health and Mental Hygiene showed that antimicrobial resistance doesn't actually seem to be the primary driver of gonorrhea transmission. So here on the right, I'm showing you a phylogeny from almost 900 gonorrhea isolates collected from sexual health clinics in New York City. So each tip on the phylogeny represents one isolate and the branch links represent genetic distance. Using this information, we can estimate transmission clusters, which I've annotated the 10 largest clusters that we found around the outside of the tree. And if you look at minimum inhibitory concentrations for ceftriaxone, azithromycin, and ciprofloxacin in these transmission clusters, they're susceptible to all of these drugs. So at the time that these were collected, empiric therapy was ceftriaxone and azithromycin, and the previous therapy was ciprofloxacin. So, you know, these lineages are not transmitting because they're resistant to our therapy. All of them were treated. And this really highlights 2 points. The first is that while drug resistance is gonorrhea, there are susceptible strains, and we're really looking our sort of antimicrobial options by empiric therapy. So to know something about susceptibility in the clinic, we could potentially reintroduce antimicrobials that we don't use anymore. And second, we really need some new interventions to prevent the transmission of both susceptible and resistant gonorrhea. So the gold standard for measuring susceptibility in gonorrhea is really culture. However, it's not feasible to do culture for every gonorrhea infection. It takes a few days and requires some specialized skills that many laboratories, at least in the United States, don't have anymore. And so what would be ideal is if we could have a point of care diagnostic using genomic material from Neisseria gonorrhoeae to predict susceptibility. Now, our work, as well as many other groups, have shown that if you have whole genome sequencing from Neisseria gonorrhoeae, you can predict minimum inhibitory concentrations pretty accurately. We really have a pretty good understanding at this point of the genetic underpinnings of resistance. So you can see here in this phylogeny of a global collection of isolates that, for example, the GYRA91S allele, which is in light green, really maps well with ciprofloxacin susceptibility. And that's great because there, in fact, is a diagnostic developed by SpeedX called Resistance Plus GC, which looks at this particular locus to predict ciprofloxacin susceptibility in gonorrhea infections. However, the picture for other antibiotics is not quite so simple. While we can predict MICs based on whole genome sequencing data, single locus for any of the other antimicrobials doesn't predict with high sensitivity or specificity. And while I think it would be great if we did whole genome sequencing every 80 million gonorrhea infections, from a research standpoint, that's just not feasible in most settings. And so we need to develop diagnostics based on a few loci for these drugs. The other reason it's very important for us to develop new diagnostics is that modeling data has shown that introduction of just a single susceptibility diagnostic for one drug might actually inadvertently drive multi-drug resistance. What are our other options besides ciprofloxacin? We could also consider penicillin and tetracycline. These are really the backbone of gonorrhea treatment for decades until around the mid to late 1980s when resistance emerged. However, like I just mentioned, we really can't predict susceptibility to these two drugs with just a single locus. And that's because there are multiple pathways to resistance. There's high-level resistance that's mediated by either plasmid-encoded beta-lactamase in the case of penicillin or TET-M for tetracycline. But there's additionally chromosomally encoded resistance, which is mediated by a combination of variants in antibiotic targets, efflux pumps, and the porin. And so our goal is really to identify variants that predict penicillin and tetracycline susceptibility with the idea that we could find a locus in the to combine with plasma for a potential diagnostic. So to do this, we use genomite association study, incorporating the presence or absence of these plasmid-encoded high-level resistance genes. So we use a curated collection of genome assemblies, as well as our phenotype, which in this case is penicillin or tetracycline MICs. We summarize all of the genetic variation present in the population, as well as the presence or absence of these AMR plasmids, and we estimate a phylogeny. And then we look for significant associations between each variant and our phenotype using a linear mixed model, which incorporates that phylogenetic information to control for population structure. So we can look at the significance of our variants in something like a Manhattan plot, which I've shown an example here. But the other thing I wanted to mention is we're very interested in looking at the effect size of these variants, which is in the model represented by beta. So because we're interested in developing a diagnostic specifically to predict susceptibility, we're looking for those variants that are associated with that have an estimated negative effect size. So here's the result of our penicillin conditional GWAS using almost 7,000 isolates along with penicillin MICs. And what you can see is that we have three major peaks of significance. One in MTRC, which is a component of the major antibiotic efflux pump in gonococcus. One in POR-B, which encodes the porin, and then lastly, a peak in Pen-A, which encodes the target of penicillin, PVP2. Now, I told you that we already have a pretty good idea about the genetic underpinnings of resistance in gonococcus, and so these peaks were not surprising to us. This is what we expected to find. And we decided to really concentrate on the variant that had the greatest impact on susceptibility. And so in this case, the sequence, which we just named Pen-A01, actually differentiates the susceptible allele from two different known resistance mechanisms for penicillin. The first is this insertion at codon 345, but it also differentiates the susceptible allele from mosaic alleles, which have been acquired from other Neisseria species. And so by just looking at this for this one sequence, we can differentiate susceptible alleles from multiple resistance mechanisms. We combine that sequence with the presence or absence of our beta-lactamase encoding plasmid, we can predict penicillin susceptibility with very high specificity. However, sensitivity, I would say, is a little more moderate in both our global data set, which we use for the GWAS, as well as a validation data set of representative isolates collected through CDC surveillance. I'm not going to go through all of the results for tetracycline, but we see a similar picture where we found that RPSJ, a particular sequence in RPSJ, which is the target of tetracycline, combined with the absence of TET-M, can predict tetracycline susceptibility with very high specificity. Sensitivity for tetracycline is a little better than penicillin with just these two loci in both our global and validation data set. So we're actually working with some folks at the Broad Institute to see if we could develop a diagnostic based on these two loci. So some short conclusions from this part of the talk is that for both penicillin and tetracycline, two loci can predict susceptibility with high specificity, but only moderate sensitivity. We added in additional loci, we could increase that sensitivity. And also that this conditional GWAS approach focused on variants associated specifically with susceptibility may be a promising approach to identify loci for molecular diagnostics and surveillance in other pathogens as well. I told you earlier in the talk that in addition to using our current antimicrobials more effectively, we also need to come up with some new interventions to control the transmission of gonorrhea. So I thought I would highlight a couple interventions that I think are, have made the news recently, and I think are likely to be introduced or are being introduced in the clinic now. The first is vaccination. So people have been working on developing vaccines for gonorrhea for a long time with not a lot of success. That's because gonorrhea infections do not produce natural immunity, and the immune evasion strategies used by gonococcus also hinder development of an effective vaccine. However, a few years ago, some retrospective analyses of a vaccine to a related bacteria, Neisseria meningitis, showed some maybe 30 to 40% effectiveness against gonorrhea as well. And so there are actually ongoing clinical trials to directly test whether this vaccine is effective against gonorrhea. But on my way here, I was reading that even though these trials aren't quite finished yet, a committee in the UK has recommended that people at high risk for gonorrhea have this Bexsero vaccine. And then the other intervention is post-exposure prophylaxis. So in addition to gonorrhea increasing in incidence, other bacterial STIs like chlamydia and syphilis are also increasing in incidence. And susceptible isolates to all of these are effectively treated by doxycycline. And so post-exposure prophylaxis recommends that someone at high risk for contracting gonorrhea take a single dose of doxycycline within 24 hours up to 72 hours after unprotected sex. Unfortunately, doxycycline PEP is a little less effective for gonorrhea. However, it is still being introduced into the clinic because of its effectiveness against chlamydia and syphilis. So we were curious to understand what the impact of doxycycline will be on AMR in Neisseria gonorrhoeae. Like I said, doxycycline PEP is not as effective against gonorrhea compared to other bacterial STIs, and this is because pre-existing tetracycline resistance exists in the gonorrhea population. We don't have any official breakpoints for doxycycline resistance, but it seems like tetracycline resistance and doxycycline MICs are going to be similar. And so in this study, we just use tetracycline as a proxy for doxycycline. In this larger data set, we found that known loci also explain tetracycline resistance well, similar to what we had seen in other studies. But most importantly, what we found is that co-resistance with other important antimicrobials really is linked to particular tetracycline resistant loci. So high-level resistant isolates, those with the plasmid, actually don't often encode resistance to other antimicrobials, while those with chromosomally encoded tetracycline resistance often are linked to resistance to tetracycline, azithromycin, and ceftriaxone, meaning that selection for tetracycline resistance by doxypep may also change the AMR landscape for these other drugs as well. So we see this in a global data set as well as in the United States. So for example, we don't have a lot of ceftriaxone resistance in the United States yet, but all of the ceftriaxone resistance that we have seen has exclusively emerged on backgrounds with chromosomally encoded tetracycline resistance. We see differences in the potential effectiveness of doxypep across patient populations and across geographic regions in the United States. And so in conclusion, this near-term impact of doxycycline PEP on Neisseria, gonorrhoeae, and AMR is likely influenced by the string selection for TET-M mediated high-level resistance, just because TET-M doesn't seem to be as linked with other antimicrobial resistance in the gonococcus population. I do want to emphasize that this is a near-term impact, as gonococcus is extremely recombinogenic, and so we might see reshuffling of this linkage or introduction of new strains. We think that doxypep is likely to vary in effectiveness across patient populations, and we really need genomic surveillance, specifically among doxycycline PEP users, to understand this impact. So just to summarize, AMR doesn't seem to be the primary driver of Neisseria gonorrhoeae transmission, although it is obviously a big problem with some multi-drug resistant strains encoding resistance to almost all of our available options. We can use GWAS to identify targets for antimicrobial susceptibility diagnostics, and this impact of doxycycline PEP on AMR is really going to depend on the strength of selection for TET-M. So I will finish with just some acknowledgements. While I'm at University of Georgia now, most of this work was done when I was a postdoc in the Unitime Grads Lab at the Harvard THGM School of Public Health. So I'd like to thank everybody who worked with me on the project in that lab, and as well as all of our collaborators who have collected and helped us sequence all of these isolates, and the people who maintain our fabulous publicly available resources, without whom these GWAS and doxypep studies would not be possible. So thanks, everyone.
Thank you, Dr. Mortimer. Myself and on behalf of my co-chairs, we thank all the speakers of this session and for their excellent work and presentation. Now the floor is open for discussion. I think must be certain questions, so that might be asked. Please introduce yourself to ask me the question.
Hi, I'm Manshu. I'm a postdoc in Sweden, and I have a question for Dr. Stephen Baker. Okay. Maybe another question for so you talked about prediction, this fascinating thing about being able to predict the MIC based on the genomic sequence. Is it based on the copy numbers, or is it based on how much they express? How is it done?
Yeah, so the prediction is based on basically having all of the known antimicrobial resistance alleles, and then looking for them in genomes, and then using the presence or absence of those alleles to build linear models to predict MIC.
Thank you.
I'm Krish Atmakuri from HSTI. So I had actually a question for Tatum and Baker, but that he's not there. I'll ask you about this. So this is regarding the exploitation of the outer membrane vesicles as potential bacterial vaccines. So we do work with mycobacterial OMBs. And it's been always a challenge that one of the reasons why OMBs are not very well supported as a vaccine candidate is because of the antigenic variation or the load with respect to each of those vesicles. So what is your take on it?
I have to say I am not an expert on gonococcal OMVs, although Sanjay Ram might have a better answer for you about vaccination in gonococcus.
Can I ask one more question? I have one question for Dr. Mathur. So this is regarding in terms of when you're looking at the surveillance across the hospitals. So have you been able to identify some of the potential reservoirs in these hospitals, which is sort of common and which is distinct across different surgical units?
So not at a network level, but through the Center for Advanced Research, we are one of the main focus is looking at the environmental reservoirs and niches in the hospital, especially for Klebsiella and Acinetobacter. So we have been doing a lot of work on understanding the reservoirs for infections in ICUs and surgical units, and we found that it is mostly water. The biofilms that are formed, because although Acinetobacter survives on dry surfaces also, we found that we've had a couple of outbreaks related to water. Even the effluent treated water from many of these hospitals is known to, we found that the copy numbers of these pathogens is immense, even after the effluent has been treated by ETP. So we know that all hospitals are supposed to have effluent treatment done. So this water after treatment is sprinkled everywhere and it is pushed into the municipal water. So all these bugs are actually going through the sewer lines. So it is basically, and the same thing during monsoon, we find that these sewer lines and the water lines they have these small leakages. So it is the main reason in Indian hospital is water. And of course, we find that high touch surfaces, the bed railings and the lockers and everything are heavily, colonized with these MDRs. But where does it come from? It is the gut colonization, of course, and the water. So we've seen sequentially that within 5 to 6 days, the gut flora changes and it becomes highly resistant. So gut colonization and water is the weakness.
Thank you.
Dr. Ganguly, my question is to Dr. Mortimer, that you made a statement that AMR is not responsible for transmission of Neisseria gonorrhoeae. Up to now, our main hypothesis was in the STI, that drug resistance Neisseria gonorrhoeae is a real major problem. And my another question is that what's your position in doxycycline? Because this particular dogma is prevalent very strongly and people use it. So could you make a clear-cut statement that whether doxycycline should be or should not be as an after pill?
Sure. So I think that for STIs, doxycycline post-exposure prophylaxis, at least in the United States, it's being introduced into clinics, and CDC is working on guidelines, but even without official guidelines, people are asking for doxycycline post-exposure prophylaxis. Some modeling work out of Yonatan Grad's lab has shown that the excess use of doxycycline because of doxycycline PEP, it's going to be enormous. And I think that there probably will be impacts on resistance for certainly gonococcus, but other organisms as well. But it also is clearly extremely effective against chlamydia and syphilis is increasing in incidence and has a very severe impact, especially neonatal impact. So I guess that's probably not a clear-cut statement, except to say that the doxycycline PEP is happening. And so we just need to be prepared for the potential impact.
So thank you for the talks. I'm Liliana Brown from NIID. My question is for Dr. Mathur regarding the hospital surveillance and the way that the data are shared to the database. Are they anonymized per hospital? We see a lot of reticence in the United States from hospitals wanting to share this type of data. They don't want the public knowing what level of infections are occurring, especially drug-resistant infections. So I'm wondering how those negotiations have been, what kind of data, what level of anonymity exists at the hospital level.
Yeah, thank you for that question. And this was the major icebreaker when we started this surveillance. The main query or anxiety amongst the hospital was their data going public, or even within the network, the hospital knowing the other hospital's rate or their own hospital's rate being made available or known to other network hospitals. So it's all anonymized. Every time a new hospital is included, they are trained first, and then they are given a code. That code is known only to the central team at our hospital. No other hospital knows the code number. And within the hospital, the various ICUs are given subcodes. So even in an institution, supposing there is a gastrointestinal ICU and a neurosurgical ICU, even within an institution, the various ICU in charges don't know each other's code. So everything is anonymized. The kind of data which goes out into the ICMR's domain is all generic. There is no individual hospital. We just give it according to how many pediatric ICUs, how many neurosurgical ICUs, medical ICUs. So the idea is that a medical ICU in India can compare the rate with other medical ICUs somewhere in the, and probably down the line, we would like to analyze it geographically, that what is the rate in North and South, but not beyond that. So it's all anonymized coded data. Which is good. And every hospital can see their own data in the database, the graphs and the trends, but only the central team, the ICMR and LCTC can see the entire network data and the graphics and everything.
So my question is Dr. Tatum. So I'm Nishit from THSTI. So you have shown the GWAS-based studies to diagnose the drug-resistant gonorrhea. So I was wondering, like, is this based on the study localized in US, or you have also identified, this trend in the different geographical locations?
Sure. So when we do our genome-wide association studies, we generally try to include all of the available Neisseria gonorrhoeae sequencing data. However, so that comes from many different geographic regions. However, I will say that genomic surveillance for gonococcus is definitely geographically biased. A lot of these sequences are coming from either the United States, the UK, or Australia, and sequences from other parts of the world are less represented in the data set. And so I think it's certainly the case that, there might be I think the loci we're finding are probably generally applicable across Neisseria gonorrhoeae, given how well these strains transmit internationally. But I think we could do a much better job at genomic surveillance globally.
Thanks.
Hello.
Dr. Shanta from ICM and NICET. My question goes to Dr. Mathur. It's a very wonderful study, and altogether you have shown all the hospitals are actually responsible for this infection. So there must be we all know that hospital control infection committee is present in almost in all hospitals. So it is a huge task to control this infection. What is your recommendation, like how to start with and how to go ahead with to control the infection?
So I think infection control is more like a behavioral science and a teamwork. So all these hospitals which are part of our network, when we start enrolling them, we do a baseline assessment, and one of the major component or most essential is that they have to have some kind of infection control committee already existing, because there is no point in doing all this exercise unless there is a loop closure. As I said, that all this data that is coming has to be used for infection prevention and control. So an infection control committee, which is multi-sectorial with all the surgeons and medical people and microbiologists and the nurses, that is mandatory. And then looking at your data, that whether your rates of CLABSIs are high or UTI are high or VAP, in which ICUs, ultimately infection control is also requires a lot of manpower and time. So you have to use your own data and see which are the ICUs which have a higher infection, and you can focus on that, and you can improvise your own preventive bundles. That is available in the literature, but you just have to take out which particular maneuver or protocol is applicable to your setting, given the resource limitations and the manpower, and select that bundle and implement. So it's a continuous science, but yes, infection control committee is very important for preventing HAIs.
No, but there are recommendations, but it is not an easy task to implement all those.
It is not an easy task, and it's always a top-down approach that it has to come from the administration. So what we have done at the AIMS Trauma Center is that the chief of trauma center is involved, and so it has to be a top-down approach. The administration has to be a part of that, and for that, you have to give these reports that what are the rates. Unless you have a communication, you give these rates to the administration, there will be no support and implementation.
No. Hello. This is hello. Hello. Yeah, this is Trust. This is Dr. Madhav from Scan Research Trust. My question is from Dr. Mortimer, that you said that whole genome sequencing can predict the minimum inhibitory concentration, but doing the whole genome sequencing of all the bacteria is not possible. But can the same thing be done using whole genome metagenome sequencing?
Please.
So my understanding of using metagenomic sequencing for gonococcus specifically is that just the coverage has not been very high so far, but perhaps advancement in technology can make that better.
Okay, thank you. And thank you for this nice discussion. And thank all the speaker and audience for this excellent session. Thank you.
Thank you to all the session chairs and the speakers for the highly informative session. So now we may start with the felicitation ceremony. I request Dr. Rupa K. Bhadra to hand over the memento to Dr. Purva Mathur.
Dr. Tushar K. Mehti to Dr. Tatum Mortimer. Now I request Dr. C.V. Shrikant to kindly come up on the stage to felicitate the stage session chairs. Sir, please. First, for the session chairs, Dr. Rupa K. Bhadra. Dr. Ranjan Nanda. Dr. Tushar K. Mehti. So we may have a group photo of this session. Thank you, ma'am. Thank you, sir. So we now break for lunch. All the conference volunteers are requested to assist the dignitaries to the area for lunch. We request everyone to join us for the lunch and stall visits outside the auditorium. And the poster participants are requested to be present near their respective posters for the evaluation. So all are requested to assemble back by 2 p.m. for the start of the next session. Thank you.
Hey, hello. Check, check, check.
Hello.
हेलो हेलो 1 2 3 सर आ रही है आवाज सर वो नहीं बीप हेलो हेलो 1 2 3 माइक टेस्टिंग 1 1 1 नहीं आ रही ना
मैम बैठ गया मैम मैम का कोई इलाज नहीं
I request everyone to please take their seats in the auditorium. We shall begin our session soon. I again request everyone to kindly take their seats in the auditorium, please. Welcome back, everyone. We hope you all had some engaging discussions over lunch. We now begin our scientific session 3 of the conference on the theme Emergence and Dissemination of AMR, New Concepts and Evidences. For this session, I request Dr. N.K. Ganguly, Dr. Arvind Sahu, and Dr. Debashish Mohanty to kindly chair the session. Sir, I request you to kindly join us on the stage, please. Dr. Nirmal Kumar Ganguly is the former Director General of the Indian Council of Medical Research, ICMR, and was also formerly a distinguished biotechnology research professor at the Department of Biotechnology, Government of India. He has also served as the president of the Jawaharlal Institute of Postgraduate Medical Education and Research, as well as that of the Asian Institute of Public Health, Bhubaneswar. Dr. Ganguly has over 40 years of research and training experience, and his major areas of research have been tropical disease, cardiovascular, and diarrheal diseases. He is the recipient of the prestigious Padma Bhushan Award in the field of medicine. It is an honor to have you with us here today, sir. Dr. Arvind Sahu is the Executive Director of the Regional Center for Biotechnology. Prior to this, Dr. Sahu has been with the National Center for Cell Science, Pune, for over 2 decades. Dr. Sahu's research is directed towards understanding the role of virally encoded complement regulators in viral pathogenesis. He has been awarded the prestigious J.C. Bose National Fellowship in recognition for his outstanding research in the field of immunology. Thank you, sir, for joining us here today. Dr. Mohanty is the director of the National Institute of Immunology, New Delhi. His research is focused on understanding the structural principles that govern sequence-to-structure relationships in proteins and binding of various ligands to proteins. Dr. Mohanty and his team develop computational tools to assign functions to putative proteins that can help in identifying protein interaction networks and novel metabolic pathways. Dr. Mohanty is an elected fellow of the Indian National Science Academy, the National Academy of Science, and the Indian Academy of Sciences. He is also the recipient of the National Bioscience Career Development, sir. Welcome, sir. In this session, each talk will be for 15 minutes. After all the speakers have delivered their talks, there will be a 10-minute long question and answer session. We request our audience to kindly reserve their questions until the session is completed. I now request the session chairs to introduce the speakers and call them over to deliver their talks.
Thank you. And we start this session thanking Dr. Bhavatosh as well as the THSTI. Our first speaker is Dr. Ramamurthy. I know Dr. Ramamurthy for I don't know how many decades. He did his PhD from Annamalai University, and then he joined NICED with Dr. J.B. Nair's group, and ultimately took up cholera as his major, Vibrio cholerae as his major area of research, was trained in Japan also, and he created a very good library of the Vibrio cholerae strains. And as we know that one of the major problem in Vibrios are the AMR, and there's some in Africa created when the first the hybrid Eltor came up with a lot of drug resistance. The Sam Karaoke and Gordon Douglas Gordon Dugan, they worked out the network of that particular spread of resistant cholerae. So he is one of the most erudite surviving researcher in the area of Vibrio cholerae and the drug resistance in Vibrios as well as other diarrhea causing organisms. So Dr. Ramurthy, I invite you to give your talk.
Thank you very much, sir, for your kind words. But today I'm not going to talk about cholera, but very suffocating talk that is on anaerobic bacteria. So how this anaerobic bacteria plays a role in the dissemination of antimicrobial resistance, as well as what are the governing factors that is responsible for spreading the disease. So in the next slide, I will show you the total mechanism of spread of drug resistance. Mostly the microbial community plays a good role in spreading the species to interspecies or intraspecies spread of antimicrobial resistant genes, as well as other virulence factors also we studied. And next comes the environmental factors, known and unknown, plays a good role in the spread of antimicrobial resistance. And the mobile genetic elements, as usual, this is responsible for the spread of the antimicrobial resistance encoding genes, especially both for both to the aerobic as well as anaerobic and bacteria. Just to recapitulate, what is the difference between the aerobic and anaerobic? So everybody knows oxygen stress is more responsible, but the other factors includes, apart from oxygen, the citric acid cycle, as well as the oxidative phosphorylation is very important for the aerobic bacteria, and for the anaerobic bacteria, it produces lactic acid in the anaerobic environment. And due to this lactic acid production, so most of the distillery units exploits this mechanism, especially to produce ethanol, and also the methane and sulfur reducers are responsible in the anaerobic environment. If the aerobic bacteria exposed to the anaerobic conditions, what will happen? Maybe this is a very time-bound effect. So it influences antimicrobes the anaerobic or the anaerobic conditions. Just I'm giving two good examples. One is the Pseudomonas aeruginosa. Due to O2 starvation, it develops, it reduces the effectiveness of the antimicrobes. There are so many antibiotics. And the other good example, my favorite organism, the Vibrio cholerae, if it exposes to the anaerobic environment, maybe mimicking the gut environment, it the minimal inventory concentration goes up. Usually this Vibrio cholerae is susceptible if you do the antimicrobial assay in the aerobic conditions. But in the anaerobic conditions, the ciprofloxacin tolerance increases 10-fold and 100 times in the azithromycin. That's why the azithromycin susceptibility is getting reduced, and most of the Vibrio cholerae strains, as well as the other entry bacteria nowadays, are reduced susceptibility, means less sensitive. And most of the commensals are less susceptible to gapromycin and ciprofloxacin. These are all non-pathogenic. Quickly go to the mechanism how the aerobic or facultative anaerobes adopts the situation is the cytochrome oxidase gets increased, and due to that it uses the nitrate and nitrate reduction and use this nitrate as an energy source or oxygen during respiration. Just to quickly, I'll go through this slide. So it produces anaerobic bacteria produces or associated with important diseases in the aerobic environment. Say, for example, gum or in the lung or in the blood. These are all actually oxygen saturated regions, but the anaerobic bacteria invades into the tissues and survives and causes the infection. And also the genitals. Most of the one special session will be there after this. So they'll be covering more on the reproductive organ related infections. So this slide just shows or recapitulates how the trends of antimicrobial resistance. It increases most of the antibiotics. And in some cases, in some species, of course, there are overlap of the data. And mostly, these are all highly stable. Anaerobic bacteria infection, what are the challenges? So the two major challenges is, this is a very difficult organism to handle in the lab. And the identification is a big problem. And selecting the optimal therapy for the infection is also very, very difficult. Other practical difficulty is the two existing gold standard, the UCAST and CLSI guidelines. There is no consistency in declaring this is the cutoff point to say this is resistant or susceptible. So especially in developing countries, we face the problem, which one to adopt. The common infections caused by different species of bacteria is shown here. Usually this is bacteremia. So these are all, we cannot say it's a demarked pathogen like a Salmonella or Shigella or Vibrio cholerae like that. Mostly these are all commensal bacteria, but somehow it adopts and became resistant to so many antibiotics due to the existing pressure. So Bacteria fragilis, we have a parabacteriosis and also Provitella. So this causes so many infections. This is all very, sometimes it is dangerous. In real clinical scenario, mostly for bacteremia or brain abscesses or wound infections, the conventional method is to isolate the organism and do the susceptibility assay. But if this is the second column shows These are all facultative anaerobes, E. coli or Pseudomonas aeruginosa or Enterococcus. And most of these are susceptible to ciprofloxacin or amikacin or gentamicin or to ofloxacin groups. But in the same scenario, the same say for bacteremia, there are associated pathogens or bacteria, fragilis, the one that is marked in red or highly resistant. Whereas if we identify the other bacteria as a sensitive, we have the background of anaerobes that is resistant to the given antibiotic. So this is a very warning scenario. So these are all some of the very common resistant genes that has been identified in anaerobes, especially to Clostridium difficile or Bacteroides fragilis, as well as Provitella. The NIM gene, as well as the CFXA or the CFIA, these are commonly present, mostly present, I won't say common, so mostly present in the anaerobic bacteria. So what will happen if normal person takes antibiotic for some treatment or from the recovery from other infections? So one study conducted in 2015, which shows the two groups given 15 numbers each, male and female, including placebo. So one clindamycin group, other is the ciprofloxacin group. So if they are given, they are administered about for 10 days to a fixed dose of antibiotic. And then the volunteers followed for about 1 year. And the clindamycin group, Provitella in saliva, and the Bacteroides species in the fecal samples, it existed resistant to clindamycin group. Whereas in ciprofloxacin group, the other group of bacteria. So means this resistant power has extended beyond 1 year study period. These are benefits of antibacterials in the synergy. So that sometimes antibiotics or enhances the biofilm formation and also the virulence, and synergy also take place between some species of bacteria, Bacteroides fragilis and Neisseria gonorrhoeae, together in the pelvic inflammatory field. So again, to lack of time, not explain too much. The mobility of several genes are occurring in the anaerobic environment very fast between bacterial species, and also the tetracycline resistance that integrates in the chromosome of another same species or another species of bacteria. And anaerobes are also present in the food samples. And the resistance to anaerobes from the food is not, this is a very challenging problem because the conventional antibiotic resistant genes are lacking. Environmental reservoir, just quickly I'm describing. This is the treatment process, the pollution treatment or the waste treatment, the pre-treatment, anaerobic digestion, and post-treatment, where the anaerobic digestion is a very hot spot, especially in the exchange of or increase or reduction of antibiotic resistant bacteria as well as the resistant genes. So the last column shows the total ammonia, nitrogen, or potassium, or anaerobic waste storage, or oxygen concentration. These are all the factors responsible for the abundance of antimicrobial resistant bacteria as well as the genes. Integrons plays a very important role. This is the case in the aerobic bacteria also. The whole cassette may have about 10 to 12 resistant genes in the integron. So the anaerobic digester, usually several factors are responsible for reducing the antimicrobial resistant genes or the bacteria. Say, for example, removal of efficient bacterial taxa, production of methane or lipid stress or acidic pH reduces the antimicrobial resistant genes. And this is in developing countries, they use ultrasound or ozone to decrease the abundance of antimicrobial resistant genes or the bacteria. And some enzymes like cellulose as incorporated with or in combination with zero valent ion removes the antimicrobial resistant genes. However, there are a lot of other factors that promotes the abundance of antibiotic resistant bacteria or the genes. Say, for example, there are absorbed cellular genes or intercellular or extracellular antimicrobial resistant genes that that has been increased or abundance is increased due to the chemical oxygen demand or total nitrogen or due to heavy metals. And there are antibiotics that reduces the methanogenic bacteria. That is the native flora of the anaerobic digesters. So in the case of reduction of methanogenic bacteria, there is an increase in antimicrobial resistant bacteria. So this is my last slide, which shows the pharmacodynamics of pharmacodynamic properties of several antibiotics or antimicrobials against anaerobes or different compared to the aerobes. However, the clinical implication of these differences needs further exploration. I'm happy to see some of the young scientists are now started doing concentrating the research on anaerobic bacteria in this country, especially in THSTI, where doctors established the anaerobic system where they are doing the abundance of anaerobes as well as in relation to the antibiotic resistant. So thank you. Now we can have a good brief. Thank you.
We will take questions at the end of the session. This was a very unique talk because very few people work in this area, and you have not only emphasized the antimicrobial resistance properties and mechanism in anaerobes, but the hypoxic condition in the aerobes enhances the concentration and enhances also the populations. And he has started this. For those who are working on microbiome in this organization, they're using your anaerobic chamber now. So it's very good. Come, have a seat. I give it to Dr. Saud.
Yes, so the next presentation is by Dr. Ranjana Patania. Dr. Patania is a professor in the Department of Biosciences and Bioengineering in the Indian Institute of Technology, Roorkee. She leads the molecular bacteriology and chemical genetics group at IIT Roorkee. Before joining IIT, she was a postdoctoral fellow at the McMaster University in Professor Eric Brown's lab. And currently, her group employs two-pronged approach at IIT to tackle the antimicrobial resistance. And in the escape pathogen, the first one is involves the chemical genetics strategies to discover the novel antibacterials, antibiotic combinations, and the adjunct molecules. And the other involves the uncovering the small RNA, a mediated post-transcriptional regulatory network of pathophysiology in Acinetobacter baumannii in particular, which is the WHO classified top priority pathogen. So today her talk is going to be on the role of small RNA to mitigate antibiotic resistance. Dr. Patania, please.
Thank you for your kind introduction. And I would like to thank the organizers for inviting me. So I'll be talking on exploring the role of small RNA to mitigate antibiotic resistance. And my lab works on understanding the role of transcriptional regulators, non-coding RNA, other virulence factors in pathophysiology of escape pathogens with special emphasis on Acinetobacter baumannii. We also try to look at the role of various determinants of antibiotic resistance in bacteria. And depending on the knowledge generated on these fundamental aspects, we try to take these pathways and then try to explore small molecule libraries, chemical genetic approaches to discover novel antibacterials which target multiple pathways. We also try to look at and discover novel inhibitors of various determinants of antibiotic resistance like efflux pumps, beta-lactamases, carbapinamases, etc. And we're also trying to look at the combinations of antibiotics, both synergistic as well as antagonistic combinations. So depending on what we've worked in the last few years, we've generated some know-how, and based on that know-how, we've discovered some boronic acid-based beta-lactamase inhibitors, some antibiotic-antibiotic combinations, some molecules that target persistence in escape pathogens, some efflux pump inhibitors that revive the activity of antibiotics, and antibacterials that target multiple pathways. However, today I'll be focusing on the pathophysiology of this pathogen, Acinetobacter baumannii, which is a top priority pathogen. It is a gram-negative nosocomial pathogen that causes urinary tract infections, pneumonia, bacteremia, etc. And this is community-acquired pneumonia is predominant in tropical climate, and that's the reason, the two talks in the morning session, both of them mentioned Acinetobacter baumannii, goes to tell you about how important Acinetobacter baumannii infections are in terms of antibiotic resistance and carbapenem resistance for countries like India. And the strength of the pathogen comes from the fact that it adapts very swiftly to changing environmental conditions, such as inside the host, overcomes the nutritional immunity posed by the host, survives desiccation conditions in hospital setting, and the conventional pathways of gene expression don't control all of this. All of this is controlled by post-transcriptional regulation that mediates swift adaptation. At least in rest of the escape pathogens, we know that post-transcriptional regulation, because of its quick response, because of flexible regulation, because of genetic fine-tuning, these pathways, the riboregulation pathways, they confer pathophysiological fitness and adaptability to many of these pathogens. So in most of the other pathogens, most of the bacterial pathogens, it's known that riboregulation comprises of three major partners. One is non-coding RNA. Non-coding RNA, something that's shown here in red, non-coding RNA goes and binds to the target mRNA with the help of, now this binding is not perfectly complementary, and because of that, they need a chaperone, which is HFQ in escape pathogens, and based on whether they recruit RNase E or they protect the system from RNase E, the pathways can be turned off or on, and that's how most of the various physiological pathways are controlled in most of pathogens. But these pathways are not known in Acinetobacter baumannii. So my group, a couple of years ago, we characterized the unique HFQ in Acinetobacter baumannii, which is very, very unique in the sense that it has a unique C-terminal length. And we showed that this HFQ is essential for virulence in Acinetobacter baumannii in systemic models of infection as well as in pneumonia models of infection. And we are characterizing the non-coding SRNome of Acinetobacter baumannii, and we've identified and confirmed 31 novel small RNAs in Acinetobacter baumannii. And I'll be talking about two of these non-coding RNA today, since the session is focused on new concepts. So I'll try to show you how these control the fitness of Acinetobacter baumannii. So one of the small RNA, ABSR25, negatively regulates the expression of an efflux pump, which was designated as AIS1331 in Acinetobacter baumannii. When we induce ABSR25, the expression of ABF goes down. And the phenotypic characterization of this showed that we looked into the mechanism, and we found that ABSR25 downregulates this efflux pump. And when we knocked out the efflux pump, we found that the knockout was sensitive to phosphomycin. We went on and characterized this efflux pump to be responsible for intrinsic resistance of Acinetobacter baumannii to phosphomycin. So we found that the knockout is compromised in phosphomycin efflux pump. So we renamed this gene as ABAF, ABA for Acinetobacter baumannii, F for phosphomycin. So ABAF is involved in phosphomycin efflux. It's also involved in efflux of biofilm matrix and is also involved in virulence. So based on this data, and we know that phosphomycin is a very effective antibiotic with great potential for uncomplicated UTI treatments in all escape pathogen, except it doesn't work in Acinetobacter baumannii because it's intrinsically resistant. And now we know why it is intrinsically resistant. So based on this information, we went ahead and we screened a library of small molecules, about 8,000 molecules, and we picked up 28 non-antibacterial efflux pump inhibitors that were active against another efflux pump. We screened these 28 molecules against ABAF, and we found that this molecule lead series 8367 restores phosphomycin activity below the UCOS breakpoint in more than 90% of clinical A. baumannii strains tested. We tested around 200 clinical strains of Acinetobacter baumannii, which had an overexpression of ABAF. And we went on and characterized the pharmacokinetic properties of this molecule. It was non-toxic in PBMCs, was non-hemolytic, and this was a bona fide, the quinacrine assay shown here showed that this was a bona fide efflux pump inhibitor that acted by affecting the pH gradient. We also tested its virulence in C. elegans model, and it reduces the virulence of A. baumannii in C. elegans. The combination reduces biofilm formation on catheters, and the combination of phosphomycin and A367 reduces the bacterial colonization in urinary tract infection models. So in summary, we were able to, show that A367 sensitizes A. baumannii, resensitizes A. baumannii to phosphomycin by disrupting the efflux pump that was responsible for intrinsic resistance. Now, another small RNA, which we named as ABSR28, seems to repress type 6 secretion system in A. baumannii during oxidative stress. Now, the reason I'm talking about oxidative stress is baumannii faces immense oxidative stress as well as metal starvation in neutrophils, especially when it causes infections. And as we were characterizing the non-coding small RNA, when we made the knockout of this small RNA, it led to altered type 6 secretion system transcripts. And when we looked at these transcripts, it turns out these transcripts were involved in assembly of type 6 secretion system. And the deletion of this non-coding RNA, ABSR28, results in increased type 6 positive population. The wild type cells, they predominantly show type 6 negative population, whereas the deletion knockout mutant results in increase in T6 type population. Then we started looking at what was type 6 secretion system doing in baumannii. So in the literature, it is known that in baumannii, type 6 secretion system induces contact-dependent killing of other microorganisms. So the baumannii cells, they express type 6 secretion system by losing PAB3, a plasmid. The repression for type 6 secretion system is on a plasmid. So when they lose the plasmid, they become type 6 positive. And when they become type 6 positive, it's a contact-dependent killing. There's a needle-like apparatus which goes and punctures other microbes and then kills them. So the killing, actually, the killing comes at the cost of losing this plasmid. So Acinetobacter baumannii type 6 minus cells, the cells that don't express type 6 system, they retain PAB3 and are antibiotic resistant. But A. baumannii cells, they switch to type 6 positive cells at the expense of losing antibiotic resistance. Now, when we look at the evolution of these plasmid in clinical strains, it turns out this plasmid not just hosts the repressor for type 6 secretion system, it also hosts a lot of antibiotic resistance genes. And in the clinic, The earlier isolates, they have resistance to only 2 antibiotics, but the modern clinical isolates, they have a lot of antibiotic resistances, virtually all groups of antibiotics, aminoglycosides, beta-lactams, chloramphenicol, erythromycin, tetracycline, spectro, and streptomycin. So the losing of the plasmid comes at a cost. Now, the role of this type 6 secretion system in the host has not been studied yet. So since our knockout of non-coding RNA was primarily, showed a phenotype in type 6 secretion system, we started looking at it. And it turns out that type 6 positive cells are more sensitive to phagocytic cell-mediated killing. They cannot tolerate oxidative stress, and they cannot tolerate oxidative stress because they accumulate reactive oxygen species. In Bomini, the reactive oxygen, Bomini and other bacteria, the reactive oxygen species are sort of taken care by SOD and CAT. And when we evaluated their levels, the expression of SOD-B and CAT-E are upregulated in type 6 positive cells, although they cannot break down the ROS. So we looked at the metal uptake systems because SOD and CAT metalloenzymes, they use metals that are taken up by these transporters. And to our surprise, one of the cofactor for SOD, manganese, its transporter was significantly downregulated in type 6 positive cells. So type 6 positive cells are defective in manganese uptake and accumulate manganese ions. And that's the reason they cannot break down ROS. So then we made a knockout of the manganese transporter, and turns out the manganese transporter, the deletion of manganese transporter resulted in increase in type 6 phenotype. And when we complement it, the complemented strains are type 6 secretion system negative. So it turns out there is a crosstalk between manganese uptake and type 6 secretion system, which seems to be controlled by a mechanism. So we looked at all the regulators, the known regulators of manganese transporter and other known regulators of type 6 secretion system. Their levels did not change in type 6 positive, as well as the knockout of the transporter. And to our surprise, the manganese deletion, the transporter mutants were predominantly type 6 positive, despite of retaining this plasmid, which shows that the plasmid was not playing any role in regulating this. So we looked at all the candidate small RNAs, and one of the small RNA, ABSR28, was downregulated in the knockout mutant, which indicated that ABSR28 is the regulator of this whole pathway. So coming to the how of it, I'll just summarize these results. So these results show that ABSR28 potentiates RNase E-mediated degradation of a structural gene of type 6 secretion system, TSSM. So disruption of this repression of type 6 secretion system leads to diminished virulence in Acinetobacter baumannii, both in neutrophils as well as in pneumonia murine model. So in summary, here is what we think happens. Under oxidative stress in the host in neutrophils and other phagocytic cells, Acinetobacter baumannii cells upregulate the manganese transporter to increase manganese uptake. The manganese binds to non-coding small RNA, which allows it to complementarity base pair with TSSM, and with the help of RNase E and HFQ, they degrade the type 6 secretion system, and this degradation represses the type 6 secretion system, and these cells evade host-mediated immune response while retaining antibiotic resistance. On the other hand, the baumannii cells that lose this controlled regulation are unable to withstand oxidative stress and hence are cleared by the host. So in summary, we do see that non-coding RNA control pathophysiology and antibiotic resistance. In one case, we've used small molecule libraries to restore the efficacy of fosfomycin in Acinetobacter baumannii. We could take similar approach, do a screening, and maybe inhibiting this whole pathway will result in type 6 positive cells, which will be cleared easily by the host. So understanding the pathophysiology of bacterial pathogens is crucial, seems to be, comprising the Achilles heel of pathogens and can be targeted for drug discovery. With this, I would like to thank my brilliant team, the funding agencies, especially the part of the work that I showed you today was funded by India Alliance DBT Welcome and DBT. And I'm also thankful to other funding agencies, ICMR, DST, SERV, UCOST, and Research Council of Norway for believing in our efforts. And I'm thankful to the resourceful collaborators that we have. And thank you for your attention.
So now we go to the third talk in this session. The speaker is Dr. Neisseria Agrawal, who is a well-known TB researcher in the country. Dr. Agrawal is a professor at THSTI, the host institute, and he completed his PhD in biochemistry from Delhi University under Professor Tyagi, and he received his postdoctoral training from Johns Hopkins University, USA. Dr. Agrawal's extensive research is on mycobacterial pathogenesis. His research aims to understand drug-induced phenotypic tolerance and genetic resistance in MTB. His work also includes identifying small molecule inhibitors against MTB. Dr. Agrawal is a recipient of Arthur M. Danberg Award for Tuberculosis Research at the Center for Tuberculosis Research in Johns Hopkins. He is a member of Society of Biological Chemists India, and he is also in the editorial board of Journal of Global Antimicrobial Resistance. Thank you. Agrawal, please.
So, very thank you for your kind introduction. And I would also like to thank the organizers, Dr. Batosh and his team for inviting me here. So, today's talk, I'm going to introduce a little bit different flavor of the antimicrobial resistance. What we know about is the post-antibiotic effect. And my subject area is the Mycobacterium tuberculosis in the TB infection. So I don't think that I need to introduce the problems which all of us are facing, particularly in Asia, about the tuberculosis. Nonetheless, the recent report from WHO reveals 10 million cases and 1.3 million deaths because of TB. It's considered as the second leading infectious killer only after COVID. Otherwise, it was the top leading killer in the past. And although it is curable, but multi-drug resistant strain causes a major problem. So not only on our health, but also on our economy. There's a report released on the 7th November, which reveals that India accounts for the largest number of cases, which is not very good news for all of us. And despite the fact that we have defined regimen to control the TB infection, particularly those which are the drug-sensitive cases. But as I can show here, the treatment goes for 6 months. So that is the major driver, for causing a lot of problems. As can be seen from this slide, that approximately 500,000 of drug-resistant cases were reported for the TB. So the major challenges which we have is to how to control the increasing load of drug resistance and how to shorten the TB therapy, which we believe is the major driver of the drug resistance. So when we, this is a model experiment which we all of us do in the lab. So when we give the regular drug exposure to the bacterium, we never see killing. Ideally, the killing trend should be like this, but often the growth slows down and what we call the biphasic curve, which is mainly because of the persisters. So we have tried to understand what is the phenotype of these persisters, and often we believe that this also fuels the resistant population. It has also been observed that apart from the drug resistance persistence, the bacterial resilience plays a major role in the emergence of resistance. And what is the resilience? Is the capacity of bacterial system to recover their initial state after certain disturbances. So it's different from resistance. Resistance is the insensitivity to those disturbances, but resilience is to change their profile of recovery. It can be measured either by the speed of the recovery or by the time, by the amount of the disturbance which requires to undergo the resilience. So what we can do in the lab is that if we grow bacteria in the presence of antibiotic, and here we are monitoring the rate of recovery by giving high exposure for a shorter duration. And when we profile them for the appearance of the colonies after drug treatment, we see the appearances, the recovery time is slowing down. And that is precisely what we call is the, post-antibiotic effect, because of the more time they take after the drug exposure to grow on the cell. There are numerous studies which have been performed in the past. I'm just quoting one such study which has shown that the post-antibiotic effect majorly is controlled by the protein synthesis, either the slow degradation of ribosome or the fast synthesis of ribosome. Both basically improves the recovery, as we call the reduced pH. So we all know that bacteria have a very well-articulated proteostasis network, and majorly it aims to protect the function of proteome. It starts from the synthesis of RNA to the level of unfolded protein synthesis, which undergoes folding with the help of chaperones, or if they fail to fold properly, they undergo the degradation so that the load of unfolded protein is taken care of. Our lab works on different proteins which cover the proteostatic network. And in today's talk, I will be talking about a transcription factor, which we, when we started, we did not know its functions, RE1830. So if you look at the homology, it is very well conserved, but the conservation is showing a dichotomy between slow-growing and the fast-growing organisms. And if you look at the database, it is still annotated as the unknown conserved transcription regulatory protein. To our surprise, while we were working on this, we came across this seminal paper in MBio, where it was shown to regulate the cell division of mycobacteria by regulating a protein called YB2, which is a cell division regulator, and they've identified the canonical sequence, which is recognized by this particular transcription regulator. Another paper came, which was another surprise to us, where they've shown, as was published in Science last year, December, where it was observed that this protein is associated with the antibiotic resilience. And they've observed that clinical isolates across the world show a lot of non-synonymous mutations in many genes, including this transcription regulator, which they call the RESR. And the frequency of mutation was much higher in the case of drug-resistant strains compared to those in the drug-sensitive group. They created one of the point mutations were created in the lab-grown strain, and surprisingly, what was observed that this mutation leads to faster recovery after the antibiotic treatment, and that is how it was attributed as a drug resilience. However, nothing was known at that time that at that paper that why this resilience is happening and what is the role of this protein, because this is an essential protein. So we framed our question that whether it is essential, and if yes, then what is the reason of its essentiality? How does it control drug resilience? So far, we have been calling it 1830, but after these two publications, we started calling this RSR-MCDR. And the third objective was can we target this with some small molecule inhibitors and subsequently target the post-antibiotic recovery. So our lab has long ago developed this, CRISPR interference tool, where we have modified the Cas9 into dead Cas9 by mutating two of the nucleus domain. And this was very helpful to us, basically in characterizing genes which are essential, because the genes which are essential will stop growing if we conditionally express the relative, specific guide sequences. So we created a library of the mutant strains in our lab, and then we picked this mutant, which is the RSR-MCDR from this library, and we started characterizing it. So first we did standardize the conditions for its downregulation with the help of inducer, which is the N-hydrotetracycline here. And subsequently, we measured growth, which showed that unlike what we were expecting, the growth is bacteriostatic. It's not decidal. So it's essential, but it's somehow stopping the growth. At that time, again, we were not knowing what is the reason behind it. But then we also went ahead to do the animal infection experiment. And what we found that in the animals, the strains are not showing the static behavior, but rather the CFU is going down. Like in case of lungs, the CFUs were down by 10 to the power 4 fold. And the spleen also approximately more than 100-fold reduction was observed. Not only that, we also found a significant difference in the pathology of the lung, both by the gross lung pathology as well as the tissue sections, suggesting that this gene plays a major role in the bacterial virulence. Subsequently, we performed the RNA-seq-based transcriptomic analysis of the mutant, And what we observed that among many gene set of category of genes, those which are involved in the information pathway were mostly downregulated in the mutant. When we closely looked at them, I've listed them here, all the ribosomal genes were downregulated. So this was the first information which we obtained from this, that this gene is somehow, this regulator is controlling the expression of the ribosomal genes expression in mycobacteria. And this was also found to be corroborated by the metabolomic studies, where we found many metabolites as well as the amino acids getting upregulated in the in the mutant because of their underutilization due to weaker protein synthesis. So FAME at RCB helped us to profile the ribosome of the mutants and the complemented strain. And we found that in the mutant, the translating ribosomes were downregulated by more than 50%. And this was also evident by analyzing the protein synthesis using the puromycin uptake. So what we found that the initiation of translation was severely impaired in the case of mutant compared to the control and the complemented strain. So this was the first observation which gave us a clue about the role of this protein and so-called its essentiality. Further going deeper into the mechanism, we picked this particular operon, which we found to be downregulated, cloned this promoter and checked its expression in the mutant and found it to be downregulated. And when we compared its sequence, we found it showing the partial sequence, recognition sequence to what was reported in that MBio paper, giving us a hint that probably there might be direct regulation. So we took two different probes, one is the full length and one is the truncated one, and did the EMSA kinetics. And what we found that the mutant probes are unable to bind, but there's a very good kinetics of binding which we observed. And at the KD level, we observed that it's the binding kinetics at the 700 nanomolar. So we then searched for the literature, how it is related with the post-antibiotic effect. So we came across this paper where it was found that with the different number of ribosomal operon, this is the E. coli which contains the 7 or RRN operons. So sequentially deleting one by one each of them, they found that it increases the bacterial susceptibility to bacteriostatic drug, and also it affects the bacterial recovery time from the antibiotic treatment. So we thought, okay, let's analyze that post-antibiotic recovery time, and that what we observed that in the case of this mutant, there was a significant defect in the post-antibiotic recovery of the mutant compared to the wild type. Similar was the observation with the RPLN silencing, the same profile which we got. However, we did not find any effect of the YB2 downregulation, which was found to be regulated in another study by this particular regulator. So this is my last slide. So what we have just started doing was that we picked this library of molecules and use this electrophoretic mobility shift assay as an assay to screen the molecules which inhibit or alter its function. So we found 4 different type of profiles. One which D1 is inhibitory, which doesn't let to this protein to bind with the DNA. Where there's many molecules which do not show any effect, molecules which alter the stoichiometry of this binding. And we also came across several molecules which stabilize the binding. So this is a summary of those molecules which affect the binding affinity of the protein with the target sequence. So we are currently, trying to analyze their effect on the post-antibiotic recovery with respect to different drugs. So if I can conclude my today's presentation, I've shown that it controls the post-antibiotic effect in MTB. The conditional silencing of RSR-MCDR leads to static growth in vitro and decreased virulence in vivo. The transient silencing of this protein results in the massive transcription reprogramming, as we observed around 20% of the entire transcripts showing differential regulation. We showed that it regulates RPLN operon and the regulation is direct. And, we have also found that PA is controlled by the RSR-MCDR via the regulation of RPLN operon, but YB2 doesn't seem to play any role in it. And small molecule inhibitors exhibit varying effects on the DNA binding activity of RSR-MCDR. So finally, I would like to acknowledge the 2 talented students in my group. Pramila was the lead researcher in this work, assisted by new entrant, Yunus, and all the collaborators and the funding agencies for providing their funding support. Thank you very much.
Our next speaker is Dr. Marcus Guen. He is from the Argonne Center, University of Chicago, and he's a data scientist and works in the Data Resource Center there. He is a biologist and also has a physics background and works on the bacterial, viral and bacterial genome databases to create a meaning out of this. And his skills are the machine learning, the simulation, and the modeling. And his work is on the future of AMR. His topic is future of AMR, but his work is not only restricted to this, is much more vast there. So this is something very new, and we are very keen to listen to you, Dr. Marcus.
Awesome. Thank you for the nice introduction, and thanks for the organizers for giving me the chance to give this talk. So I'm going to give you a quick talk on predicting AMR phenotypes using conserved genes. So quick, who am I again? Data scientist from Argonne. My past papers mainly revolve around predicting AMR using whole genome sequencing. I've also done collaborations doing virulence prediction, predicting chromosome plasmidophages or classifying them. And yeah, so I'm funded out of the BVBRC, which is a Bioinformatics Resource Center funded by NIAID. And we focus on bacterial and viral genomes. And I'm going to be talking mainly about the AMR database we have here. The AMR AST data that we have composes of over 90,000 genomes and over 107 species across 168 antibiotics. These are all scraped from over 400 publications that are manually scraped and edited and put onto the website. This data collection is still growing, and we have built models using all of this data as well. So the main goal of this was to eventually move from whole genomes to metagenomes, since that's where the world seems to be moving. So with whole genomes, we have a lot of WGS data just available, and even more important, a lot of that is labeled. We have labeled metadata for a lot of this, and that's led to a lot of successful papers. With metagenomes, we have a limited data when it comes to when we're looking at community versus individual level, and the labeling tends to be at different levels and makes it a lot more complicated to run ML on. Additionally, we have the additional limitations of being a metagenome. We've got a smaller feature set available. You're not going to have the entire genome in any given metagenome, and the homology between samples is not going to be the same. If I've got two genomes, A and B, well, A might have these genes, B might have these genes. They're not guaranteed to have the same number of genes, and that makes it difficult to run ML on it. So my approach was to see, well, can we start to walk in that direction of doing AI on ML on metagenomes by using conserved genes or core genes? And this allows us to use a very small percentage of the whole genome. And because it is WGS based, we get all of that labeled metadata I talked about earlier. I can also control the size and see what is the minimum amount of genes that I actually need to make a prediction and how large can we go. And I still see this more as a stepping stone between whole genome sequencing and metagenomes rather than being a direct application of metagenomes. But you could kind of see that can eventually become, you could possibly eventually use this to classify on metagenomic bins. We can force homology between the data set, one of the issues I was talking about earlier, by using protein families. In BVBRC, we have something called Patrick genus-specific families. Prior to BVBRC, it was called Patrick when there was only the bacterial side of stuff. So this is BVBRC's implementation of protein families. Essentially, they're gene sets that share a function, share sequence similarity, and they more or less represent homologous genes. I predicted quite a bit with this data set already, mainly antimicrobial resistance, as well as accessory gene content. So I'm using the core genome to predict what accessory genes this genome might have. Unfortunately, due to time constraints, we'll only be talking about the AMR side of this. Quick overview of the training pipeline. We start here with the BVBRC database and all its metadata. I'm going to grab all the PLFs and count them all up just to see what it looks like for a given species overall. And I want to get the percentage of genomes with a given protein family, the average number of copies of that protein family per genome, the median number, as well as the average and median length of each protein family. From there, we can get to conserved genes, and we're going to define them right here as being in more than 95% of all of the genomes that we're playing with. We have filtered out all of the AMR-related protein families for the purposes of any metagenome may not hand you that particular gene, and we don't want that to kind of bias the accuracy of any of these models. We do require them to be singly occurring. That just makes it a little bit easier to deal with. And we're going to select X PLFs. X could be 25, it could be 50, 100, 250, 500, so on and so forth. And we replicate it 10 times to get rid of sampling bias. From there, we can get the training set. And with the training set, we have an additional filter on that to make it a little bit more stringent. And we require that all the genomes have all of these X PLFs. So if we chose a 25, all the genomes have to have those 25 PLFs. And we want to make sure that these particular protein families are greater than 50% the median length, but less than 200% the median length of that particular protein family. We don't want to have a scenario where we're missing the majority of that protein family, but we also don't want duplicates in that particular genome due to some sampling issues, or not, sorry, sequencing issues. We use KMC as a K-mer counter to get features for each of these genomes. It's sent over to a training matrix, which I'll go over in the next slide. That is sent over to XGBoost to produce models. XGBoost is one version of an ML that you can apply. There are many others. This is a decision tree based one. It's like random forest and bagging. There are many others, as I just mentioned. So the training matrix has one genome and one antibiotic per line. So we get the K-mer counts for that particular genome, followed by the antibiotic. It's a 1 if we're testing on an antibiotic, it's a 0 otherwise. So you can see for the first genome, it's going to be in multiple rows because we're going to test it with antibiotic 1, antibiotic 2, so on and so forth. The label is in the last column here. We'll split that out and actually do the training on that. So that's just an overview of what the matrix looks like. Our data set, we're going to be looking at 4 specific species here. We wanted to make sure that our models worked on multiple species and it wasn't something specific to, let's say, Klebsiella, for example. So we're looking at Klebsiella pneumoniae, Mycobacterium tuberculosis, Salmonella enterica, and Staphylococcus aureus. A lot of this was due to the amount of data that the VVBRC does have for these particular data sets. So on the right side here, you'll see a very large table, and the antibiotic is on your left side with each species having its own column. The numbers in there represent the number of susceptible and resistant genomes that we have at the time of the paper. So for this particular one here, you've got 846 susceptible and 214 resistant genomes for CAP. I don't remember what that actually stands for. So we've got a few variables that we have to go over first when we are doing these models. The first one is selecting how many PLFs or protein families we want to use. And the second one is going to be pertaining to XGBoost, and that's going to be decision tree depth. A deeper decision tree can get you more features being used, but it also has a tendency to eventually overfit. Plus it has the added bonus that you have to spend a lot more time training. But if you have a too shallow tree, you won't get the accuracy and that you want out of the model, and it will overly generalize everything. So the charts to your right here, you've got your macro F1 score on your y-axis, the number of genes that we selected on your x-axis, so 25, 50, 100, 250, and 500. And each line is going to be representative of a different tree depth of 2, 4, 8, 16, and 32. The things to take away here are that if you look at Klebsiella and Salmonella here, starting at around 100 genes, you start to kind of plateau. You don't quite see that with Mycobacterium and Salmonella, sorry, the other one's staph. But we did end up choosing 100 genes just to keep training times down. Additionally, if you look at the lines for 16 and 32, they more or less overlap regardless. So the other thing we did want to look at was how does the length of each gene, or how does the average length of the genes over the data set influence the accuracy? So with this, we didn't choose random genes. Instead, we ordered them by length and selected a set that had an average length of 2,000, average length of 1,000, so on and so forth. And the main takeaway here is that your accuracy increases as you get longer genes, and this kind of makes sense as you've got more k-mers to discriminate with. I also added in the ME, which is going to be a resistant genome labeled as susceptible. You don't want this because it's an error. That stands for major error, and the VME stands for very major error, which is a resistant genome labeled as susceptible. As you can see, in general, the accuracy increases as you get longer genes. The last thing we looked at was building individual models for each antibiotic versus a single model for all antibiotics combined. And the idea behind this is, well, if I've got two antibiotics A and B that are in the same family, so you know there might be some stuff related between them in terms of features, but I don't have a lot of samples for B, but I have more samples for A. The model could in theory use the samples from A, the data that it learns from A to apply to B and improve the accuracies. And what you see here is that it actually does do that overall, as the accuracies do improve with the merge models over the individual models for the most part. Like I said before, we have done a lot of whole genome stuff, and we do want to see what the accuracies look like between those. And this is very, very apparent that you can see major accuracy drops when you go from the partial genome stuff to the whole genome stuff. And this is kind of expected as we have a lot less data. One thing to note is that when you're looking at the very major error and the major errors, although our whole genome models would actually suffice for an FDA device to do AST, our partials do not. We did a bunch of controls here. One of these was to deal with phylogeny, where we rated based off the size of each clade. So we grouped each of the genomes into a clade, and the larger the clade, we negatively rated that particular sample. So the model wouldn't rate a single clade. We want to make sure that the model isn't just learning one clade, and the smaller clades are just being tossed out the window. We don't want that. So we rated by no weight, the clade size, the SR, the susceptibility, resistant distribution within a clade, as well as the size and the SR distribution. And what you can see here is overall the accuracies don't change too much. They do change somewhat, which means that there is some phylogeny being played into this. Knowing that we're playing with core genes, that's kind of expected. Note that the scale here starts at 75%. The other thing we want to make sure is that the model just isn't memorizing the answers, and this is done by just shuffling the labels. And as you can see with the shuffled models, your accuracies sit right around 50%, which is more or less guessing. Feature importance, we looked at the feature importance. Actually, Boost has something built in, and it does feature importance by k-mer. We link those k-mers over to the gene that came from, and we sum up the total feature importance for that particular gene. And we can see that there are hundreds of genes used, and we analyze the top genes per each model. Keep in mind, we didn't look at any of the AMR genes because we had filtered those out, and those are from ARDB, CARD, and NDARO, which is from NCBI. So some of the feature important genes, they mainly consist of virulence factors, which you would kind of expect to be correlated to AMR, biofilm formations, which again would be expected to kind of interact with the antibiotic. We also see some genes that are actually related to resistance in general. So we have this OPCA gene, which is conferring isoniazid resistance, and the RMLH gene, which is methicillin. I am running out of time, so I could go over the limitations and the future work, but I'll probably leave that to discussion if you want to find me after the talk. This conclusion just sums up what I just told you. I'd like to give a special thanks to the BVBRC team, Jim Davis, who is my mentor, the lab members of my group, as well as the NIH and NIAID for funding my research, the DARPA iCentry project, which also helped fund some of this work as well. And NARMS have provided us with a lot of Salmonella data, and Houston Methodist provided us with a lot of Klebsiella data. So that's all I got, and I think we're ready for questions.
This was very rich, at least for the 4 organisms you identified. Very nice secretomes and the basic gene elements. Thank you. So they're now open for discussion.
I had 2 questions, one for Dr. Ramurthy and one for Dr. Rangana. So to Dr. Ramurthy. So is it possible that you have a lot of conjugation going on between the anaerobes and the aerobes, especially in the gut microbiome or the gut area? Is that one of the reasons why you have most of these aerobes becoming MDR and XD? And for Dr. Ranjana, so I was just wondering, what is the kind of natural distribution of the type 6 positive and type 6 negative Acinetobacter population, given that it is causing so much of prevalence? And if there was a subpopulation of the type 6 positives, what is the reason behind why that plasmid is lost?
The plasmid loss or plasmid gain is a stress to the organism. So that's why if it gains, maybe it can survive in the better environment, especially in the antibiotic enriched environment. Suppose the same organism goes to the condition where there is no antibiotic stress, though, so the organism don't need the plasmid or any AMR encoding genes. That's why it's losing. So gain and loss, especially the gain, is always a stress to the organism. So I'm able to answer your question. Use your mic, please.
So the frequency between the anaerobes and aerobes in terms of the conjugation.
Frequency
But
of the
aerobic organism, as I mentioned, say facultative aerobes or facultative anaerobes, especially say for example Salmonella or E. coli. So it survives for a moment, means very brief part of time. So that's why it changes its gene expression, etc. But that's not always stable. So in due course of time, the anaerobic facultative anaerobes dies. So this is a very quick mechanism. So this exchange is not always constant.
You had a question to Dr. Patania also?
Yeah, so that was regarding the type 6.
Your first part of the question was what happens in the natural environment. So natural environment is a relevant, very, very relative term. So if you're talking about soil, or if we talk about when other microbes are not surrounding Acinetobacter baumannii, most of the cells are type 6 negative. But as soon as the other microbes, they start surrounding in soil, they switch. But the switch does come at the cost of losing the antibiotic resistance as well as the plasmid. And also, it's kind of suicidal behavior. The cells that secrete type 6 positive, they themselves also die, right? But in the host, the type 6 negative population is what we see because the type 6 positive cells are cleared off in the host.
As shown by her, the transcriptome plays a role because there is a lot of work going on. See, forget about all the AMR genes. How the transcriptome plays a role. Because a recent study shows there are so many genes responsible for antimicrobial resistance indirectly. Say, for example, type 3 secretion system or type 3 or type 6 secretion system, or commonly known as a biofilm formation. So these are all the mechanisms. The transcriptome plays a good role.
Thank you for the very nice talks. I have two questions, one for Dr. Ranjana and one for Dr. Nishit. The first one for Ranjana. Straight out of the box, why do you think it's magnesium? Why magnesium? Manganese, sorry. Why manganese? Have you looked at it structurally or have you seen with other metals?
Go on, sorry.
If you just you mentioned about the soil. So if you see any particular preference, why manganese is a choice of metal in your case of the RNA?
Yeah, so as far as our experiments go, we've tested with zinc, magnesium, manganese, and iron. So, and we see that, so this is all done in vitro because it's very difficult to do it in vivo. So in vitro, we see that when manganese binds to the small RNA, it changes the structure of the small RNA. This is something that we've seen with ITC, isothermal calorimetry. So the binding is not there. So it's not all divalent. There's a lot of difference between manganese as well as magnesium also. So the EMSA gels that I showed due to the positive time, I didn't go through it, but the binding with the target, the binding of the small RNA with the target mRNA, APSR28 with TSSM, does not happen in the presence of other metal ions. It only happens in the presence of manganese, right? So, and manganese is a cofactor for SOD. So then it makes sense because the whole pathway is correlated.
And I have a follow-up question for Dr. Nishit. So in one of your slides, you talked about the ribosome population, which I guess you see a decrease in 70s population. So did you all, so your outcome, your conclusion is based on the 70s population, or also you look at the dynamics or the ribosome composition? Because in mycobacteria, changing the ribosome composition, protein itself is one of the alternates that happens under stress.
So we did not look at the composition. We primarily took this in the wake of transcriptomic analysis. And if you have noticed, all the ribosomal genes were down. So this is what we were predicting. And that is what precisely we found that, when we profiled the ribosome under the, single, same condition, like we did not change any condition or we did not do the proteomic to find out the composition. There was a significant reduction in the translating ribosome, as well as the 30 and 50s were also downregulated, although they were not apparent because of the high peak of the 70s, but all of them assembled ribosome subunits were downregulated.
Thank you.
Great talks to all the speakers. So Marcus, this is a question for you. Actually, we have a good overlap with the kind of work we are doing. And so I deal with a lot of gene copy numbers instead of k-mers in your case. And but I have noticed is that when we do not exclude a clade or the related genome, the models are much biased. So is it just in case of antibiotic resistance, in your case, that you notice that removing a clade didn't really affect the model accuracies? Or is it that in other cases you have observed that clades do bias the models? And the other part was why k-mers and why not gene copy numbers when you have a lot of prepared data sets already available in terms of the gene copy numbers for all the phenotypes that we want to analyze.
So the first one dealt with how clades will influence the outcome of the model. So if you exclude a clade, you could get you might get better generalizability out of that. You may not. Part of the issue with removing a clade is there's something called the convex hull, which is basically the decision space of your input for the input of your model or what it's been trained off of. And in general, most models cannot predict outside of that convex hull. So if the clade you remove is too distant from everything else, the model's ability to now predict on that particular clade is going to be lost, or it's not going to be guaranteed. So that's one of the things you will see when you remove a clade or put in a clade. Your second question referred to k-mers versus gene counts. So two parts to this. The first part is with the conserved gene stuff. We made sure that each of the conserved genes appeared once. And that means I just have a matrix of all ones, which is not informative of anything. But to the more generalized question of if I applied to a full genome, why would I use genes over k-mers? And one of the things with k-mers is that the model can grab intergenic regions. And I've seen a model do this actually. And what can end up happening is the model might hit an intergenic region that obviously may not be related to AMR. But when you look around that particular k-mer, you'll see 4 or 5 AMR genes flanking that particular k-mer. So the model can use that one k-mer to basically do predictions on 5 or 6 antibiotics at once.
That makes a lot of sense, but that brings me to a follow-up question. So do you also consider the context of the k-mers of the genes which you are targeting, basically? Because then using k-mers actually makes it more useful if you also can add the context of the presence of the k-mers over the length of the genome.
So currently we really aren't. We're just handing the model all the k-mers and letting it try to learn what is related to AMR and what isn't. We could give it to context, and that would probably improve the accuracy or require less deep models, something a little bit simpler to do. It'd make it easier on a model if we did, but part of the question from a data science perspective is, can a machine learning actually just learn it?
Thank you. After this rich session, it gives me great pleasure to thank Dr. Mohanty and Dr. Sahu, the co-chairs, as well as Dr. Patania, Dr. Agarwal, Dr. Ramamurthy, and Marcus for giving these brilliant talks. So thank you very much. Thank you, everyone.
I thank all the session chairs and the speakers for the wonderful session. I now request the session chairs to kindly felicitate the speakers. I request Dr. N.K. Ganguly to kindly felicitate Dr. T. Ramamurthy with a memento. Thank you, sir. Dr. Arvind Sahu to Dr. Ranjana Pathania. Thank you, sir. Dr. Debashish Mohanty to Dr. Nishit Agarwal. Thank you, sir. Dr. N.K. Ganguly to Mr. Marcus Guyen. Thank you, sir. Now, I request Dr. Prasanjit Guchet to kindly come up on the stage to felicitate the session chairs. To Dr. N.K. Ganguly, please. To Dr. Arvind Sahu. To Dr. Arvind Sahu. And to Dr. Debashish Mohanty. Thank you, sir. We shall now break for tea and reconvene for the fourth session by 3:45 p.m. The volunteers may kindly assist the delegates to the area, and I request the poster presenters to be present near their respective posters for the evaluation. I also request everyone to please visit the stalls placed near the entrance to the auditorium. I request the session chairs and speakers to please have a group photo.
Thank you. Thank you. Thank you.
I request everyone to please take their seats in the auditorium for the last session. I request everyone to please take their seats in the auditorium for the last session of the day. I request everyone to please be seated in the auditorium for the last session of the day. I again request everyone to please be seated in the auditorium. We'll be starting the session. The theme for today's fourth and the last session is Chemical and Biotherapeutics for AMR. For this, I request Dr. C.V. Shrikant, Dr. Prasenjit Guchet, and Dr. Ramandeep Singh to chair the session. Sir, I request you to kindly join us on the stage, please. Dr. C.V. Shrikant is a globally renowned biomedical scientist, a great visionary who mentors several students on infectious and autoimmune disorders of the gut. He focuses on the key events that cause inflammation during infections and autoimmune disorders of the gastrointestinal tract. He has also been extensively examining the involvement of post-translational modification pathways in shaping inflammatory processes of the gut. Sir, it's an absolute delight to have you here with us. Dr. Prasenjit Guchet is a well-known Indian virologist at the Regional Center for Biotechnology, who is popularly known for his research on molecular signaling of thrombosis, inflammation, and innate and adaptive immune responses in human health and diseases, and identifying biomarkers and molecular targets to develop potential therapeutics. He has majorly contributed to SARS-CoV-2 research by focusing on molecular signaling of inflammation, thrombosis, and thrombophilia, promoting lung injury and cell death. It is a pleasure to have you here, sir. Dr. Ramandeep Singh is a senior professor at THSTI. He is well known for his studies on the pathogenesis of Mycobacterium tuberculosis and understanding the metabolic pathways that enable M. tuberculosis to survive in stressful conditions. He has also undertaken many vaccine development projects as the principal investigator. He is the recipient of the DBT Wellcome Trust India Alliance Senior Research Fellowship. Thank you, sir, for joining us here. In this session, each talk will be for 15 minutes. After all the speakers have delivered their talks, there will be a 10-minute long question and answer session. I request the audience to kindly reserve their questions until the session is completed. I now request the session chairs to introduce the speakers and call them over to deliver their talks.
Thank you very much. It's actually a pleasure to be here and to be chairing this session. The first speaker of this session is Dr. Sanjay Ram. Dr. Ram is an infectious disease specialist from Worcester, Massachusetts, and a professor at the University of Massachusetts Medical School. He received his MBBS degree from the Seth Medical College in Mumbai. Dr. Ram's lab focuses on complement evasion mechanisms of pathogenic Neisseria, gonorrhoeae, as well as the meningitis strains. His current focus is to use the knowledge gained pertaining to complement evasion mechanisms to design novel immunotherapeutics and vaccines against rapidly emerging multidrug Neisserian pathogens. Parallelly, his research aims to define the basis for the increased gonococcal transmission and burden of infection in women in pre-existing chlamydia co-infection. So without any further delay, I will invite Dr. Ram. The floor is yours.
Thank you. Thank you so much for the kind introduction. And I'd like to thank the organizers for this opportunity. It's really an honor and a privilege to present our work in front of this group. Of course, I know I can't compete with the fact that Virat Kohli is batting, so we might explain the thin audience. But anyway, here are my disclosures. So I think Tatum had set up gonorrhea perfectly, but these are some of the clinical manifestations, and this is primarily a mucosal infection, cervicitis, urethritis, conjunctivitis, and in some rare instances, the bacteria can disseminate through the bloodstream and can cause septic arthritis, tenosynovitis. The reason I put this slide up is just to show the protein manifestations of this disease. And while designing a vaccine or immunotherapeutic, one has to take into consideration how many different sites a drug or a vaccine has to work. Again, this was already alluded to. We've got a really big problem worldwide. The situation in the United States is terrible. The numbers keep increasing. So, and that coupled with the fact that this bug has become resistant to every antibiotic that's been thrown at it, further compounds the problem. So what do we do? To really win a battle, one must know oneself and one's enemy, with wise words by Sun Tzu from The Art of War. So the story I'm going to tell you actually has its origins about 50 odd years ago. And this was actually quite an amazing story and highlights the importance of studying bacteria in their natural habitat. In this experiment, Ward and colleagues took bacteria straight out of male urethral secretions, exposed them to homologous complement, and then looked for decrease in CFU over 60 minutes. And they noticed that every strain was completely resistant to killing by complement. However, a single passage onto routine media, artificial media, rendered many of the same of the strains susceptible to that same source of normal human serum. Took close to 20 years for Harry Smith's group in Birmingham, England, to figure out what was happening. This bacteria was stealing this molecule, cytidine monophospho-N-acetyl-neuraminic acid. It's a mouthful, but it's basically CMP sialic acid, stealing it from the human host and putting it onto its LOS. And it does so via this LOS, or lipooligosaccharide sialyl transferase, which is a highly efficient enzyme. It's about 100-fold more efficient than mammalian sialyl transferases. So the host CMP sialic acid is used, and the sialic acid is put onto the lipooligosaccharide, which is the most abundant molecule in the gonococcal outer membrane. And now, if you add back CMP sialic acid into the growth media, you can then convert a completely serum-sensitive strain to full serum resistance in a dose-dependent manner. This single molecule, sialic acid, does very many things. It renders the bacteria completely resistant to killing by the alternative as well as classical pathways of complement. I'll talk about this a little more. It engages Siglec receptors, which dampen the inflammatory or immune response, and it also renders the bugs resistant to cationic peptides such as LL37. So I promise this is the only slide of the complement cascade I have, and you must be very familiar with the three pathways. The alternative pathway has this peculiar positive feedback loop, which is kept under very, very tight control by a molecule called factor H. So let's take a closer look at this molecule. It's a beautiful molecule. It's like a string of pearls. Each pearl is called a domain. And the first four domains are important for complement inhibition, whereas the rest of the molecules involved in binding with or interactions with host cells, including glycosaminoglycans, and our very own Arvind Sahu has done the seminal work trying to sort of defining the specificity of glycosaminoglycan binding to factor H. Over the years, many pathogens have been discovered to bind to factor H, and they do so via the same mechanisms or the same sort of domains that factor H binds to host cells, namely domain 6 and 7 and 19 and 20. It's a beautiful example of convergent evolution. And I should point out that the sialic acid binding site is here in domain 20. So many years ago, when I was a fellow in Boston University, we discovered that sialation of gonococci resulted in them binding the full-length factor H. And subsequently, we figured out that the regions that bound to sialated gonococci were really the C-terminal domains 18 to 20. Specifically, it's actually domain 20, which contains a sialic acid binding site. So we reasoned that if we were to design an immunotherapeutic where we fuse the bacteria binding region of factor H with FC of an antibody, we could get like a broad spectrum monoclonal antibody because this is sort of a common mechanism used by gonococci as well as other bacteria. However, if we did it without modifying the factor H part, and it ended up binding host cells, we'd end up destroying our own kidneys and red cells. So therefore, we introduced a mutation in domain 19, which abrogated binding to host cells but maintained binding to the gonococcus. And we were quite happy to see that this molecule continued to mediate very good complement-dependent killing of gonococci in a dose-dependent manner. This is the original superbug described in Japan. We went on to the mouse model that was originally developed by Ann Jers's lab, and we showed the topical delivery of this factor HFC given intravaginally. This is a vaginal colonization model in mice, resulted in a rapid clearance of gonococci. One way we'd like to describe the data is using area under the curve, which is a nice measure of overall burden of infection. And we saw that the area under the curve was also significantly decreased with factor HFC treatment. So how does this function in vivo? It's important to determine the mechanism of action. In the next experiment, I'll show you data with a C6 knockout mouse. Here, all the proximal parts of complement are intact, including opsonophagocytosis, because C5a is generated normally. I know it's a busy slide, but the bottom line is that while wild-type mice clear the bacteria, the C6 knockout mice do not clear the bacteria, clearly suggesting that membrane attack complex was very important for clearance of the bugs. We also rendered the FC region through some genetic engineering, complement inactive, and that also failed to clear the bugs. So importantly, the activity of the factor HFC remains intact after spray drying and lyophilization. This is important because now it allows us to use this molecule as part or to formulate it into intravaginal rings, work we're doing with John Moss at Oakcrest Institute. We want to design multi-purpose prevention technologies using intravaginal rings where we combine this with an anti-HIV and a contraceptive so that women can sort of empowers women to take their health into their own hands. So the other little trick we used is to see if we could use analogs of sialic acid, so tweak the human-like sialic acid a bit so that it and sort of trick the bug into taking these analogs up and see if these analogs prevented immune evasion. So this is the sort of chemistry of the human-like sialic acid. So we use, I'll show you some data with legion-aminic acid where the change is sort of the exocyclic part of the sialic acid tweaked a little bit. Turns out the gonococcal silyl transferase is rather promiscuous. Okay, so this is an SDS gel with no sialic acid added. It migrates, the LOS migrates faster with the addition of human-like sialic acid. You can see the retardation suggesting addition of sialic acid. So several analogs are taken up, but some are not. But we were happy to see that legion-aminic acid and KDN were both taken up. So we said, well, what will happen if we deliver these fake sialic acids into the vagina of mice? And we were happy to see that all three resistant antibiotic resistant strains we tested were rapidly cleared. So something else that sialic acid does, the human-like sialic acid, is confer complete resistance to cationic peptides such as LL37. And it was very interesting that these modified sialic acids, though they were incorporated in the LOS, provided no protection against LL37. So in order to find the mechanism of action, we used cationic peptide knockout mice, and we saw that that sialic acid did not, these modified sialic acids did not clear the infection in cationic peptide knockout mice. These forms of sialic acids are highly resistant to sialidases. This is important because bacterial vaginosis-like organisms are very frequently seen with gonococcal infections. It looks like I'm kind of running out of time, but okay. So I'm going to quickly go through a monoclonal antibody that was discovered by Peter Rice and Sunita Gulati at our institute. And this epitope is recognized by this antibody, is expressed by almost every strain in vivo. And importantly, this antibody is bactericidal in a complement-dependent manner against 100% of tested isolates. I'll go through this. It turns out that sort of serendipitously made the observation that this site was also a site for sialic acid, and it limited complement activation on the bacteria. I'll skip that. It turned out to be important in mice. Just 1 minute on the vaccines here. So over the years, we've used this antibody to find a peptide that mimics this LOS structure. And when configured as a multi-antigenic peptide, it elicits an IgG response against LOS, which is bactericidal, and also mediates clearance in the mouse model of gonococcal infection. So in a collaboration with GenMag, I'm going back to the monoclonal now, they had technology to enhance complement activation, and we saw that even a single microgram of this antibody given to mice effectively cleared infection. So it turns out that IgG systemically can be transported very readily into the vagina. We showed that the mechanism of action was, again, membrane attack complex because the treatment was ineffective in C9 knockout mice. And there is plenty of complement in human cervical secretions. This antibody was active in the dual chlamydia gonorrhea co-infection model. Again, an important consideration because many patients have both chlamydia and gonorrhea. And it's important to note that this Bexsero vaccine actually has very limited efficacy in co-infected people. We skipped that. We delivered this via a DNA antibody, and that was effective as well. We've now started, Thomas Shaughnessy in our group has done extensive CDR grafting, back mutations, and designed a humanized antibody to enhance complement activation. She found that the IgG3 version was extremely active and has shown efficacy of both the IgG1 version as well as the IgG3 version in the mouse. The IgG3 version worked at the lowest dose tested, whereas the IgG1 version did not. And we've collaborated with the Anteria Anderson's lab in Oslo, and they've got a set of mutations that enhances the half-life of IgG1. And here you can see doubling of the half-life in mice. And it also enhances complement-dependent cytotoxicity, as shown here. We can see the survival is so. Again, I won't reiterate all the conclusions, but I hope I've convinced you that LOS is a key virulence factor and how important sialic acid is and how we've used our knowledge of how sialic acid modulates the innate immune system to design therapeutics. Of course, nothing's possible without collaborators. It's been a wonderful journey over the years with many, many groups, and I'm very grateful to the NIH for providing us funding. Thank you.
Thank you, Dr. Ram. So next speaker, it's a great pleasure to introduce Dr. Anil Kaul. So Dr. Kaul is currently clinical professor, director, and director of a clinical high complexity clinical laboratory in Oklahoma State University Health Science Center. So he's a senior health advisor. He served as a senior health advisor to the United States Department of States and received extraordinary service award from the United States. And he has 6 patents, several publications, more than 50 publications, and several awards. So without delaying too much, so I just request Dr. Anil Kaul to go through his presentation. Thank you.
Thanks a lot for that introduction, and thanks to the organizers, particularly Dr. Das, for inviting me to be here. And I'm glad I was able to make it. I just landed this morning from US. So I may be a little jet lagged, but I hope I can get through this. Dr. Sanjay, I think this Virat is still playing, so take it easy. Thanks a lot. I'll talk a little bit about, I saw a lot of presentation on gonorrhea as an OBGYN infectious disease persona. We have worked on gonorrhea for a long time, and biggest challenge in US right now is STIs, particularly gonorrhea as well as syphilis. We have a huge issue with it, but we have to face it. Anyway, I'll talk a little bit about what we have done, because we are trying to use some different approaches, out of the box thinking about how to handle the healthcare crisis. I'll just focus a little bit on UTIs. As you know, UTIs are, they account for about 13 million physician and emergency room visits, costing about $3.5 billion in the US. As we know, women have a higher susceptibility, particularly post-menopausal women. There is a hormonal regulation. I don't want to get into that right now. And there is no effective vaccine. And despite antibiotic treatment, about 25% of women will develop at least one episode of UTI. I just want to skip this. UTI, I don't want to get into the clinical side of it. I don't have time. We have urethritis, which is infection of the urethra, then cystitis. I'll focus a little bit on bladder infection. And then we have
one fact is that most of the UTIs are caused by E. coli, and we have been working on E. coli for a long time, and we have identified some receptors and complement regulatory proteins, particularly CD55. We have worked a lot on that, and also on C1q, on gonorrhoeae. So there's a lot of things we can talk later. And some uropathogenic E. coli, particularly the ones which we have, they evade the bladder epithelium and they go inside the bladder cell wall and just stay there. And there's an antibiotic resistance. We have a huge issue with it. I've just digress a little bit right now. We are all focused here on how to face the challenge of antimicrobial resistance, but I think there's a lot which can be done at a policy level. Unfortunately, not much is being done on the policy level, on the antimicrobial prescriptions and everything else. So that's for some other day. Half of all women experience at least one UTI by the age 35. So we have a huge issue with UTIs, asymptomatic bacteriuria resulting in premature births. Again, 27% that has been reported by Mark Martin's group. And the postmenopausal, as I said, we give HRT. We have seen the results are a little better, but again, there's a lot of multiple reasons for that. Right now, antibiotics are the main course of treatment for UTIs, and overuse, as we know, is a challenge. And not only in the human health, but also antibiotics are being rampantly used in the ag industry. That's again a whole different challenge. Actually, more antibiotics are being used in the agriculture, poultry industry. It's huge usage of antibiotics. And of course, they don't need prescriptions. So those are the challenges we face. Some strains of E. coli cause persistent infections and they basically hide inside the bladder wall. And so it's very difficult to reach those sites with antibiotics. So as a viable alternate strategy, we have been looking at nanotechnology. And as luck would have it, we bumped into a few material scientists who were working on nanodiamonds. So initially we thought they take diamonds and crush them and make nanodiamonds, but it doesn't look like that. So compared to traditional antibiotics, nanoparticles can have numerous benefits, such as longevity, absorption, controlled release, distribution, and delivery. So these are all the advantages we have with nanoparticles. So we focus, I'll just give you nanomaterials. I'll just rush through these. These are different classifications. I don't want to get into this. Inorganic, we have organic, composite-based, and carbon-based. We'll just focus on carbon-based. Because nanodiamonds are carbon-based, so we decided to use them. Again, they have different advantages. Number one, they are small size. They are chemically inert core and are internalized by human cells. Nanoscale of nanodiamonds provides large surface to absorb and covalent link to various drugs. And we also, they are being used as carriers of drugs, different drugs, not only for infectious control, but also for in oncology, we're using some of these. Nanodiamonds have generally been considered biocompatible with broad variety of eukaryotic cells. Nanodiamonds can be made in two methods. One is a chemical explosion. Actually, there's an explosion detonation. Use detonators to sort of create these nanoparticles, and also by decomposition of graphite using under high pressure and high temperature. The size of nanoparticles, it can be 4 nanometers to about 100 nanometers. They are biologically inert, surface can be chemically modified. So they are a promising candidate for drug delivery, and they are biocompatible. And even though they have a small size, but they have a large surface area to absorb different drugs. And they can be tested as a candidate for treatment of infectious disease. We hypothesize that nanodiamonds will be taken up by human bladder cells and facilitated in killing intracellular pathogens. That's what the intent was. We tested the hypothesis by evaluating the ability of nanodiamonds to be internalized by the human bladder cells. So we use an in vitro UTI model comparison of T24 human bladder epithelial cells. And we use an invasive E. coli, it's a uropathogenic E. coli, it's a clinical isolate, which was expressing DRADs and which binds to the CD55, which is decay accelerating factor. So we used those, the bacteria and the cell line, and we decided to use nanodiamonds of two particular size, 6 nanometer and 25 nanometer. Initially, we wanted to characterize the nanodiamonds, and we were able to sort of see a peak with a 6. Actually, we got more purification with a 6 nanometer. So we decided to focus more on 6 nanometer. So this purification was done by acid treatment, where we'd use different concentrations of sulfuric acid and nitric acid, 9:1 ratio, and treat them with acid. And then towards the end, we clean it up with sodium hydroxide. We did some experiments with the bladder cell line, and the 6 nanometer nanodiamonds were less toxic. And when we treated the nanodiamonds, when we treated the bladder cell lines with the nanodiamonds of two different sizes, we found that 6 nanometer nanodiamonds were less toxic, and more than 80% of the cells survived, even at the highest dose of 500 micrograms per ml. Whereas in the larger size, there was a little less survival. And this is just a table to just describe our experiments at different concentrations of nanodiamonds and the set of survival. You can see at even at 200 micrograms per microliter, the survival rate was more than 82%. And we used the nanodiamonds 6 nanometer because it was less toxic. So we used 6 nanometer nanodiamonds to kill the extracellular cells. What we did is basically we used these cells and we incubated them with 6 nanometer nanodiamonds. And as a control, we use amoxicillin. You can see that the amoxicillin as a control. So 6 nanometer at 200 concentration, we got almost 50% killing of extracellular cells of the bacteria. Okay, this one, we have those T24 cells were treated with nanodiamonds for 4 hours. And with the TM study, we found that the nanodiamonds were utilized much more than 25 nanometers. So smaller size, obviously they went more inside. Intracellularly, 6 nanometer at 200 concentration, it was significantly killed the internalized bacteria. So in addition to external killing of the bacteria, internally also 6 nanometer was able to kill. So this is just a comparison of the 6 nanometer and 25 nanometer. See that at 200, there was significant killing of the internalized bacteria. Just a confocal, we wanted to see what mechanism these nanodiamonds use to get inside the cell. So they use actin, so we used actin blocker to confirm what was happening. So in this study, what we did is we infected cells with E. coli for 1 hour, and external bacteria was killed by gentamicin for 1 hour, and then we treated the cells with cyto-D, which is anti-actin, for about 1 hour, and treated cells with nanodiamonds for 24 hours. At the end of 24 hours, we realized all the cells with right on 100, and then we were able to inhibit intracellular bacteria. So as you can see that higher concentration of 10 micromolar concentration of Cyto-D, there was a significant reduction in the intracellular bacteria survival, because the nanodiamonds were all able to get in and kill the bacteria. So just to conclude, 6 nanometer acid-treated diamond nanoparticles are relatively non-toxic than 25 nanometer diamond nanoparticles, particularly in T24 bladder cells, which we use. 6 nanometer acid-treated can kill extracellular bacteria, and also are able to kill intracellular bacteria in these particular cell lines. Internalization of nanodiamonds is important for killing and reduction of intracellular bacteria. These findings provide the foundation for developing a 6 nanometer acid-treated nanodiamonds for in vivo therapeutics to treat UTIs. So currently we are working on some projects related to this. We are testing targeted delivery of 6 nanometer nanodiamonds using IV route to deliver nanodiamonds intravesically into the bladder to treat resistant UTI in an in vivo animal model. We basically use C3H-HEJ mice, and we also are assessing antimicrobial effects of nanodiamonds in infected bladder and other major organs. We are performing studies for loading nanodiamonds with antimicrobial agents. We have succeeded in loading nanodiamonds with low dose of amoxicillin. And we are working on seeing if we can deliver amoxicillin intracellularly at a very low dose. And also, we are using these nanodiamonds to coat them with low dose of anti-cancer drugs. So those are side projects which are going on. We're also investigating a therapeutic effect of nanodiamonds in treating UTIs in preclinical in vivo animal model. Well, we are hopeful that the administration of 6 nanometer acid-treated nanodiamonds can serve as a novel therapeutic for treating UTI and make us less dependent on high-dose antibiotics and maybe being away from antibiotics. So this was published recently. Just want to acknowledge some of the team members we have. We have people from the chemical engineering department. We have people from the material science department, and of course the immunology lab group, which I have at Oklahoma State University. Thank you for your attention, and I will be happy to answer any questions later. Thanks.
Thank you. Thank you, Dr. Kaul, for a nice talk.
Our third speaker for the session is Dr. Deepti Jain. She is an associate professor at Regional Center for Biotechnology. She obtained her PhD degree from National Institute of Immunology and postdoctoral training from Rockefeller University. In her lab, she employs different approaches such as structural biologies, biophysical techniques, biochemical methods, and in vivo models to investigate the molecular mechanisms of transcriptional regulation. She is also a recipient of the Innovative Young Biotechnologists Award by the Department of Biotechnology. On behalf of the organizing committee, I would request Deepti to deliver her talk entitled Structure-Based Targeting of Bacterial Biofilms to Overcome Antimicrobial Resistance. Deepti, all yours.
Thank you, Ramandeep. Before I begin, I want to thank Babatosh for inviting me to present our work. So our lab looks at the bacterial biofilms, which are leading cause of antimicrobial resistance. And we largely utilize structural biology-based tools to target biofilms. And our model organism is Pseudomonas aeruginosa. So Pseudomonas aeruginosa is an opportunistic human pathogen, and it is a cause of substantial morbidity, mortality, especially amongst immunocompromised patients, such as those suffering from cystic fibrosis or burn or wounds. And it is listed as a critical pathogen in priority list of antibiotic-resistant bacteria. And its persistence is because it has this ability to form biofilms, which result in chronic and hard to eradicate infections. Biofilms render the organism 1,000 times resistant to antibiotics. This is the electron micrograph showing the wild type organism, which has a single polar flagella. Now, Pseudomonas flagella is very important for establishment of biofilm because it acts like a mechanosensory organ. So it senses the surface, and that is what initiates the irreversible attachment to the surfaces. This is then followed by microcolony, macrocolony formation, where you can see that flagellar genes get downregulated. So organism does not move around, and it is encapsulated in a layer of exopolysaccharide matrix, which makes it very difficult for the antibiotics to penetrate. This is followed by mature biofilm formation and biofilm dispersal, where virulent strains of the bacteria or even aggregates of bacteria are released, which can cause fresh infection. So this is the biofilm life cycle, and the organism becomes 1,000 times resistant to antibiotic because of this protective layer surrounding the colonies, as well as this layer protects the organism from the host immune system. Now, every organism has a very specific composition of this biofilm. In case of Pseudomonas, this is made up of alginate, PEL, and PSL. These are some of the microscopic images showing the existence of all these three different kinds of exopolysaccharides, particularly specific to Pseudomonas aeruginosa. At the molecular level, the regulation of genes which are responsible for production of exopolysaccharide is quite complex. However, one common theme is that the secondary messenger concentration within the bacterial cell increases when the organism decides to switch from the planktonic free-floating flagellated form to the sessile biofilm form. So from this to this transition involves increase in this concentration of cyclic diGMP, which is the secondary messenger within the bacterial cells. When this happens, there is a production of these three exopolysaccharide, PEL, PSL, alginate, and hence the biofilm lifestyle is established. So one of the transcription factors which is very important for this switching between the flagellated and the biofilm form is FLEQ, which is a master transcription regulator of flagellar genes as well as biofilm genes. This FLEQ is, so this is the schematic of a typical bacterial flagella, and the flagellar genes, there are about 50 different genes that need to be expressed for the formation and assembly of this flagella. Then there are another set of genes that need to be expressed for the formation of biofilms, and both these set of genes are regulated by this transcription factor FLEQ. So if you knock out FLEQ, you can see there's several fold down regulation of flagellar genes, as well as there is upregulation of PEL and PSL, the exopolysaccharide genes. So during the planktonic stage of the bacteria, that is when the bacteria is motile, there is FLEQ acts as an activator of flagellar genes and repressor of biofilm genes. However, when cyclic diGMP concentration increases in the cell, it binds to FLEQ and it acts as an activator of exopolysaccharide production. So it's a dual regulator which activates flagellar genes as well as activates biofilm genes in the presence of cyclic diGMP. So we determined the structure of FLEQ in our lab complex to ATP, and ATP binds in the crevice between the two subdomains. This is the alpha beta subdomain and alpha helical subdomain. This is the central ATPase domain of FLEQ to which ATP is bound. And you can see that in crystals, it is organized in the form of hexameric discs. So these are hexamers of FLEQ, each of them bound to one molecule of ATP. Next, we look at the details of interaction of ATP with, at the active site, and we see that there are conserved residues like Walker-A, residues of which form hydrogen bonding interactions with ATP, Walker-B, which is important for hydrolysis. There is sensor 1 and sensor 2 residues, which sense the presence of the nucleotide in the active site. So the nature of sensor 1 and sensor 2 is important because when we mutated the sensor 1 histidine to asparagine, which is what is present in this family of proteins. So this is NTRC family of proteins. We align the sequences of these conserved motifs in all the family members, and we see that FLEQ has a histidine in sensor 1, whereas it is asparagine in most of the other proteins. So we mutated this histidine to asparagine as well as to alanine, and we show that asparagine mutant has 2.5 times increased ATPase activity, whereas alanine mutant shows no ATPase activity. And at the same time, we checked the ability of ATP to bind to these mutants, and we showed that there is no difference in affinity of the ATP. to all these mutants. So ATP can bind to all the three proteins, but it cannot hydrolyze in case of alanine mutant, and it can hydrolyze more efficiently in case of asparagine mutant. So when we complemented the bacteria with these mutants, we saw that for H287N, asparagine mutant behaves just like the wild type as far as the motility is concerned. So this is the motility assay on the soft agar plate. And we see that in case of the knockout of FLEQ, which is complemented with asparagine mutant, the motility is as good as the wild type. Whereas in the case of alanine mutant, there is absolutely, the motility is compromised. So ATPase activity of FLEQ is important for motility, is what we concluded from this experiment. We also tested the ability of these mutants to form biofilms, and we see that both asparagine as well as alanine mutant were defective in biofilm formation. So therefore, we thought that the FLEQ will be a good target for mitigation of biofilms. Simultaneously, crystal structure of cyclic di-GMP bound to FLEQ was also released in the PDB, and we saw that cyclic di-GMP binds to a site which is adjacent to the ATP binding site. So this is the ATP binding site, this is cyclic di-GMP. These two structures are overlaid over here, and this is the magnesium which is important for catalysis. So C-di-GMP binding is important for exopolysaccharide production. And what we showed is that ATP hydrolysis is necessary for flagella formation. So we want to target both these sites so that complete abrogation of biofilm can take place. Interestingly, cyclic di-GMP is also inhibits the ATPase activity of FLEQ. So when cyclic di-GMP is present, ATP cannot bind to FLEQ and it acts as an inhibitor. So FLEQ is a good target because when the cyclic di-GMP concentrations are low in the cell, it affects the flagellar genes and we can target the surface attachment. And when cyclic di-GMP is high in the bacterial cells, it actually regulates the production of exopolysaccharide, a lot of other adhesins, also is responsible for cell aggregation and biofilm dispersal. So we employed structural biology tools in silico screening, virtual screening library for FLEQ using this particular protocol, where we are employing a lot of databases, small molecule databases, which has several compounds. And these compounds are docked into the structure of FLEQ. And the scoring, we are looking at the scoring function in order to shortlist the compounds. This is followed by MD simulation and MMGBSA analysis, followed by lead compound identification. So this is just to show you a bunch of 10 compounds and the kind of scores that we are looking for when we are docking these molecules in the ATP side. Now, about 100 of these molecules we procured from NCI library and we tested for their ability to inhibit biofilms. And we see that 35 of these were causing about 50% reduction in biofilm formation. In the secondary screen, we saw that 18 of them were actually lowering biofilm more than 50%. This was then followed by, so we have purified FLEQ and we tested the ability of these compounds to inhibit the ATPase activity of FLEQ because ATPase activity is important for flagellar gene formation, which we had already shown. Also, it is important for biofilm formation because any alteration in ATPase activity also led to reduction in biofilm. So we see 6 of these compounds were actually able to inhibit the ATPase activity. We calculated the IC50 of these compounds and we show that all of them are actually inhibiting the ATPase activity quite well. And we also carried out concentration-dependent inhibition of biofilms by all these compounds. 5 of them were shortlisted for this because one of them was showing antibacterial activity. So we see that they do show concentration-dependent inhibition. And then we selected 3 different time points to look at the inhibition because by 4 hours, we think that initial attachment has taken place. In 24 hours, micro and macro colony formation occurs. And in 48 hours, we are looking at the mature biofilms. So we see that 2 of these compounds could inhibit biofilm by 50% at least by 24 hours. This was followed by, also we looked at ability of these 2 compounds to eradicate the preformed biofilms. And we see that there is quite a lot of reduction in biofilm formation using confocal microscopy. Now, looking at the mechanism, and if we wanted to see if FLEQ is really the target, then flagella formation should also be compromised. And indeed, both the flagella formation, so there's 40% reduction in flagellar formation as well as motility in the presence of these two compounds. And more recently, we have also determined the structure of the FLEQ bound to one of these lead molecules, the C32. This is the FO minus FC electron density map. And this is the compound sitting in the active site. And you can see that there are a lot of hydrogen bonding interactions. And this is comparison of the ATP bound to FLEQ, whereas this is the compound and it binds very close to the ATP binding site. And also we tested these compounds for their ability to inhibit the biofilms formed by clinical isolates. So we procured about 5 clinical isolates. We generated the resistance profile, and we can see here the CS1, CS4 is resistant to almost all antibiotics. And these two compounds were able to reduce to some extent the biofilm formation by these isolates as well. So the idea now here is to make some modification in these compounds based on the structure data that we have, and also to test the ability of these compounds in presence of the antibiotics on these clinical isolates to see if they can show a better activity. That's all. With that, I want to acknowledge all the members of my lab, the X-ray diffraction facilities, collaborators, funding agencies, and I will be happy to take questions. Thank you.
Thank you, Dr.
So next and last speaker of today's today and this session, it's Mr. Sachin Dubey. And Mr. Dubey is the co-founder of Module Innovation, so one company, and also he serves as a executive director of that company. So Mr. Dubey is a Charming Chris Fellow from University of Oxford, UK. He holds BTech, MTech degree in nanotechnology. So he has been involved with several institutions like NCL as a technology for technology commercialization, NCL Pune, I think. And he has been Chief Operating Officer of Fountaina Optima Publishers. He has been involved as an accelerator programmer in IIM Ahmedabad. So he has a long record of, he has been our twice back-to-back IKMC fellow, and so there are many other things he goes in favor of Mr. Dubey, but without any delay, I just request Mr. Dubey to go for his talk on next generation phenotypic test platform for rapid AST, the role of diagnostic in bending the curve of AMR. So,
thank you to the organizers. And I am sure that because all of you have given your 15 minutes leaving the fantastic cricket match that is going on, I may be able to justify these 15 minutes usable for you. So a little bit about Module Innovations. Module Innovations is a deep tech healthcare startup, and it's primarily headquartered in Pune in India. And we primarily work in the area of infectious disease diagnosis and also in the space of AMR. How we started to work in AMR again is a story that dates back, but today's talk would focus primarily on the next generation phenotypic test platform that we are developing to do a rapid AST. Well, these numbers are something that all of us know, but as somebody growing up in life, seeing the fact that by the year 2050, superbugs would actually kill more people than cancer was actually a startling fact. And something that jolted us and all the founders in the team that came together to think about, can we do something about this space? Now, why diagnostic? Lord Jim O'Neill in 2014 created a report, a very important report to highlight the menace of AMR that is upcoming. He gave 10 recommendations, and of those 10 recommendations, he was asked by journalists that if out of those 10, you had to choose one, what would it be? And he clearly pointed out that I would say diagnostics, and I would put my bet on diagnostics. Why? Well, the development of new antibiotics is, of course, under threat. We know that a lot of pharmas are pulling out because there are no financial incentives. From the year 1940 to 1990, resistance to antibiotics took approximately 2 years. It used to take 2 years, but given the timeline that is needed to develop a new class of antibiotic, which is approximately 9 to 16 years, the rate at which new antibiotics are meeting the fate of having a resistant infection is much, much faster now. So since 1990, resistance to linezolid was happening shortly after its launch. So if this kind of trend continues, even the new antibiotics, which are very less in the development phase, will meet the similar fate. And therefore, rapid diagnostic tests are actually really, really important. So Model Innovations is actually building a device which we very happily call as AST Sense. So all the products that we are developing has sense as the last word. And here AST is very obviously for antimicrobial susceptibility testing. So it's an ultra-rapid detection of antibiotic susceptibility in hours and not days. It packs in some very key features that we've very consciously built in by discussing with a lot of clinicians around the world. It's an ultra-rapid test with time to result of as short as 2 hours. The intention is to keep the test very much affordable because the primary market that we want to build it for is India. It's a connected test, of course, with the results being disseminated to the clinician for quick action. It's a multiplex test. And also, importantly, what we are doing, we are trying to actually test different kinds of antibiotics like the traditional, non-traditionals, and even like phages to be compatible with the system. So once there are new antibiotics which are there in the market, they can be used with this test as a companion diagnostic. So how actually is this test working? It utilizes a very simple principle on paper. The reaction that we very commonly know that glucose with reaction with glucose oxidase produces gluconic acid and hydrogen peroxide. Now we are using this hydrogen peroxide as a catalyst to increase the size of seed nanoparticles, which are 5 nanometer particles that we've built proprietary. And in the presence of chloroauric acid, the size of this grows. So eventually this finally in the assay is read spectrophotometrically. So when there is bacteria, it is incubated with, again, a component that we have built in, which we call as nanoprobe. Nanoprobes, of course, has specific antibodies, class specific, that binds to the organism. They bind, the excess amount of probes is filtered through, and then eventually the addition of chloroauric acid happens. It allows the seed particle to grow, and that is recorded spectrophotometrically, which is depicted in this fashion. So when there is no antibiotic, you see the graph, which is on the top. When the bacteria is sensitive to the antibiotic, there is a dip in the absorption. And when it is resistant, it looks very much similar to the case which is without an antibiotic. So this is how, in a nutshell, in a very simple format, the assay is able to actually interpret the organism's profile. The system also has a component called as thresholds. So thresholds are actually some values which the system has been programmed to understand if there is a new case coming that allows it to confer it is resistant or sensitive. It's because it is actually measuring percentage reduction in the assay. It compares all the assays with the no antibiotic panel, which is already part of the whole assay. And this one value, so if the reduction lies above this value, it is conferred as resistant. If it is below, it is conferred as sensitive. These thresholds so far have been developed by doing assay of around 3,500 patient samples, all done in triplicate. So this is an example of the categorical interpretation the system does. So like, for example, Klebsiella pneumoniae, Eficalis, and Pseudomonas aeruginosa. So as you can see on the bottom side, there is the threshold values. And if we take this case of Klebsiella pneumoniae, then it is sensitive to only one antibiotic, colistin, where the threshold lies, the value lies below the threshold. So the beauty of this test is also that it allows the information of MIC to be interpreted. So this is again an example of how the system interprets MIC. So MIC is basically the lowest concentration of antibiotic at which the fall of the value is below the threshold. So in this case, it's a Klebsiella bacteria, and in case of amikacin, the threshold value is minus 13%. So this at 1 microgram per ml, it falls below minus 13. That's how it interprets the MIC. So this is how the system is built to interpret MICs. Right now, these are several other examples, and it has been now quite robustly tested on clinical samples also. So this is how the system has performed on the samples that were taken clinically from hospitals based out of Pune. As you can see, from the 3,500 patients that we have tested, we have got a categorical agreement of 99.1%, an essential agreement of 93.75, with less than 2% of very major errors, falls very much in accordance with the FDA's approval for AST devices. Now, it was very important for us to select what antibiotics will go into the panel. And because right now the initial focus is the Indian market, we consulted with the Indian agencies. So the antibiotics were selected by consulting a lot of hospitals in India and with ICMR as well. So it has two panels. One is, of course, for gram-positive bacteria. One is for gram-negative bacteria. The gram-positive panel has lesser number of antibiotics, and gram-negative has larger number of antibiotics. We also tested its performance when with leukocytes as well as with glucose. So normally the urine will have a lot of leukocytes that will be coming in. So if there are excess quantities of leukocytes, do they affect the performance of the test? And it clearly indicated that there was no interference on the categorical interpretation of the test. Similarly, we mimicked the glucose concentrations as high as 16 millimolar, which reported to be the highest concentration in a diabetic patient. So the test also did perform well in this case. So while developing the test, we also took a different approach of talking to the stakeholders directly, like the clinicians who would use this test, the diagnostic labs that will use this test, and different kind of people who would be using this test. It spanned different parts of the country. We did our survey as well in US and UK also, but that's not represented here. And I would say that 80% of the respondents said that they would be okay to wait for 2 hours to start the specific antibiotic based on the AST results, which was a positive affirmation. There were still people, because of the high loads of patient in India, it's still a large time, and the clinician would want to give the antibiotic or the treatment right away. But this was still a positive news for us when we compare our device to other devices like Vitek or maybe Phoenix. So yeah, the distribution of respondents included different categories of institutes that include tertiary care in private, tertiary care in public, and also different kinds or different levels of care. It included primary care, tertiary care, and secondary care, and also the span of the professionals who would be using this test also varied. Right. So just to give a glimpse of the kind of commitment module has been in the space of AMR. So I remember, I think we started this program back in 2019, particularly coming from the fact that we really lack different kinds of diagnostic tests. I got the experience of the problem of AMR, not the AMR, but lack of diagnostics. Very recently, I was in the state of Jammu and Kashmir. It's a very cold place. I got a little bit of fever. I talked to my clinician friend and they said, you start an antibiotic. Even I had the confidence that it is a viral infection, but they started antibiotics, so I had to take that. So I was of the opinion that if we had a very rapid test to confirm that we had if it's a viral infection or a bacterial infection, I wouldn't be taking 1,500 milligrams of azithromycin. But yes, Module Innovations is committed in this space. Module is the only company in Asia in the diagnostic space which is funded by Carbex. Carbex is a Boston University-based agency that funds primarily deep tech research in AMR. So out of the 93 companies, Module is the only diagnostic company other than Works and Gangajan, which are into the therapeutics development. And we are also competing in a prize called as Longitude Prize in the UK, which is an £8 million prize, primarily given to tests which work at the point of care with several set of features. Again, just one thing that I want to bring with this short video is that after being in this field for a very short span of time and talking to a lot of stakeholders, what we've realized is public engagement is really, really important. If we do all this research and the normal public pushes the clinician to give an antibiotic right away, they do not have an option. So we are also trying to create some small awareness videos in a very common Manish language. Of course, the audience here would know what it speaks, but I just thought I'll play this to break the monotony of the whole day. Does it have a sound? Well, I can speak what is going on, but it has some background score as well. By the way, that's the last thing.
I still remember meeting that lady in a regional hospital in India, whose child died of a resistant bacterial infection. He could have been saved if a timely diagnosis had been done. Antimicrobial resistance, or AMR, is the next pandemic. In 2019, AMR killed more people than HIV and malaria combined. Antibiotics have transformed modern medicine, but its inappropriate use has led to dangerous bacteria develop deadly resistance. The current methods to diagnose bacterial resistance are time-consuming. We need evidence and personalized antibiotics. For that, we need rapid tests. Module Innovations is building AST-Sense, which is one of the world's fastest phenotypic tests to detect bacterial resistance. AST-Sense is ultra-rapid, affordable, near point of care, and a connected test. Based on our patented technology, we see bacteria grow or die faster than conventional method. We will not let bacteria win the war. Are you with us?
So yeah, I think with this, I will also encourage any kind of feedback and input on how can we disseminate this information to general public, which we feel is also really important, and we must include all the stakeholders together. I would like to end by saying that because bacteria knows no boundary, so should our work be very much collaborative, and let us all come together to solve this problem. Thank you.
So thank you, Mr. Sachin. So now the floor is open for discussion and questions, please. And I request those who are taking the microphone, please take quickly.
Thank you for the very nice talks through the session. I have a specific question for Dr. Deepti. So cyclic di-GMP is more of a secondary signaling molecule as well. And I guess it's not really relevant to your study because you clearly show that it specifically works in your case to one specific target that you talked about. But do you think there are other points that you can also hit specifically by utilizing cyclic di-GMP, which you are probably already following up on, or are points of interest for you for this biofilm formation or inhibiting the sort of the signaling pathways? I mean, I'm saying that we know that it's sort of a signaling molecule, right? Especially in Pseudomonas. And if it's something that you are following upon apart from the one target that you talked about.
Involved in biofilm formation, and FLEQ being the target, we are looking for also whether cyclic diGMP mediated signaling. So what I showed you here is only ATPase inhibition myself, but we are also looking at whether it is these compounds are specifically messing up the signaling, cyclic diGMP mediated signaling for biofilm inhibition.
Thank you. What's your question?
Yeah, hi. Thanks, Sachin. It was really nice talk. My question is whether this test can differentiate different bacteria. Does it detect bacteria or only the AST?
So this is an only AST test. We have a different product which differentiates like what bacteria it is. So that is that is actually a 15-minute test which can be done at any kind of clinical setting. This is more inclined to be placed at diagnostic labs and facilities which are slightly little higher in category.
So the test which you say can differentiate between bacteria, so that can tell you whether it's E. coli or it's Klebsiella or only gram-positive, gram-negative?
It differentiates, like it tells you if it is E. coli, it is Klebsiella, it is Pseudomonas, it is Enterococci. So right now in this test, the primary focus has been for UTI. It is now being expanded to other kind of matrices like blood bacteria and respiratory tract infection based bacteria.
So clinicians are okay with a test which can only tell you the AST, whether bug is sensitive without differentiating. Is that enough?
I think for a clinician to make better judgment, ID and AST are both important. But in this test, because we have a ID test already, which gets coupled with this, so this works for them.
Okay, thank you.
Sachin, I have a question for you. Like right now, how many antibiotics you are using for your test?
So if you see the panel, the gram-negative panel has 16 antibiotics and the gram-negative positive panel has 11. And this has been cut short. We have tested it on much higher numbers, but primarily we do not want to increase the cost of the test. And also these are the initial category of antibiotics which are important for clinicians too.
And may I ask you, what is the cost right now and can we reduce it for further?
So this is yet to come into the market, so I cannot comment. I cannot disclose the cost, but I would say that it would be slightly higher than what culture does, but at par with what Vitec and BD Phoenix.
And how much time it may take? Can you?
Yeah, so this is actually a direct from sample to our test. It takes slightly longer time when if it is Pseudomonas, but primarily for Klebsiella and E. coli, it gives the MIC in as short as 2 hours.
Thank you so much.
Yeah.
Sachin, you said that you want to get comment, am I right? Take care about the reproducibility of nanoparticle. It's very sensitive, all right? It's a gold nanoparticle and you are using hydrogen peroxide. First, you cannot, you cannot tell exactly what is the nanometer. And second thing, if for a little bit longer time, with our experience, it will settle down or it gets different kind of, aggregator. That please take care of that part. Otherwise, your result will be varied.
Yeah.
That part you take care because even you have patented and you will go to the market, it will have different condition. So it will reflect on the result. Second question I have, Dr. Jain. You are working on biofilm. You are working on biofilm and you tested a lot of compounds. Yeah. What is the next step? Because the compound you take from NCI or other places, all are proprietary, am I right?
Yes,
and then
so-
are you entering the drug discovery system or you just want to make the lead or heat out of that and you get to some?
No, so we now have crystal structure of these compounds bound to the target protein, and we can now make specific modification based on the structural data to improve the efficacy of these compounds. So it is not just lead identification. We will make modifications and derivatives of these compounds and then come back and test them. And also another aspect will be to test these compounds in association with antibiotics combination because these are not bactericidal. They are only disrupting the biofilms. So we would need antibiotics in combination with these compounds.
So just to tell you with my experience, because I was working on biofilm, and we have the paper in PNS also international patent, and it is in phase 2 clinical trial. Even if you start today, you need minimum 10 chemists to develop a product, and most of the product which will develop it they are already in this zone. So I think you need a huge amount of force to make this hit to lead, and then animal study and all those kind of things.
Yes, we are aware of this. And we are in collaboration with chemists who are already derivatizing, and the structure that we solved, this compound has been chemically synthesized, not from the screen. So the screen always gives you very little amount. And so we then collaborated with the chemist to synthesize this compound in large quantities. That is how we were able to do all these experiments. And we are aware of all these difficulties, and just because it's difficulty shouldn't be doable is not the.
It should be.
So any more question from the audience?
Yeah, I have one question for Professor Kaul. Professor Kaul here. So I was asking that you have some nanoparticle dimer, so that can kill the intracellular bacterial pathogens. So do you know what is the specific target these nanoparticles recognize and kill the bacteria? Because if you are giving this nanoparticle, if they cross the eukaryotic cell, they could also inhibit the similar function to the host also. In that case, they're going to be toxic, but you're saying this is not toxic. That means they must have some specificity for the bacteria. What is the target for your particles?
I think that's. Can you hear me?
Yes, I can hear you.
Yes, we are working on identifying the target, but just for internalization purpose, we have identified that it's going through the actin sort of structures, goes through in, but specifically what exactly it does to the bacteria, or how does it function, we have not been able to do that as yet. But that is work in progress. I think we should have something, probably I would say next 2 to 3 months, we should have the identified. We are working on it right now. We're also developing some analogs. Once we have that, we'll develop some analogs and see how best we can approach that. Does that answer?
Yes, another question I have for this Sachin. So Sachin, when you are talking that the device you have, so that can do the AST in 2 hours. So have you tested with different types of clinical sample, or does that vary depending on the microbial load in the clinical samples? Because some of the clinical sample like blood microbial load is very low. If you go to the fecal sample, microbial load is very high. Does that basically affect your test time?
So this has been primarily tested on UTIs where the loads are slightly higher. Yeah, I think with blood-based infections where the loads are low, we might need to do a culture to bring the numbers to a point where the changes can be seen fast. So the data that you saw were mostly for UTIs, and in some cases the bacteria were from different matrices also. But yes, I mean, if the numbers are low, we'll have to sort of bring them to one particular quantity. And even I think with the CLSI guideline, it has to be brought to one particular CFU to be correlated with the breakpoints. So we probably will in that case have to bring them to a particular number, and it will need a culture step before that.
Okay, thank you.
So last 2 questions, one from me, one from Ramandeep. So my question to Dr. Ram. So you have been nicely showing that that mutations. So my question is that factor H is to some extent I did work with factor H related protein. So do you think that healthy individuals of that has certain percent of that polymorphism in factor H related proteins? CFH3, CFH. So do you think this polymorphism has any, you have been showing that mutations in that region has correlated with the severity of the disease. So do you think that natural polymorphism of CFH and CFHR has any correlation with the infection?
Yeah, that's a great question. The short answer is we absolutely don't know. And one of the questions is why these FHRs, which are part of they actually don't have complement inhibitory function, but they may modulate binding of factor H to surfaces. We have no idea what the concentration of these different FHRs are at various mucosal surfaces. So in my simplistic view, I believe that these molecules might, at least one reason they might have evolved is to prevent microorganisms from stealing the full length factor H, which has complement inhibitory activities. But I mean, I'm sure there's more functions for these molecules. It's sort of the red queen effect, right? I mean, the bacteria have learned to steal the full length factor H and the host responds by making these FHRs that prevent the bugs from stealing the full length or binding to the full length factor H. I'm not sure I fully answered your question, but yeah.
So my question is for Sachin. So using your system, can you say whether it is MIC 50 or MIC 90 values for the drugs you are testing? You said you can measure MIC values, right? So, but how much growth inhibition you can measure, like MIC 50, MIC 90, something like that?
I think I do not recall exactly that it does both of them. But probably I think it is MIC 50, if I can vaguely recall. Because it has the, so what exactly is happening that we are now programming the system to stop at a particular point. So we have the ability to stop when to take the measurement, when not to. So in that case, I assume that we can still go with MIC 90.
And I had one more question for Deepti. So have you measured the intracellular activity for your compounds, like in a macrophage model or in?
We done experiments on the bacteria, not on the anything with the host.
It's okay. No.
I thank all the session chairs and the speakers for sharing great knowledge. I request the session chairs to kindly felicitate the speakers. I request Dr. C.V. Shrikant to felicitate Dr. Sanjay Ram with a memento. Thank you, sir. Dr. Prasenjit Guchet to Dr. Anil Kaur. Thank you, sir. Dr. Ramandeep Singh to Dr. Deepti Jain. Thank you, sir. And Dr. C.V. Shrikant to Mr. Sachin Dubey. Thank you, sir. I now request Dr. T. Ramamurthy to kindly come up on the stage to felicitate our session chairs. Dr. Bhabatosh Das will be felicitating Dr. C.V. Shrikant. To Dr. Prasenjit Guchet. and to Dr. Ramandeep Singh. Thank you, sir. I request everyone to please have a group photo here, the session chairs and the speakers. We now come to the end of the first day of our Indo-US conference on antimicrobial resistance and human microbiome. We have an announcement for the workshop participants. All the workshop participants are requested to assemble at the MSc Lecture Theatre 1 in the Office of Connectivity building by 8 a.m. tomorrow. I thank the audience for their patient hearing and for making the Q&A sessions interactive. Hope you have enjoyed the sessions. The end of today's session is just the beginning of tomorrow's very interesting and informative sessions on the human microbiome in health and disease. We welcome you for the dinner at 7 p.m. outside the auditorium and hope to see you all at 9:30 a.m. tomorrow for the second day of the conference. We wish you all a very pleasant evening. Thank you all.